Okay. Okay, well, let's get started. Good morning, everybody. I'm Sue Lynch. I'm a professor of medicine here at UCSF and have the pleasure of serving as the director of the Benioff Center for Microbiome Medicine. And I'd like to welcome you all to this, our third in our series of annual Benioff Center for Microbiome Medicine symposia on the microbiome. And before I introduce our co-chairs for today, I just want to make a few brief comments on our center and the incredible team that keeps it ticking over, and some words of gratitude. I specifically want to thank our executive committee, Sergio, Peter, Julie, and Tiffany, who've just been incredibly supportive in driving the direction of the center and all of our efforts. And I also want to thank Joelle and McDad, our trainee ambassadors for the center, who are bringing just an extra aspect of what our trainees need on campus to be successful in the field of microbiome research. I also want to thank the incredible team of people who really keep all of our research ticking over across campus, who lead and contribute to our collab plugins in data analytics, metabolomics, notobiotic animals research, microbial genomics, and microbial culture facility. And I specifically want to call out Chedi Lal Gupta and Rahim Khan, our newest additions to this incredible team of people who just keep the lights on and keep all of the research going with their incredible efforts. And as many of you know, we're incredibly proud of the work that goes on in microbiome research across UCSF. It's really pretty incredible, the breadth and depth of research that our faculty and learners are engaged in. And I'm particularly proud of some of the efforts that we've built over the last couple of years. We're really focused on building research efforts that generate resources, that are not just one-hit wonders with a key question that we move on from, but rather collect samples and data that allow many of our investigators across campus to continue to build on our human-focused research on campus. And I'm incredibly grateful to the team that has brought many of these large-scale initiatives to life, particularly over the last year. We've launched the PRIMO study. This is our study of very early life microbiomes and development in premature babies, a major area that we think we could make a large impact on. And we just, in fact, enrolled our first participants, our first pregnant mothers into this study earlier this year, and the first babies will be born into the study next month. Actually, this month, it's already June. And there's efforts, obviously, in developing microbes and understanding their biochemical capacity to build out a microbial pharmacy, work that's going on in the multiple sclerosis microbiome, and some work that's going on within the center, the microbiome and COVID. And these are just some of the larger initiatives that we have funded and initiated through the center that are making their way and progressing. And then lastly, I'd just like to mention that beyond these kind of larger kind of resource building research initiatives, we've also funded a large number of trainee staff and faculty who are engaged in a number of studies across campus that engage other sister programs, including ImmunoX, the Collaborative Laboratories, or CoLab, and the Baker Computational Health Sciences Institute. So we're really moving microbiome research into this very kind of integrated multi-omic arena, where we really move from population-based studies right down to microbes, molecules, and cellular engagement that dictate host health. And so it is with great pleasure that I get to introduce our incredible two co-chairs who have put together today's symposium, Peter Turnbaugh and Sergio Baronzini. I'm incredibly grateful to them for bringing together an amazing lineup of speakers who are just at the cutting edge of microbiome research. But I also want to acknowledge and mention my gratitude to Kristen, Sarah, and Kaylee, who've just been behind the scenes and doing incredible work to bring this symposium to you today. And so with that, Peter, do you want to take the helm?
Okay, thanks, Sue. Thanks, everybody, for coming. I'm really excited for today's talks. I think we have a really great lineup for you guys. I don't know if can they see me? Or I don't know. Hopefully they can see me on Zoom. Good.
Yeah, you look good, Peter.
Excellent. Thank you, Sergio.
As always.
So yeah, just a little bit of housekeeping stuff. With the exception of the keynote speaker, all of the talks are going to be live over Zoom, followed by a brief Q&A. And so feel free to, if you're on Zoom, feel free to enter your questions in the chat box throughout the talk. And the moderators will call on you after the talk for your questions. And so as Sue mentioned, this is the third symposium that we've done. The first one was really a grab bag of highlights of microbiome research across UCSF. The second one focused on the early life microbiome. And then today we're going to talk about precision medicine, mainly in adults. And so to sort of set the stage for what we're going to be talking about today, I found this quote from President Obama back when he launched the Precision Medicine Initiative in 2015. He said, doctors have always recognized that each patient is unique, and doctors have always tried to tailor their treatments as best they can to individuals. You can match a blood transfusion to a blood type. That was an important discovery. I guess I should be doing an Obama impression. What? I can't do it. What if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of medicine was as simple as taking our temperature? And, I think this really ambitious and inspiring dream has motivated a lot of the work you're going to hear about today. I think there's been tremendous progress in terms of precision medicine, mainly in terms of integrating electronic health records with human genetic information. And Sergio, my co-chair, has really helped pioneer this with the knowledge network that's being put together that incorporates data from the UC health system. But by and large, all these efforts have really ignored the microbiome. They don't incorporate either mechanistic or descriptive information about microbial communities. And so today you're going to hear a series of talks that are trying to address this oversight. And they really take a lot of different approaches. So we tried to capture multi-omics, sort of large-scale human data that you're going to hear about in the keynote, in vitro screens, chemical biology, in silico modeling, and phage biology, among many other approaches to tackle this really complicated and hard question. And so we really hope that you'll enjoy the talks and that this will help us really renew this effort towards integrating the microbiome into precision medicine here at UCSF. And so with that, I want to introduce our pre-recorded talk from Iran. So Iran has a background in computational biology and gene regulation. In 2015, as you probably all know, his lab made a big splash by publishing a paper in Nature suggesting that the microbiome explains the effect of artificial sweeteners on glucose tolerance. Notably, for today's symposium, a year after that, he published a paper showing that you could use microbiome data as well as other types of data to predict the rise in glucose in the blood after a meal. And that used machine learning tools and really has set the sort of foundation for a lot of similar studies that have come out in different disease areas. And it also led to a company that they started called Day 2. So if you're curious, you can go pay Day 2 to sequence your microbiome and tell you what to eat. I think what's really notable about his lab's research is it's really massive scale. So they have huge human cohort studies analyzed across many different ways. They're incredibly creative in terms of their computational approaches that they take to analyze this data and experimental approaches to validate it. And it's obviously really translationally relevant. They focus on things like blood glucose that are clearly relevant to disease. So unfortunately, Iran was traveling today. He's not going to be here live. So I will turn you over to his video, and then I guess we can briefly discuss what we thought of the video afterwards.
Okay, hello everybody, and thank you for the invitation to speak here today. Sorry I could not be there in person, but hopefully this will be an opportunity to tell you what we are working on. So I decided in the talk today to talk more broadly about the projects that we are working on, and which cover, for the most part, microbiome work, but even more broadly, basically using a very deep multi-omic profiling of human cohorts to derive insights, with again, microbiome being a very strong component in it. So as a computer scientist and somebody who's been working in the field of AI for over 20 years now, applied to computational biology, I very much relate to this quote from Benjamin Franklin that an investment in knowledge always pays the best interest. And over the past 20 years, we've really been trying to generate a lot of data and to derive biological insights from it. And one of the key reasons is if you look at what happened in the Human Genome Project. This is a project that started or was started many years ago, but the first draft of the Human Genome was published in the year 2000, a little over 20 years ago. And if you think of what happened, this has really been a transformative project, and we went from knowing just about a few variants for some diseases to knowing now hundreds, sometimes even thousands of variants for essentially every type of disease. So this has really been transformative data that, if you look at the fruits of it over 20 years, it's been tremendous. And I guess these days you can't think of treating cancer without sequencing the tumor to understand what you should be doing. So genetics, though, is just one component, and it does not take into account any environmental factor, any lifestyle effects, which we know are very dominating. And so for this reason, people over the past several years have been looking at other types of data that we can measure, other types of omics, like, for example, RNA sequencing of gene activity from different tissues, of course, the microbiome, metabolites, the immune system, and many other components. But typically, when researchers study these, they study each of these data sets in isolation in kind of a narrow fashion, focusing on each data set together. And we asked what would happen if we were to put all of these different types of measurements on the same people and really obtain a very deep multi-omic profiling of individuals. And so because we believe that any clinical condition, much before there's actually clinical symptoms, there is some molecular signature, then if you were to measure everything, you probably would be able to see and detect that disease is about to occur before clinical symptoms actually happen. So you'd be able, in other words, to predict disease ahead of time. With these markers, you might be able to identify which are correlated and which are causal. So maybe that could lead to novel treatments and to prevention of disease. Because we are measuring things on people, it can help us with personalizing treatments, and ultimately, with this data-driven approach, to be much more certain in the different targets that we go after, and by that significantly lower the cost of drug development, which has really been today just too much time and too much money to bring a new drug into the market. And so based on this reasoning, we initiated a few years ago a project that we call the Human Phenotype Project. It's a project where at the moment it's not the biggest data set, but as far as we know, it's the most deeply and broadly phenotyped data set that exists. And the idea is to build this data set to derive discoveries. We think of this as having three main layers. At the core, there's the data, which we actually call a library, because a library represents knowledge, not just raw data. On top of it, we have a platform that does all of the data cleanups and provides access to the data, all the computational and AI tools that are needed. And then on top of that is what you do with this data. So all of the different applications for many different areas, which I'll demonstrate in this talk. And the outcome of all of this is what I think will lead us to the next generation of precision medicine. So this is not just an idea, this is an actual active project in my lab for over 4 years. We call it the Human Phenotype Project. It's, as I mentioned, perhaps the world's most deeply characterized data set. It has these 3 pillars. And so far we recruited over 8,000 people. It's a longitudinal cohort, so we follow people physically every 2 years, virtually every year, to see what progress they have, whether they maintain their health or whether they deteriorated in some ways. And so in total, we have about 28,000 human years that have been followed. And we do everything in my lab, so all the way from building the website to recruit participants, approaching them, bringing them on site to campus, collecting and measuring all of the data on them, and then processing samples for multiomics. And of course, the core expertise of my group is in the data science and data analysis phase. So I believe that by creating this type of data, this will have many, many different applications, medical applications, pharmaceutical applications, but also educational and social applications. Really, let me tell you now what we are actually measuring. So every participant that comes to the Institute, at the core, there's the data, and what we measure about them is can be divided into 3 main parts. On the right, you have the physiological and clinical assessments that we do. On the left, top left, we have all the molecular testing, and then we have the biobanking of samples that we can use in the future for future assays. So in terms of the physiological testing, when people come in, we take all of their, we have all of their blood tests, historical blood tests. We use ultrasound to look at fat in the liver and to look at the carotids, which gives us some assessment of cardiovascular health. We use the DEXA machine to look at full body composition and bone density, EKG and ABI to look at cardiovascular health. We take high resolution images of the retina that allow us to look at blood vessels. And then we use sensors like continuous glucose monitors that people go home with. So they measure glucose levels for 2 weeks, so we can get a good assessment of the metabolic health of participants. Then we have a device that measures sleep at the grade of a sleeping lab, but it really provides us with several analog continuous signals to assess sleep. And I don't think that sleep quality was ever assessed across a cohort of thousands of individuals coupled with all of these other multi-omic data sets. We have all of the intake of medication that people are using, their dietary assessment, through food logging, hundreds of questionnaires about medical background and physical activity and lifestyle that we take from people, and of course, all of their diagnoses. In terms of the molecular testing, we're doing full genetic sequencing, metagenomic sequencing of the oral microbiome and of the gut microbiome. We use mass spec to look at thousands of different metabolites in the blood. We're doing proteomics from the blood, and we have a novel assay of the immune system whereby we synthesize hundreds of thousands of antigens, and then we identify which of these antigens are recognized by the antibodies in the serum of each participant, which we read off by sequencing. And then we do bulk RNA sequencing to look at gene activity from peripheral blood cells. In terms of biobanking, we biobank live cells, PBMCs. We also biobank serum and urine, and these can be very valuable for future testing. So I mentioned that in this first milestone in the lab, the aim is to get to 10,000 people, but in addition to that, we're also building clinical cohorts, so cohorts where we collaborate with local clinicians here to look at either cohorts of cardiovascular systems, oncology. So we have several oncology cohorts I'll talk about, the cohort of NAFLD, and some interventional cohorts that we're doing in atopic dermatitis. So this is a busy slide, but I don't expect you to read all of the details here, but what I wanted to impress on you is that even though the baseline cohort of people in the age of 40 to 70 is a cohort of healthy people, there's actually a lot of different conditions, a lot of different drugs, abnormal measures, abnormal blood tests that people actually have in this cohort. And so the point is that even across a healthy cohort, there is a lot of variation that we can study. So that was the core, basically the data, the library that we collect about on each participant. And I should mention that all of the tests that I showed you before are tests that we apply to every one of these participants. Then there's the platform that allows access to the data. This, we believe, will generate a lot of interest once we open, and there is a plan to open this data set quite broadly. Because if you look at what happens, for example, in the UK Biobank, a study of 500,000 people for the past 15 years, this study is really accessed by tens of thousands of researchers. There's thousands of active projects, many papers, many citations. This generated a lot of impact, and I believe that the cohort that we are building, because of its depth, is actually very complementary to a cohort like the UK Biobank, and therefore will generate a lot of interest. So finally, now for the core part of the talk, I want to tell you about various applications and insights that we've started to derive from this data, starting from the many different applications that could be worked on, starting from therapeutics. And from therapeutics, I want to focus on work that we've been doing around the microbiome, where I'll show you a novel discovery platform that we started to develop, which we could develop because we have really a very large cohort of close to 10,000 people measured and standardized in the same fashion. So I'll tell you about a discovery pipeline that we've developed that allows us to identify bacterial strains that we think could be beneficial for some human conditions. Once we identify them, because the cohort of individuals is actually a cohort, is our cohort, we can go back to selected individuals, those that harbor the specific strains of interest, get a stool sample from them and try to isolate the relevant bacteria. And once we have that, the idea is to generate a formulation, a consortia of several of these bacteria, and go into clinical trials to test the efficacy of this consortium for the indications that we work on. And the first two indications that we're going to go after are indications for type 2 diabetes and for weight loss. And I want to show you some results that we have on weight loss. So just a few words about the discovery pipeline. So as you know, a lot of the analysis that has been going on in the microbiome field has been at the level of compositional analysis, so which bacteria are there, which species are there, what abundances they have. But with the depth of data, the number of samples that we have, we could take this a step further and analyze not just the species, but really record every read that we get, record its mapping to the genome, record the variation, and then try and associate differences in genetic variation at individual nucleotide positions with the trait of interest, in this case, BMI. So here's an example of one, just to illustrate, just 3 samples from 3 individuals. We have a read here that maps to some location in the genome of one bacteria. And you can see that there is variation in this position highlighted in blue, whereby the individual at the top has an A at this position and a BMI of 31, whereas individuals 2 and 3 have a T at this position and a much lower BMI. So this may constitute an association. Of course, this is just an illustration. In reality, we do this on thousands of participants. And when we do, we actually find a lot of signals. So this is a, this is a Manhattan plot that you're typically used to seeing in human genetics, but where the x-axis are human chromosomes, and the y-axis is the significance of the association of a single nucleotide polymorphisms. But so this is a similar Manhattan plot, except that on the x-axis here, we actually now have a human bacterial, we have gut bacteria. And so the x-axis represents different species, different positions within these species, and every dot represents what I showed you in the illustration, genetic variation in a single position that associates significantly with the trait. So yeah, you can see that we actually identify here many different signals that are very, very significant. But I think that much more impressive than the significance of these associations are actually the sizes of the effects. And so here, it's the same data. Every dot represents a SNP within a bacterial genome. But now the x-axis is the size of the effect. So in other words, how many points of BMI are actually explained by each of these dots. And I think what you can see here is that some of these effects go up to 1 or 2 points of BMI. And these are very, very strong effects. So I think bigger than the strongest effects that are seen even in human genetics. So I think very, very exciting. And not only that, but we can identify specific bacterial strains like the one that's shown here, where if you focus on the middle panel, you will see that there's 2 different strains that we identified that have some genetic variation between them. Let's call them the green and the blue strain of the species. And you can see that those individuals that have the blue strain have on average 2 points lower BMI than those individuals that have the green strain. And 2 points of BMI, again, it's huge, can amount to even 15 pounds of body weight. And so this is a significant association, but now we can also leverage the longitudinal nature of our data to see what happens to these individuals 2 years later. And you can see that those individuals that had the blue strain, the one associated with lower BMI, they don't gain weight after 2 years, whereas individuals that have the green strain actually gain another point of BMI. So this is, of course, by itself not proof of causality, but at least this weight gain after 2 years came after we saw the strain being present, the strain that is associated with higher BMI. So maybe it's suggestive that this can actually be causal for BMI. Ultimately, this will have to be tested in clinical trials, which is where, as I mentioned before, we are gearing towards. Here's another strain that we identified, another bacterial species with 2 strains, again, about a 1 point difference in BMI, and also longitudinally, those individuals harboring the strain associated with lower BMI maintain their weight, whereas individuals with the green strain gain weight in 2 years' time, suggesting that maybe these bacterial strains indeed provide some propensity for obesity. So the idea would be to isolate these blue strains now, make a formulation out of them, and give them to people to see whether they can aid with weight loss. Another thing that we're doing with these bacterial SNPs is to study not just interactions between bacteria and traits of the host, but actually interactions between bacteria and other bacteria. So now we're searching for SNPs that in one bacteria associate with abundance of another bacteria. And the reason we're doing that is because we're hoping by that to actually identify some potential novel antibiotics where you would see that some genetic variation in one species may be responsible for a change in some toxin, some gene being produced that may affect the abundance of other bacteria. And I won't go into the details of this, but here too, we are finding many signals that we're now going to study. So I want to show you one type of work that we've done where to show that with alteration of the gut bacteria, one could actually have therapeutic benefit. And this is in a disease called atopic dermatitis. So atopic dermatitis, or AD, is a skin disease. It's a very prevalent one. It's present in about 20% of children, in about 10% of adults, and people who suffer from it quite a bit. And the question we asked ourselves is what would happen if we intervene in the microbiome, in this case in a very crude way, not the precision approach of identifying and isolating specific bacteria that I mentioned before, but in this case with fecal microbiome transplants. And what we did was a proof of principle study, whereby we took only 9 individuals and we gave them initially placebo capsules for 2 dosings separated every 2 weeks, and then 4 more dosings of actual fecal material from a healthy donor. And we asked what happens to their clinical symptoms. So before I show you the results on the microbiome, I'll show you clinically what happened. So here's a participant after week 4, so after 2 dosings of the placebo. And you can very clearly see the clinical manifestation of disease. And if you look at what happened 4 weeks later, so after 2 dosings of FMT material, fecal material, you can see clearance of clinical symptoms. Here's another participant after week 4, and this is even many weeks after the last dosing, we still see clearance of clinical symptoms. And when our clinical collaborators quantified the effect, they actually saw that in all 9 patients, there was improvement. There was no improvement after the placebo, but then all of them improved by an average of 70% reduction in clinical symptoms. If you want all of the details, we just published this paper last year. Of course, we were then interested in understanding what is going on under the hood in terms of the microbiome. And so what we did was to initially analyze the baseline samples and compare the genetic distance between the genome of each bacteria between samples of different individuals. So this is what's shown here. The y-axis is represents different species of bacteria. The box plot, sorry, in gray represents genetic distances between the genomes of this species across unrelated individuals. And so you can see here that on average, there's maybe a 99% identity, or about 10 to the minus 1% genetic difference across the millions of base pairs of bacteria. So this is the typical distance between unrelated individuals, and the dots here represents comparisons between the genetic distance of the donor and the patient at baseline, so prior to doing any intervention. And of course, we see, as expected, that the dots fall right where the box plots are, because the donor and the patient initially are like any 2 unrelated individuals. After we give the placebo capsules, we shouldn't expect any change, and indeed the dots remain in the same place. But after we actually do provide the fecal material, you can see that that many of the dots shift all the way to the left. This means essentially identity between the genome of the donor and the genome of the patient, indicating that with full confidence there has been a transfer of bacteria and colonization and growth of bacteria from the donor to the patient. When we look at a broad T-SNE view of this, we see that here in ellipses, all of the samples of the same donor, we have 3 donors are clustered together, and they are far away from the square samples of the patients. After we give the placebo, there's really not much movement, no change in the gut bacteria of the patients. But once we start to introduce the FMT, we see that the patient has a major shift in his microbiome, and his microbiome now becomes much more similar to that of his donor, and this happens in virtually all of the patients that we examined. So, so we believe that this FMT is actually the cause of this improvement in clinical symptoms of AD, and we're now in the process of expanding the study to more participants and to also look for additional biomarkers that would tell us what may be the causative changes. And we'd like here too to isolate specific bacteria and be able to make a formulation that hopefully could be a novel therapeutic modality for atopic dermatitis. Okay, so back to the story about this data and what we can extract from it. I want to tell you now about several projects that we have to identify biomarkers for different diseases. And here the idea is that because our cohort of thousands of participants is a healthy cohort, we can leverage it to connect to smaller disease cohorts that we assemble, and then be able to find matched healthy controls for our disease cohort, matched in terms of age, gender, BMI, whatever other parameter you want to match on. And then any analysis that we do that would identify biomarkers that distinguish healthy controls and disease patients would hopefully be much more robust disease biomarkers. So we've done this in the context of several different diseases, and I want to show you those results. Before I show you those results, I'll do a small digression to talk about what we're doing with metabolomics, because we are trying to identify determinants of metabolites in our blood, because that can help us to then shed light on biomarkers for disease that we identify. So, in this part, what we are trying to understand is for each of the thousands of molecules that we identify as circulating in our bloodstream, we're trying to find what are the determinants of it. Is it diet, microbiome, genetics? For each metabolite, of course, it may be different. And the reason that this in itself is important is because we know that many of these metabolites actually have a major impact on health and disease. So by identifying their drivers, we would be able to maybe alter these drivers, change the microbiome, change the diet, for example, in order to alter then the levels of different metabolites, and by that, hopefully improve people's health. So we published a paper a couple of years ago where we've analyzed the many metabolites coupled with all of these other types of data, like bacteria, diet, and genetics. And here you can see some gut bacterial signals. For example, some metabolites that we could predict out of sample with very high accuracy using the microbiome, other metabolites predicted well by diet, and others by genetics. Just to give you kind of a positive control here, you can see that caffeine, for example, is predicted very well by diet, just by the amount of cups of coffee that people report they drink. So this is, of course, expected, but then when we analyze the gut bacteria and analyze the models, we can identify various novel findings, like specific bacteria that underlie certain predictions that may be responsible then for synthesizing or degrading specific metabolites. And so equipped with this information, we can then shed light on our other cohorts. I will mention that those metabolites predicted by the microbiome, we also independently validated in the Twins UK cohort that Tim Spector runs. And so in collaboration with them, we took our top predictions, and we got from the Twins UK only their metagenomes. And using those metagenomes in our models, we made predictions for levels of metabolites, sent those predictions to Tim Spector's group, and had them validate without us even ever seeing the metabolomics data. And all of the top 50 metabolites predicted and derived on the Israeli cohort actually replicated in the Twins UK cohort, so indication for us that these are probably truly robust signals and connections between gut bacteria and metabolites. We also asked whether some of these associations may be causal. And so in order to do that, we demonstrated this in the case of diet because it was the easiest. And so for those associations that we found with sourdough bread, we had some strong positive associations, some strong negative associations, meaning meaning that certain metabolites that seem to be higher in those individuals that consume more sourdough bread, and vice versa. And so we did a simple and proof of concept study where we took 20 participants, we gave them sourdough bread for 1 week, and indeed we saw that metabolites that were predicted as positively associated with sourdough bread, in other words, metabolites that should increase after sourdough bread consumption, indeed they increased, whereas metabolites that were negatively associated did not increase. And what was also nice is that when we gave these same participants white bread, we didn't see a change in these metabolites predicted to be affected by sourdough bread. So this is, at least in this case, validation for the causal nature of these findings. Going back now to biomarkers for several diseases, I'll tell you about here one project that we did and recently published for acute coronary syndrome patients who recently underwent a heart attack. And here we also developed what I believe is a novel personalized medicine approach. So we took, you can take even just a single participant, and you can find matched healthy controls for this participant in our large human phenotype cohort. So for example, if you have a 60-year-old male, you'll find as many 60-year-old males with the same perhaps BMI as our patient. And then we can analyze any layer of data like this metabolomic layer. And you can see, for example, now we have the distribution of what each metabolite should look like metabolite number 1, but what it should look like in the relevant healthy reference set for this one patient. And once we have this distribution, we can see where the levels of this particular metabolite for our patient lies. And by that, extract the deviation that we have for this participant. And we can do this for all of the different metabolites. And by that, derive the deviations of metabolites in this one individual compared to his own reference control, healthy controls. So once we do that and we have the set of metabolic deviations, we can go to the part that I showed you before and ask, are these metabolite deviations driven by dietary factors? Are they driven by gut bacteria, by genetics, by other lifestyle factors? So by that, we can highlight for the metabolite deviations of each person whether these metabolite deviations likely arise from which layer of data, diet, microbiome, genetics, and so on. And when we do that, we find that even though sometimes we have patients that reach the same clinical endpoint, it seems that the reasons may be quite different. So for instance, here we have two 60-year-old males, but they both reached the same clinical endpoint. They both had a heart attack. But when we look at their metabolite deviations, that we see that for one, the deviations seem to be more driven by diet. In fact, some metabolites that seem to be enriched higher based on the diet that this person is eating. Whereas in the other one, it seems that, for example, microbiome components may be responsible for the depletion of some metabolites that are part of the metabolite deviations of this person. So in other words, even though we have patients reaching the same clinical endpoint, it seems that the reason for it may be different, and therefore the reason, the way of treating this participants these patients should probably be different. So this is how this can shed light on this. In another project, we analyzed patients with multiple sclerosis, and I bring this as an example for something that we identified on multiple layers of data. So here's a pathway that eventually produces indolactate. And indolactate is actually, or one of the derivatives, indopropionate, is known to protect neurons from oxidative damage. And what we find here is that the level of this metabolite is actually lower in MS patients, but also one of the precursors for this metabolite is also, which is produced by gut bacteria. We find that the bacteria that produce it are also lower in multiple sclerosis patients. So by having seen the depletion of this pathway on two different layers of data, this gives us more confidence in the robustness of this result, and potentially in that increasing the output of this pathway either by providing directly this metabolite or by increasing the levels of the bacteria that produce this metabolite, we might have a novel therapeutic avenue for MS. Here's in the case of IBD, what I want to impress on you is that we were able to have only for 100 in one IBD patients, we were able to find close to 1,400 healthy control participants with similar age and gender. And so by doing this analysis, one could now look at an analysis not of 100 IBD patients, but actually of 1,500 healthy matched controls and IBD patients, and arrive at a very robust signature, in this case of gut bacteria, where we can identify specific bacteria that seem to be, in this case, higher. E. coli seem to be higher in IBD. But then we also find other broader signatures, like more human reads in IBD patients, maybe indicative of shedding of epithelial cells in this inflammatory disease, and also lower richness of bacteria in IBD patients. And from all of this, we can actually predict with near perfect accuracy using a signature of the microbiome who is an IBD patient and who is not. So in the final segment of the talk, I want to tell you about our efforts in the oncology space. So here's several cohorts that we are building, mostly focusing on the role of microbiome. Some of these cohorts are actually interventional cohorts, like a cohort in colorectal cancer and ovarian cancer, where we are performing FMT, a dietary intervention that we are performing in breast cancer that I'll tell you about, and then other just cross-sectional but also longitudinal profiling that we're performing. And in all cases, I think one of the strengths is the ability to couple the usually smaller cohorts of cancer patients in the hundreds of samples, to couple them to match healthy control and derive insights based on that. You can see where these projects lie in terms of our progress with them. So some of them are still in the planning phase, like a FMT experiment that we want to do in lung cancer, and others are at the FDA stage, and for others we actually already produce data and are in the process of analyzing. So I want to show you, here's a result of a signature that we were able to identify for pancreatic cancer. And here too, we had 86 pancreatic cancer patients, but we were able to match them with close to 400 healthy controls, and from that build a quite robust signatures that give us classification out of sample with an AUC of 0.93 based only on gut bacterial signals. And when we look at the feature attribution using the SHAP framework, we can also pinpoint the specific bacteria that at least are driving the predictions of this model. In another case, in the case of GI types of cancers, we also are able to identify a signature of gut bacteria that again provides quite a robust separation between patients and matched healthy controls. In the case of breast cancer, here's also another example for a gut bacterial signature. You can see specific bacterial species, but also just bacteria that produce, for example, butyrate, a short-chain fatty acid. They seem to be lower in breast cancer patients compared to healthy controls. There's also lower richness of bacteria in breast cancer patients. So this signature then also allows us to provide a fairly accurate prediction or classification of breast cancer patients compared to healthy controls. Okay, and the final part I want to tell you is about something we're excited about dietary interventions that we are performing in breast cancer patients. The reason we are doing this is because in about 75% of breast cancer patients, the patients are HR positive, hormone receptor positive, and then the women receive endocrine therapy. But this therapy actually has a side effect whereby over half of the women gain significant weight following this therapy. And this, of course, high weight has by itself many different health complications, but also it causes some of the women to stop taking their medication. And so for that reason, we wanted to perform a dietary intervention where the idea would be to aim to see if we can avoid this weight gain that results after the use of this endocrine therapy. So in order to do that, we built on previous work that we've done. The first paper published in 2015, where we developed an approach for personalized nutrition. The way we did that was to connect people to continuous glucose monitor, look at their blood glucose responses to meals, and what we identified, the first result that you see here on the left, the first key result was that every person is different. People have very different blood glucose responses when they eat even the same food. You can see here 2 lines of the same color represent the same meal consumed by the same person, but on different days. You can see very robust results, in this case for bread consumption. But you can see that there's a lot of variation across different participants, highlighting the need to personalize our nutrition, at least as far as blood sugar levels go. In this same work, we were also able to develop an algorithm that with very good accuracy predicted blood glucose responses to meals based on microbiome data and other clinical data. And then finally, we were able to perform an intervention where we could take participants who are usually pre-diabetic, like this one here, and normally have many spikes in their blood sugar levels, and be able to, with a diet that we tailor according to the algorithm with exactly the same amount of calories, be able to fully balance their blood sugar levels. So, so this first story we actually published in 2015. And since then, we've been working on a long-term randomized clinical trial that we just published last year on people with prediabetes. The idea was to take 200 prediabetics, randomize them into either a standard of care Mediterranean diet or a diet based on our algorithm. And this is anecdotally showing one participant where we track them using CGMs. You can see that for the first month of profiling, prior to providing the diet, we see many spikes in blood sugar levels, indicating that this person is indeed pre-diabetic. But then in the following month, the 1 month after the intervention, we can see full normalization of blood glucose levels in this participant. So the dietary approach really works. And when we look, this is of course an anecdote of one person, but when we look statistically at the entire intervention, we see that in red here, the Mediterranean diet, the standard of care diet, after 6 months of intervention, was able to lower the blood sugar levels. But oh, I think my presentation just crashed, but I will hopefully revive it. But as you can see, as you could see in those results, the personalized dietary approach was actually working much better in terms of the ability to lower hemoglobin A1C, which is the key parameter that people look at in diabetes and in prediabetes. Let me share my screen again. Yeah, hopefully you can now see this. And we got double the effect on the personalized arm after 6 months, and this effect was maintained in the next 6 months of follow-up, in contrast to the reversal back to baseline levels that we saw in the Mediterranean diet. So this, again, we published last year, and we now apply this to breast cancer patients, where we recruited already over 100 breast cancer patients. The study is still ongoing. And we also did a 6 months intervention here, and we could see, and this was the most exciting result, is that not only do these women now not gain weight, but actually they lose weight on average. There was no significant difference in weight loss between the two arms of the study, the Mediterranean diet and the algorithm diet, but in terms of the blood glucose response, we did see a much lower blood glucose response on the algorithm diet as compared to the Mediterranean diet. With that, I want to go back to the high-level story of the cohort, and we are now, I hope in this talk I convinced you that there's a lot of power in collecting deeply phenotyping human cohorts and deriving many different insights from this. And we're now in the process of seeing how we could scale this up on several different axes. So we'd like to increase the number of people, to increase the robustness of the results, to allow us to look at more rare conditions. We also want to expand the different types of data that we look at. We want to expand the different disease cohorts that we have that are shown here. And we also want to increase the diversity of lifestyle and ethnicities and genetics that we look at, which is why we're looking to now expand this study to additional geographies. As I mentioned, in my lab, we actually have been developing all of these different layers from the ability to build and recruit a cohort to actually profile it, the cohort in the clinic, to extracting the samples and carrying out the assays, and of course to analyzing the data. And I believe that if we expand such a study and go and very deeply now phenotype human individuals, this will really allow us to build the next generation of precision medicine. And so with that, I'd like to thank you all for listening. And this is the team, the wonderful postdocs and students in my lab who have been really driving all of these different projects. And with that, I'd like to thank you for your attention.
Okay, so we have a little bit of time now. I think maybe we'll take a break until 10:30 when we'll start the first session of the day. And so feel free, for those of you that are here, to stick around, have some snacks, and we can talk about what we thought about the keynote. See you back here in half an hour.
Hello. Sorry. So welcome back, and well, thank you for connecting back on Zoom, I guess. So I'm going to introduce our next speaker, the first session this morning. So our first speaker is Dr. Liza Meyer. So Dr. Liza Meyer is a biochemist and infectious biologist by training. She received her PhD from the Institute of Microbiology at ETH Zurich in Switzerland. And she followed by a postdoc in the laboratory of Nassos typus at EMBL in Germany. Since 2019, she has led her independent research group at the University of Hospital in Tübingen, where her research investigates whether drugs can be used to precisely modulate the composition of the human microbiome. The title of her talk is Systematically Dissecting the Interactions Between Drugs and the Human Gut Microbiota. Thank you for being here, Liza.
Okay, good morning. Can you hear me?
Yes, we can hear you.
Perfect. Okay. So thank you very much for the kind introduction. Thank you very much for the possibility to present our work here today on a systematic dissection of the interactions between drugs and the human gut microbiome. So recent years have been really eye-opening in understanding how many interactions there actually take place between commonly used drugs, the microbiome, and the host. So by now it's pretty clear that these interactions are bidirectional. So on the one hand side, drugs can impact the composition of the microbiome, but microbes can also modify drugs. It's not only bidirectional, it's also multifaceted. So it's not only these direct interactions, but also indirect interactions that are actually mediated by the host. So I want to give you a quick overview on all possible scenarios that we know of so far, but then would like to dive deeper into the interactions I'm actually most interested in. Now, in terms of direct interactions, we can have several scenarios. So drugs can, for example, impact bacterial growth or change bacterial physiology, meaning the genes that they express, but also the metabolites that they secrete. They can change the overall community composition. However, microbes can also influence drugs. So for example, drugs can be bioaccumulated by microbes, meaning that they are just intracellularly stored and therefore decrease the bioavailability. They can also be modified, meaning they can be inactivated, activated, or converted into toxic byproducts. So this is pretty clear that bioavailability and drug efficacy can be microbiome dependent. In terms of indirect interactions, you can imagine that you take a drug and that changes overall host physiology. For example, you have an increased stomach pH that then changes the environment and different set of microbes can colonize the body. Also, the gut microbiome influence, for example, gene expression and enzymatic activity, and therefore they can interfere with host drug transport and metabolism. Also, the gut microbiome can modulate immune responses, which then has an influence on checkpoint inhibitor therapy, for example, so in cancer treatment. So this is really now a big overview. However, we are particularly interested in the direct interactions. So the microbiome is a driver for individual differences in drug efficacy and toxicity. So we are particularly interested in the direct interactions between drugs or small molecules in general and the gut microbiome. So this is the focus of my talk here today. So why are we interested in those kind of interactions? So from metagenomic-based cohort studies, it becomes more and more clear that actually medication is the biggest cause for individual variation in gut microbiome composition. So this is now here one cohort, but has been shown for other cohorts as well. So there is for sure a strong influence. And then you can imagine several effects. On the one hand side, antibacterial effects, so that individual members of the gut microbiome are inhibited or killed by certain compounds. But then you can also imagine that certain just can better grow under these conditions, or that by inhibiting other members of the microbiome, this open niches and these freeze niches basically for others to outgrow or to bloom. And that has compositional and functional consequences on the host as well. For the antibiotics, that's pretty clear. The collateral damage has been reported many, many times. We have side effects like associated diarrhea. For the non-antibiotics, that is rather a new insight that can also result in side effects. And for certain drugs, the interactions of these drugs with the gut microbiome can be part of the mode of action. So this interaction is required for the drug to work. Now, the second reason why I'm so interested in is that if you understand the precise underlying mechanisms, the molecular player, you can actually use knowledge to have a targeted modulation, a precise modulation of the gut microbiome. But therefore, we first have to go from these anecdotal examples to a more systematic overview. We want to understand whether effects of drugs on the microbiome are actually really direct and host independent, community mediated, or host mediated. And as I mentioned already, so for a handful of different drugs, we actually know that they have a strong impact on the composition of the microbiome. However, if we want to identify the causal elements, the interactions and the underlying mechanisms, it can be really helpful to also complement these metagenomic approaches with cultivation, so cultivation-based approaches to drug microbiome interactions. And what we usually do is we really start with the simplest possible scenario. So we start with pure cultures and then stepwise increase complexity. So as soon as we've understood the effects in pure culture, we use defined communities, then humanized communities or communities that are derived from fecal samples. We add host cells, then employ gnotobiotic mice models, and in the end compare two large population studies. Now that comes, of course, with some problems. on the one hand side, we have to tackle a variety of different microbes. And on the other hand side, we have to work with fastidious gut microbes. So to overcome those hurdles, we actually use high throughput assays and robotics in anaerobic chambers. So we perform scalable, automatable assays that can easily be scaled up. And we have brought assay designs now running in the lab so that we can ask a variety of different questions in these setups. Now, a few years ago, we actually started this endeavor by and wanted to put together a systematic map of drug microbiome interactions. So we started by putting a representative strain collection of the healthy human gut microbiome. And there we started with 40 strains covering 38 different species of 21 different genera that cover the healthy human gut microbiome by roughly 80% of the average assignable abundance at the genus level and 60% at the species level. And these are mainly type strains, so derived from many different sources. So this is by no means an individualized microbiome and by no means reflecting any strain diversity that might occur in an individualized microbiome. However, it's really useful to get a first overview. And with that collection, we screened a drug library of FDA-approved drugs. That's 1,200 compounds, antibiotics, human-targeted drugs, other non-anti-infectives. And we screened them all at one concentration of 20 micromolar, which in average is reached in the large intestine. And then we performed our high-throughput assay. So that was really a major endeavor. And the results are basically summarized in this map here. So for the antibiotics that we've tested, we find that 77% inhibited at least one strain from our selection. And that's basically what you can see nicely in this color code here, that in most of the cases, all of the 40 species were inhibited. For the human-targeted drugs, we find that 24% inhibited at least one strain from our selection. And also here in the color code, you've seen that's really dark, meaning that only a few species were inhibited by those kind of drugs. Now, for the human-targeted drugs, we were pretty surprised that such a high percentage actually inhibited gut microbes. And we did a lot to convince ourselves that this is also reflected in humans. So on the one hand side, for the few drugs where it was possible, we compared cohort studies and see similar trends. We also looked in the side effect patterns of drugs that had impacts on gut microbes and basically could show that depending on the side effects that those drugs have, it's to a certain degree possible to predict whether a drug also have impact on gut microbes. If we look into the different therapeutic classes, we find that there are effects across all classes, basically. However, they are particularly enriched for the antipsychotics and for the antimetabolites. Now, for the antimetabolites, that is basically, does make sense. Yeah, antimetabolites target conserved pathways between pro and eukaryotic cells. So this is understandable why they also inhibit gut microbes. However, it's more difficult to understand for the antipsychotics. So there the primary targets are lacking in gut microbes, so it's a little bit more difficult to understand what actually happens. However, given the many ways, given the many communication ways between the gut and the brain, it's also conceivable that these kind of drugs actually also act by modifying the gut microbiome composition. So some insight that we also got from that data set is illustrated here. So when you take a look into the different species and count for every species how many antibacterial drugs would inhibit a particular species here on the x-axis, and how many human drugs would inhibit this particular species, you see a nice positive correlation, meaning that species that are more sensitive to antibiotics are also more sensitive to human-targeted drugs. And this is a very general property that is not necessarily explainable whether they are gram-positive or gram-negative bacteria. Now we reasoned if this is actually the case, then if we take E. coli and we knock out an antibiotic-resistant mechanism, it would not only make E. coli more sensitive to antimicrobial drugs, but also would make E. coli more sensitive to human-targeted drugs, meaning it would basically move across the diagonal of the plot. And this is exactly what happens if we take E. coli and knock out an antibiotic efflux pump, TOL-C. It becomes more sensitive to antibiotics and to human-targeted drugs. And this is also actually true for other species, like here for Bacteroides uniformis. If you have an isolate that is more resistant to antibiotics, it's also more resistant to human-targeted drugs. Now, to put this, to go even the next step, to put this further, we actually went ahead and did a chemical genomics approach where we took E. coli and took an E. coli genome-wide overexpression library and asked the questions, which genes of E. coli do we actually have to overexpress to rescue growth of E. coli in the presence of seven different human-targeted drugs? And the results you can basically see here. And the answer is easy. So genes that have been previously associated with antibiotic resistances, if you overexpress those, you will rescue E. coli in the presence of these human-targeted drugs. So there is a clear cross-resistance of antibiotics in human-targeted drugs, and that basically implies a possible risk of requiring an antibiotic resistance by consuming a non-antibiotic drug. Now, this is basically what I wanted to tell you about the human-targeted drugs. The impact on gut commensals, the direct impact, is quite extensive. We think that our results are nicely reflected also in humans, in particular in the side effect pattern these drugs usually show. We see that specific therapeutic classes consistently affect gut microbes, and we see antibiotic-resistant mechanisms that can also protect against human-targeted drugs. Now, for the rest of my talk, I really would like to focus on the antibiotics. So this is a class of drugs that we had in our initial screen, but did not put too much attention in the first round of analysis, however, followed up now in more detail. So this is work that has been started during my time as postdoc in Nasso Stipper's lab, together with a postdoc in his lab, Camille Goemans. Now, the collateral damage of antibacterials on the gut microbiome has long been known. It's more appreciated in the recent past. And it's pretty clear that it has long-term bacterial compositional changes of the microbiome, including the development of allergic, metabolic, immunological, and inflammatory diseases. But despite this increased interest, there is basically limited resolution on their effect on gut commensals. And I brought an example from Europe here. So in Europe, we have this European Committee of Antimicrobial Susceptibility Testing that is basically used in clinics. If you have diagnosed a patient with a certain infection, you would here look up the clinical breakpoints and decide on which antibiotic you would give. However, if you look into gram-positive anaerobes or gram-negative anaerobes, what you will find is that for all of them bind together, there will be one value. I mean, it's even acknowledged, so this group of bacteria includes many genera, but then there is only one clinical breakpoint. So the resolution is not very high so far. However, what we already have tested in our screen is for 144 antibiotics across 40 species, the sensitivity, and we followed up for 33 antibiotics across 17 species and performed MIC testing across basically all the major antibiotic classes. So both datasets are in high concordance, high precision, and high recall. And we then went ahead and compared these MIC values with what is available in publicly available databases like Campbell or UCAST. And for non-pathogenic human-associated microbes, these MICs expanded the current knowledge by 80%. Now, what do we learn from these new MICs on gut commensals? So first of all, there is an effect depending on the class. So if we count here the number of inhibited strains, it's pretty clear that aminoglycoside won't do too much harm. This is actually known because aminoglycosides have to be actively imported in the cell. So that is an energy-dependent process. Energy levels in the anaerobic environment in the gut are low. So this was expected that aminoglycosides won't have a big effect. Now for the fluoroquinolones, we actually see a generation-dependent effect with newer variants having a stronger impact on gut commensals. For the beta-lactam, it's basically all over the place. If we take a closer look and do MIC testing for 19 Bacteroides on 8 beta-lactam antibiotics, and then we cluster according to resistant profiles, we can nicely see here that phylogeny is not recapitulated. So if we test different strains from the same species, they are basically all over the place, but not clustering together. So it's really a strain-specific trait. And that we see for the beta-lactams and the Bacteroides. For macrolides and tetracyclines, they would just inhibit all of the species that we have tested. Now coming back to the beta-lactams, yeah, that makes sense. It's a bactericidal antibiotic. The activity is really strain-specific. So if you selectively kill individual members of your gut microbiome, of course, this will change the overall composition of your microbiome. Now, this is a little bit more difficult to understand for the macrolides and for the tetracyclines. So those are textbook bacteriostatic antibiotics. So they would just inhibit, not necessarily kill the cells. Now, if you inhibit everyone, it's difficult to understand why this would shift the overall composition of the microbiome. However, these are the classes of antibiotics that clearly have a strong effect, because this has been shown in various animal models, but also in various studies on macrolide treatment in early childhood. So we decided to take a closer look, and we went ahead and tested for 2 macrolides, so erythromycin and azithromycin, and 1 tetracycline, so doxycycline, the effects on the gut commensals. And so we had a handful of different commensals, and we tested the survival rate after 5 hours of treatment. And what you can nicely see in this graph here is that in many cases, there is a drop below 0.1%, which is a drop below 3 logs, and this is per definition bactericidal activity. So what we basically found is that these textbook bacteriostatic antibiotics for certain microbes would just act bactericidal and kill those. This is nicely seen also in time-lapse movies. So here for Bacteroides vulgatus, upon erythromycin treatment, you see shape defects, plebbing, shortening, and so on. So also in growth curves, for example, here you see clear lysis effects. So what we think is that this differential bactericidal activity, so for some it's bactericidal, for others it's bacteriostatic, can then explain the large effect in the microbiome composition that those antibiotics actually have. And it matters in particular in a community context. So for here, doxycycline, we had the case that we had 2 E. coli strains. In one case, the action was bactericidal and the other bacteriostatic. The same was true in a community context. And then we basically mimicked treatment. So by exposure to a doxycycline, a washout, another doxycycline treatment, and then regrowth in a rich medium. And what you can basically appreciate here is that in case of this E. coli strain that was actually really killed and not only inhibited, it's basically out-diluted of the community. So the bactericidal activity is particularly detrimental in the community context, where susceptible strains are immediately outcompeted and eliminated. Yeah, now, I mean, there is basically nothing we can do about, because certain situation just requires us to take an antibiotic. So we thought, yeah, is there basically another idea that we can do, anything that we can use to protect our gut microbiome under antibiotic treatment. And we came up with an idea, and that was basically based by a study that was ongoing in the Tippers lab at that time, where it has been shown that drug-drug interactions are very species-specific in bacteria. So we thought it would be cool to use the species specificity to selectively protect, so to have an antagonistic drug interaction that would selectively protect commensals, but the antibacterial activity would still work against the pathogens. And we decided to just give it a try, test whether we can find an antagonistic drug interaction that would protect, in our case, Bacteroides vulgatus or uniformis, so just the most abundant species in the gut, by screening compound libraries that could actually do the job. And it was surprisingly easy to find such antagonistic drug interactions that we call antidotes to protect gut microbes. And for the sake of simplicity, for the remaining slides, I would like to focus on 2 of those antidotes. One is dicumarol, which is an anticoagulant. The other is benzbromarone, which is a uricosuric drug. So what we usually do is we perform checkerboard assays to assess antagonistic drug interactions. So here it's Bacteroides vulgatus, we have erythromycin, and we assess the area under the growth curve. And one is basically perfect growth without perturbation, what you can nicely see here. So here it's orange. At a certain concentration of erythromycin, Bacteroides vulgatus won't grow any longer. However, if we add the antidote, so here benzbromaron or dicumarol, we can actually rescue Bacteroides vulgatus in the presence of the antibiotic. We can not only rescue growth, we can also rescue the percentage of survival cells. And not only vulgatus can be rescued, but also fragilis ovatus, Thetaia omicron, and Prevotella copri. So other bacteroidals as well. However, if we test a pathogen, so in this case, Enterococcus faecalis, we do the same assays. What you can nicely see here is that there is no protection, even like a synergistic effect, both for benzbromaron and also for dicumarol. And this is also true for other pathogens like Staph aureus, Streptococcus pneumoniae, and Coccus faecium. So yeah, it seems to be selective antagonistic interactions for Bacteroides. Now, in the next step, we went ahead and tested synthetic communities. Exactly the same. At a certain concentration, the overall community growth is inhibited, however, can be rescued in the presence of the antidote. Now, if you look into community composition, so this is the composition without perturbation. If there is erythromycin, it's basically only Prevotella copri. And if we add the antidote, we can shift the overall community composition back to a state that is more similar to the original one, but for sure we can rescue some of the Bacteroides. Now, if we have a community that has a pathogen, we can do exactly the same thing. So again, the overall community is inhibited, can be rescued, overall community growth in the presence of benzbromaron. However, if we do selective plating to determine the number of Enterococcus faecalis, we cannot detect it any longer. So it's a clear selective protection of the commensals, but still an inhibition of the pathogen. Now, in the next step, we went ahead and took fecal samples from healthy volunteers and derived ex vivo communities from these individuals. And in the first step, we treated those communities with erythromycin and determined the number of erythromycin-sensitive OTUs in these individuals. And this is basically here the size of the circle. And what you can nicely see that basically in all individuals, we have some OTUs that are affected by erythromycin, except those two, where there were no bacteroidal OTUs, because all of them were resistant against erythromycin. And of course, if they're already resistant, I mean, there is no need and no way to protect already resistant microbes. Now, in the next step, we put the antidote, and this is now basically here the color code, the fill of the circles. And in all cases, we could protect some of the bacteroidals OTU. So to sum up, we could protect 45% of the bacteroidals OTU across all individuals. And to be more specific, it was mainly Bacteroides that we could protect. And this is exactly what we screened for, because yeah, our screen was designed to protect Bacteroides. Now, in the next step, we tested in a chnotobiotic animal model. So this is chnotobiotic mice that are colonized with 12 different murine gut microbes. On top, we colonized with Bacteroides vulgatus. And then we first treated just with erythromycin. And what you can see here is a strong drop in the overall counts of Bacteroides vulgatus. And then there's basically regrowth. And in a very simple experiment, so it's just a one-time treatment with dicumarol or benzbromaron, we could at least partially protect Bacteroides vulgatus from the strong drop, both for for dicumarol and benzbromaron. So also in mice, we can use the antidotes to protect Bacteroides vulgatus from erythromycin treatment. Now, to sum up, I think I've presented you here a high-resolution map of antibiotic effects on gut microbes. Now we are more interested in strain level and personalized antibiotic-resistant patterns. But now we actually know what are the most interesting drugs to look for, and we are digging deeper to increase resolution. I've showed you that macrolides and tetracycline links can actually act bactericidal on gut commensals. And for me, this is really like a good example to also show that whatever we already know from the antibiotic action on pathogens cannot necessarily be one-to-one the same in commensals, but we have to carefully check. And of course, we are interested in the consequences on the decline in diversity. And last but not least, I've showed you the antidotes as a concept to selectively protect gut microbes and antibiotic treatment. And of course, here we would like to better understand the underlying mechanism, because what we screened in our initial screen were drugs, and there is of course no point to put a drug on top of a drug, but we want to have an antidote with no effect on the host site. And of course, antidotes can then also be used for other microbiome-related diseases. And with this, I would like to thank you for your attention. I would like to thank my group, in particular, Cordula Geckeler and Patrick Müller, and that were involved in the project that I've just presented, Nassus Tippers and Camille Goemans. This is where the project started. And I want to thank you for your attention, but also draw your attention to the fact that we have an open postdoc position in the lab. So if you feel like coming to Germany to do more culture-based microbiome research, this is your chance.
Thank you so much for this fantastic talk. We'll start a Q&A, and so I'm going to go across the room for the questions that we have here, and Kristen will take the questions that are on Zoom. So feel free to ask questions in the chat on Zoom, and to, I guess, raise your hand here. Yes.
Thanks so much for the great talk. So you said it was surprisingly easy to find the potential antidotes, and you focus on 2, but I was just wondering if you could talk a little bit more about, so you screened 1,200 compounds, and just kind of how many of those did seem to have some sort of antidotal effect.
So, as I mentioned, in the beginning, this was kind of, how to say, we felt like this was a little bit a crazy idea. And we thought, let's just give it a try, and we screen all the compounds that we had in our library from the previous screen. So basically, we screened drugs on top of drugs. And I mean, looking back, this was basically a stupid approach, because as I mentioned during the talk as well, you don't want to have a drug on top of a drug, but you would like more have something like a nutritional compound, like a polyphenol or something which has no effect on the host. However, when we did screen this 1,200 compounds, I would say we had like 12-ish of compounds that would protect uniformis, and also the same amount of compounds that would protect vulgatus, and only few overlapping compounds. So we had also compounds that would protect against both. And we also had compounds that would protect against different classes of antibiotics. And of course, we followed up with those that had then not only, effects on vulgatus and uniformis, but a little bit the broader ones, that protected overall bacteroidals. So there we could dig a little bit deeper. However, my favorite approach is more understanding the underlying mechanism and have a compound that has no activity on its own, because I think this is the way to go, because in the end, you don't want to have a drug on top of a drug.
We have a question in Zoom.
Lisa, the question is, have any side effects been observed in the mice using dichromerol to protect bacteroidetes?
I mean, this was a one-time treatment. And so we have very strict vets here in Germany. So we were just allowed to use a dosage that was reported before to be tolerated by mice. So we did not specifically look for, but the dosage that we chose was from previous experiments kind of chosen in a way that we would not expect to have a side effect from the antidote.
Another question here, Peter.
Great talk, Lisa, or Liza. I was wondering, so like this distinction between commensals and pathogens, like Faecalis can also, there's strains of Faecalis that are commensals, and there's also strains of Bacteroides that are pathogens. And, you know, if you look at, you know, a pathogenic versus non-pathogenic member of either of those species, does that distinguish the antidote effect?
Yeah, I think there, I mean, excellent question. Yeah, I think we should dig a little bit more into, you know, strain level diversity for all what we are doing. However, we chose the pathogen based on the drug. So basically we look what would be, when would you prescribe a macrolide for what type of infections? And there was Enterococcus faecium stuff also, that things, those pathogens that we have tested. So this was basically not our, okay, this is more pathogenic strain, this is more commensal strain, but more based on when would you give a macrolide.
All right.
And I think you would not give a macrolide for an infection with Bacteroides, would you? Not to my knowledge, but yeah, maybe I'm wrong.
Is that a question for Peter?
I mean, I think there was a reason Peter was asking, not?
I'm sure there is a reason. We're gonna. Yeah, Peter was asking this in context of the understanding the mechanism of how it works on the pathogen and not on the commensal.
On the commensal. So I mean, of course, I mean, if we could unravel the underlying mechanism, and we think this is something related to drug uptake or efflux, then we can be more sure. Yeah.
All right, excellent, thank you. We're gonna now move to the next speaker. Thank you so much, Liza, for this fantastic talk. And okay. And let's see. All right. So our next speaker is Dr. Elizabeth Johnson. Elizabeth Johnson is a molecular biologist and an assistant professor of molecular nutrition at Cornell University. She received her PhD at Princeton University investigating cell cycle transcriptomics and completed postdoctoral training under the mentorship of Ruth Lay before joining Cornell. Dr. Johnson's research focuses on understanding how lipids mediate diet microbiome host interactions with the goal of manipulating these interactions for the benefit of host health. The title of her talk is Dietary Lipids as Modulators of Host Microbiome Interactions. Thank you for joining here, Elizabeth Johnson.
Thank you, Joel, for that kind introduction, and thank you to the organizers for the invitation today. It's following up an amazing talk and what's sure to be another amazing talk after me. I just feel very happy to be here. Let's see, let me get to the beginning of this talk though. Okay, well, so I'm very pleased today to be able to share work that my lab has been doing over the past few years to try to understand how dietary lipids are modulating host microbe interactions. And I think we fit well into drugging the microbiome. I am currently drugging my own infant with human milk lipids. So he might make an appearance here today, and I hope his microbes are doing really well. But why are we focusing on dietary lipids? Well, work from the people, many of the people who are in the symposium and are probably in the room there at UCSF, which I wish we could be there because there's so many amazing scientists where you guys are. But but there has been a lot of evidence and great work showing that the dietary lipids or the composition of lipids in the diet can have a very significant influence on both the composition and function of the gut microbiome. And why this is important is you can think of some of these interactions may lead to the production of microbiota-derived metabolites. And some of these metabolites have been well characterized in their production by the microbiome and subsequently their effect on host tissue. However, there's still a significant set of unknown microbial transformations of the microbiome or made by the microbiome that could have very important effects on the physiology of distal tissues to the colon. So in my lab, one second. Hi, everybody. So in my lab, what we're really focused on is a class of metabolites called sphingolipids, which, as Liza was talking, and they're really interesting because they're well conserved pathways for sphingolipids and signaling in both host and microbe. And so we focused on this set of lipids for a number of reasons, which I will outline here. First of all, sphingolipids have been shown in vitro to promote the fitness of certain commensal microbes, also been shown to be toxic to certain pathogens, and in ex vivo cultures of infant stool, they've been shown to alter microbiota composition. Additionally, multiple groups have shown that the sphingolipid production by the gut microbiome is able to influence immune system conditioning and also modulate inflammation in the colon through interactions or through influencing NKT cells. And during my postdoc in the lay lab, we were able to understand how lipid synthesis by sphingolipid producing bacteria can alter host hepatic lipid metabolism. And so with all of these kind of pressing factors that could really signal that sphingolipids are important in host microbe interactions, especially when it comes to metabolism, when I started my lab, I really just started to think about focusing this in the infant context because sphingolipids are a significant component of breastfed infant diets. They're part of the milk fat globule membrane, and they're not so well known for their caloric contributions to the infant, but potential signaling functions. And so when I was thinking about adults during my postdoc, I was thinking they might not be the most interesting system to think about sphingolipids and host microbe interactions, but infants are getting constant exposure to higher levels or higher concentrations of these lipids through their intake of human milk. However, there's been limited investigation on how human milk's sphingolipids may be influencing microbiome composition and function, which was fascinating to me because there are large differences in sphingolipid composition between infant feeding modes that our group and other groups have measured. Here, when they first started the lab, there's actually this much formula in Target, now not so much, but we were able to go and measure all these, the composition, the sphingolipid composition of all these formulas on our LCMS. So by liquid chromatography mass spectrometry. And what you can see from these chromatograms, which many people already know, is that human milk has a complexity of lipids that are not found in most formulas. And if you specifically look at complex sphingolipids, they're completely lacking from even formulas that are milk fat globule membrane supplemented. And so the thought or the actual implications of if sphingolipids are interacting with the microbiome are quite large in this case where you have infants who are exposed to completely not exposed to some of these important signaling lipids. So that really brought myself in the lab to try to investigate how sphingolipids may be precise modulators of diet microbiome interactions, and then thinking about what those consequences may be for host health. And so to do that, I'll first talk about first talk about methods that we use to try to understand how sphingolipids may be interacting with the microbiome. And with these methods, we were really wanting to ask the question, do exogenous sphingolipids actually interact with the gut microbiome? Do members of the gut microbiome take up any of these sphingolipids? And then later on in the talk, I will talk to you about some work that we've done to try to understand what the consequences may be of the uptake of this class of lipids on host physiology. So to do this, when I started the lab, I had two really talented scientists, Dr. Henry Lee and Minting Lee, who is a senior graduate student in the lab, who were pretty fearless and we're like, okay, let's try this. Let's try to understand what methods we may be able to utilize or need in order to track precisely how the microbiome is interacting with dietary sphingolipids. And so that's just Minting, Henry, and Janine, who is a graduate student in the lab, all like patiently looking at data coming off of our LCMS, looking at these lipids and how they may be in these microbial communities and how they may be transformed by these microbial communities. So our first task was that sphingolipids are quite ubiquitous, both in the host and in the microbiome, and also in the diet. So we needed a way to be able to track sphingolipids that were exclusively coming from the diet. Because as you can imagine, we were not able to tease out the signal of where the origins of sphingolipids were in order to understand how dietary sphingolipids were interacting with the microbiome. So to do this, we're able to take advantage of techniques in bioorthogonal chemistry and use a modified sphingolipid that has a terminal alkyne modification, which allows us to differentiate sphingolipids from the diet or exogenous origin from other sphingolipids that are already in the system that are contributed by the host or the microbes that are themselves. And so with this modified lipid, we're able to detect localization using fluorescence by using click chemistry to click on a fluorophore that will allow us to understand where these dietary inputs are going. And so for this talk, anything that is from the diet, we've clicked with the Alexa Fluor 647, red fluorophore, far red fluorophore. So anything red means that it's coming from the diet in this case. But then we can, not click on a detection agent and use comparative metabolomics in order to understand what actual metabolites the microbiome may be producing from the potential interactions with dietary sphingolipids. And so what this looks like in vivo is we're able to orally introduce these modified sphingolipids into mice, isolate microbiome contents, and then sort these contents after clicking on the fluorophore, where we get a population of non-interactors and then a population of interacting microbes. And from those two separate populations, we can sequence these populations and able to determine which microbes are actually interacting with the diet and which ones are not. And then we can use techniques in mass spectrometry in order to identify the masses that are specific to what these microbes are making based off of this dietary sphingolipid input. And so not surprising, but very exciting to us when we were developing this method and introducing dietary sphingolipid into orally to mice, we were able to determine that there were indeed microbes shown here in red that were able to interact with dietary sphingonine, a particular sphingolipid. And there were also microbes that did not interact. So in here on this confocal image, the blue microbial looking structures are stained with a general DNA stain. So, so then the next question was, what is the identity of these particular microbes? And I was surprised by this, but some others in the lab weren't surprised by this. But the majority of sphingolipid interacting microbes in the system that we're using here were also sphingolipid producing microbes. So of the genus Bacteroides and Prevotella. And so, but there were still interacting microbes that were not known to de novo synthesize sphingolipids. And this was very exciting to us, and wanted to further understand the consequences of these interactions, want to be able to determine what products that the microbiome may be making from dietary sphingolipids. To do this, we took conditions where we would not detect transformations of the modified sphingolipid and compared them to the condition where mice were incubated orally with the modified sphingolipid to understand and to figure out if there are masses that were indicative of the microbial transformation of dietary sphingomine. And long story short, yes, we were able to find a suite of masses and metabolites, corresponding metabolites that were sphingolipids that were being transformed by gut microbes that was dependent on the input, dietary input of the modified lipid, which was exciting to us because not only were microbes taking up dietary sphingolipids, but were also using these lipids in their own metabolism. And so we, in the context of the infant microbiome, we could see that prominent infant microbes of the genera Bacteroides and Bifidobacterium make a suite of sphingolipids that in response to dietary sphingomine, and our future efforts were try to understand really what are the consequences of these interactions for the actual potential of being able to understand what's happening when these dietary sphingolipid inputs are missing in infant nutrition, and also can they be used in order to modulate the metabolism of certain gut microbes. So in order to better understand the consequences of these dietary interactions that are sphingolipid dependent with the microbiome, we use some of the similar techniques in order to trace microbial sphingolipids and whether they were interacting with host tissue, and specifically host tissue involved with metabolic phenotypes. And so we can use similar techniques as in the methodology that I explained to track dietary sphingolipids into the microbiome, where we're able to use a modified lipid in order to detect whether these lipids are transiting from the microbiome to host tissue. And in turn, also use comparative metabolomics to understand what are the metabolites that are actually being transferred from gut microbes to the microbiome, and also if there are any potential host transformations of these microbial lipids that would be of interest to host signaling pathways. And so how we're able to actually tag lipids in sphingolipid producing bacteria, and here our model sphingolipid producing bacteria microbe is B theta iota omicron, and in B theta we're able to introduce B theta into media that contains modified palmitic acid, and this leads to sphingolipids and other lipids, other phospholipids being tagged with this this alkyne tag modification. And here you can see that we're able to detect the production of alkyne modified lipids in B theta. And then we take advantage of genetics in B theta to be able to knock out sphingolipid synthesis in a particular strain. And then we can understand how the actual ability for B theta to produce sphingolipids is important to any observed phenotypes. So from this, our two strains are able to make alkyne modified lipids. However, in the wild type, these are sphingolipids, and take my word for it, we've done the measurements, and I'll show you some of, well, I can show you some of that later, but the majority of the lipids here are sphingolipids, versus in the mutant strain, the majority of these alkyne modified lipids are other phospholipids. And so the thought here is that can we observe phenotypes that are unique in the wild type strain incubated case, and that would be dependent on sphingolipid synthesis by the microbiome. So the experimental setup is we put these mice on a fat-free diet so that there are no lipids that are coming from the diet, so we can control for that. And then we were able to orally introduce the wild type and the mutant sphingolipid producing strain daily for a week, and then measure the effects on host tissue and the microbiome. So the dying question that we're trying to answer, are these tag metabolites from Bacteroides actually taken up by host tissue? And so again, anything in red is a lipid that's derived from sphingolipid producing bacteria or a sphingolipid mutant strain. And here I'm showing you the microbiome contents of these mice that were introduced with these strains with metabolically labeled lipids. And you can see that both in the wild type strain, the fecal contents of both the wild type and the mutant strain have microbes that are producing labeled lipids at equal levels. But what was a surprise is that when we look at transfer to host tissue, so in the colon, that we saw significantly more transfer of these modified lipids in the wild type sphingolipid producing strain versus the mutant, even though there is some transfer in the mutant strain. And we were also able to see that these lipids were also transferred to distal tissue, such as the liver. And so the thought was, can we identify what these lipids are? Because most likely, from what I had learned from all my studies previous to this, that they're being transformed immediately by the host in most cases. And so to do that, we needed to map the sphingolipid metabolome of the wild type strain for all of the tag metabolites. And then we could compare the lipid profile of the wild type strain and the liver lipidome, and see if there were any lipids that were common to the wild type strain and the liver. And this would be indicative of a metabolite that was transferred from B theta all the way to the host tissue. And so indeed, very, very long story short, that we were able to identify a non-canonical, a sphingolipid with a non-canonical amino acid, homoserine, canonically this would be serine, that was able to, that we're able to detect both in wild type bacterial cultures and also in our liver lipidomes. And this was very exciting to us because it shows that sphingolipids are indeed able to reach, to be transferred to host tissue. And we're really interested to understand what the consequences were for host phenotypes. So with this fat-free diet, this induces excess lipid accumulation or hepatic steatosis. And we saw that this was rescued in the case where mice were incubated with the wild type strain, but not with the mutant strain. And so we saw that this was somewhat, by doing gene expression analysis, that this was had to do with both sphingolipid metabolism and beta oxidation. And really, really long story short, we spent a lot of time in the pandemic isolating this particular lipid. And this is Minting, a senior graduate student in the lab, with all the efforts, like months and months of efforts. That's like 200 microliters of what we could do in order to look at the functional effects of this lipid. And what we saw when we incubated liver cells with this lipid is that we're able to see improved respiration in a model of sucrose and fatty acid accumulation in these liver cultures. And so that was giving us evidence that sphingolipid synthesis is important for influencing host phenotypes through multiple mechanisms. As you saw, there's direct lipid interaction and also influences on host gene expression, in addition to multiple other mechanisms that we are still exploring in the lab. And so with that, I want to thank the lab for all of their amazing work on this project, trying to understand how sphingolipids modulate host microbe interactions, and our funders for really supporting this project that was a little bit out there. Like Liza said, some of these, when we started, kind of sounded crazy. I thank you for your attention, and I can take any questions.
Thank you, Elizabeth. So we'll take questions in the room and on Zoom. So feel free to ask questions in the chat and to raise your hand. We have a question, Sue.
That was a really beautiful talk. I was wondering if you've actually looked at the bacteria beyond the kind of sphingolipid production that they produce when they come across these, when they uptake these sphingolipids, and ask more broadly whether these lipids change the transcriptional programming or more broadly the phenotype of the microbial cells themselves.
That is a good question. I have a graduate student in the lab who's really trying to look at this, like expanding exactly what are the consequences for the microbes for these interactions. So we haven't asked the questions that you're saying, but we would absolutely love to. We've actually worked with other lipid metabolites and done a lot more of that profiling. And so we do see, like we've worked with cholesterol and polyunsaturated fatty acids with that kind of method, and done more of that kind of phenotyping in lipid interacting microbes, but not for the sphingolipid ones, but we definitely need to.
We have 2 questions on Zoom.
Hi, Liz. This first question is from Julie Saba, who's one of our sphingolipids researchers at UCSF. She says, fantastic talk. Did you look at the specificity of the type of sphingolipids that could be taken up by the microbial species? Did you also look at the metabolism of the sphingolipids through the degradative pathway as opposed to rebuilding ceramides?
That's a really great question. The answer is not yet. So I have I mean, we're trying to synthesize more modified sphingolipid structures, and that will allow us to answer that question because as you can imagine that there are multiple forms, just even in human milk, of sphingolipids that are interacting. We've been doing some work to really understand some work that just recently got accepted in JLR to really actually suss out some of these genes in B theta or in Bacteroides, so we can ask more of the questions about kind of the recycling pathway and other ways that microbes may be modifying sphingolipids in order to also get at part of your question. But the answer really is like, if you ask me in 6 months, I will have an answer.
And the next question is from Holly, also at UCSS. Sphingolipid biosynthesis has been shown to be critical for the development of CD4 plus T cells. Do you have any information on how the sphingolipid producing bacteria might be modulating the immune system?
Yeah, that's a great question. Like, I think that not our group, but other groups at the Broad and Harvard have been looking at this, at how sphingolipids may be modulating, how actually unique structures of sphingolipids, unique to the microbiome, may be interacting with NKT cells. I don't know anything about this CD4 profiles in these actual, I don't remember them in those actual papers, but the thought is that there is, some of these sphingolipids look like agonists or antagonists of these NKT cells. So there's a real well-defined thought pathway of how they may be interacting with the immune system. Yeah, I would love to explore that. I'm not an immunologist, but whenever I get the attention of one, I always like to try to ask them how we can ask about that also in, other tissues that we're interested, that are more directly related to metabolism. So yeah, thank you.
Oh, you have the attention of multiple immunologists that are interested in
Oh,
that.
really? Okay, well, I need to get over there, or you guys can come here to Ithaca, because I'm ready.
All right. Is there any other questions in the room? All right, so we're going to wrap up here. Thank you so much for this great talk. And yeah, we'll definitely follow up with some interests in common. All right, so our next speaker for this session, and this is going to be the last talk for this session, and then there's going to be a lunch break. So our next speaker is Dr. Sloan Devlin. So Dr. Devlin is an assistant professor for the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. Her research interests are in synthetic organic chemistry, microbiology, biochemistry, and analytic chemistry. She heads the Devlin Lab, where she studies the human microbiome using chemical biology with the mission to control the chemistry of human-associated bacteria in order to understand how the microbiome affects human health and disease. She received her PhD from Stanford University, so close by. And the title of her talk is Bacterial Bile Acids Metabolites Affect Host Physiology. Thank you so much for joining, Dr. Sloan, and take it away.
Great, thank you. Can everybody hear me? Or if you can't hear me, someone send me a chat. Can you see my slides okay as well?
Yes, that looks great.
Okay, great. So thank you so much. I wanted to thank the organizers for inviting me. Echo the sentiments of the previous speakers. It would be nice to be there in person. I actually did my postdoc at UCSF, so I have fond memories of the Mission Bay campus. Indeed, my lab studies the chemistry of human-associated bacteria. This is really a summary of the lab in one slide. We want to know what molecules bacteria, in this case, mainly gut bacteria, are making, which bacteria are doing those transformations and using what genes, so what are the biosynthetic pathways, how are those molecules affecting bacteria-bacteria interactions, how are they affecting the host, and then how can we think about using that knowledge to help patients? Over the past 6 years or so of the lab's existence, we've focused our efforts on how bacteria are affecting host metabolism and immune function. And the focus of my talk today will be on the latter, how bacteria are affecting host immune function. One broad focus in the metabolite area of the lab is how gut bacteria metabolize endogenous, also known as host-produced small molecules. These are compounds like sterols and steroids and vitamins that the host makes, get into the gut, and then are metabolized into by enzymes into other molecules that we think have biological properties, modified biological properties. In this vein, we have two goals in our study of host-produced bacterially modified metabolites. We want to understand the roles of these molecules, both as groups and as individual compounds. We want to be able to modulate the levels in vivo of these compounds to elucidate pathways and also lay the groundwork for new therapies. Today, I'll tell you one longer story and one very short story at the end. The main focus of the talk will be about recent work that's in collaboration with June Hu's lab, an immunologist at Harvard Medical School, on how bacterial bile acid metabolites are affecting host immune function. And at the very end, I will touch briefly on new work where we have shown that bacteria sulfonate certain molecules and how those sulfonated molecules affect host immune function. So again, function today is really bacterial metabolites affecting host immune function. I mentioned this is highly collaborative work. I wanted to highlight the scientists who were involved here, June Hu across the street from us in the immunology department, and two talented postdocs, Seiyu and Dongyi in his lab. My lab, this work has been led by a former postdoc, Lina Yao, as well as graduate students Wei Li and Gabriel D'Agostino. When we look at causation studies, causal studies in the microbiome field that show how bacteria are affecting host immune function, there's some beautiful work out of many labs. I just highlight two examples here. Kenya Honda showed that a cocktail of 46 commensal Clostridia can induce the differentiation and function of regulatory T cells. And then in other work from Mathis and Benoit at Harvard Medical School, they showed that some of the common probiotics that we take inhibit Th17 cells. Just to zoom in on Th17 cells for a moment, we know that naive CD4 plus T cells can differentiate into many different types of cells. One of them is Th17 cells that express this nuclear receptor ROR gamma. These are pro-inflammatory cells, but they are necessary in many ways for our life and health. They help us to maintain a mucosal barrier, protect us against pathogens, but these cells are also implicated in the development of certain immune diseases. And today's focus will be on inflammatory bowel diseases like Crohn's disease and ulcerative colitis. Their differentiation is dependent on commensal microbes. We know this from studies of germ-free mice. Germ-free mice really in their guts do not have Th17 cells. So you need to have a microbiome to have these cells in your gut. But Even though we know something maybe about what bacteria or cocktails of bacteria can induce or inhibit these molecules, we don't know anything more, or we haven't yet discovered more about the mechanism, specifically what molecules could bacteria be making that could modulate these cells. One class of candidate molecules my lab has been studying in this regard are bile acids. Just to get everyone on the same page about bile acids, these molecules are produced from cholesterol in the liver. They are secreted as taurine or glycine conjugates. And the reason the host makes bile acids is to act as detergents that aid in digestion. They form micelles around fats and vitamins we couldn't otherwise absorb. They're actively and enterohepatically recirculated from the gut back to the gallbladder for storage after a meal. But a small percentage of these molecules pass into the large intestine, which is where, as we know, the majority of our gut bacteria reside. In the large intestine, bacteria have a slew of different enzymes that they use to chemically modify bile acids. Biosalt hydrolase is a widespread enzyme. I won't talk about it today, although the lab has done some work in this area. These biosalt hydrolase enzymes cleave this amide bond, releasing taurine or glycine, as well as an unconjugated primary bile acid. Bacteria also have other enzymes that convert so-called primary or host-produced bile acids into secondary bile acids. The focus of the talk really today will be on the conversion of the host bile acid chenodeoxycholic acid, or CDCA, into lithocholic acid, or LCA, and then further bacterial transformations of LCA. I mentioned that we know that these molecules act as detergents. They also act as ligands for host receptors. They have a steroidal core. We know that steroids are exquisitely selective and potent signaling molecules. Bile acids are signaling molecules too. They bind to nuclear hormone receptors and G protein coupled receptors. Two of the most famous ones are the farnesoid X receptor and TGR5. And by either acting as agonists or antagonists for these receptors, bile acids control key host processes involved in things like lipid and glucose homeostasis, energy expenditure, and immune response. And this has not escaped the notice of the pharmaceutical industry. Here's just one example of a company that's developing bile acid analogs to treat fatty liver disease. Returning to this question of which bacterially produced molecules could be affecting Th17 cells, this now returns to our collaboration with the Hull lab. It's known that ROR gamma T activation in naive CD4 plus T cells results in a differentiation of these cells into Th17 cells. In his postdoc, Jun He, our collaborator, showed that digoxin, which is a plant derivative, inhibits Th17 cells by antagonizing ROR gamma T. It's also known that certain sterols and hydroxycholesterols are agonists of ROR gamma T. When we looked at these molecules, what do they have in common? They have a sterol core in common. So in a collaboration led by the Hull lab, we decided to search for endogenous small molecules that could be modulating T cells. We screened a library of bile acids which have this sterol core and are endogenous. And in previously published work, we found 3-oxolithocholic acid, or 3-oxo-LCA, is the first endogenous inhibitor of Th17 cells, and it does so by antagonizing ROR gamma T. We encouraged the Hull lab in more recent work to test another bile acid, iso-LCA, isolithocholic acid, because after DCA and LCA, iso bile acids are actually the most abundant bile acids in the human gut. The range of iso-LCA concentrations in the fecal contents of healthy humans is, the mean is about 50 micromolar, and the range is anywhere from 0 to 150 micromolar. That indicates there is a large amount of inter-individual variability in the amount of this bile acid that's there. That means that different individuals could have different amounts of the molecule that could affect their health in different ways, but roughly 50 micromolar on average. And we found in very recent work that yes, iso-LCA also is an ROR gamma T antagonist and inhibits cis differentiation and inhibits Th17 cells. That's not surprising, perhaps, if we look at these molecules in a more three-dimensional way. Both 3-oxo-LCA and iso-LCA have a similar structure. They have this canonical bent shape of classic bile acids, and they both have oxygenation pointed in the same direction at the 3 position of the sterol core. We were able to show, in collaboration with the Hull lab, that iso-LCA and 3-oxo-LCA inhibit Th17 cells. Here on the right, you can see that LCA, that what we think is the parent molecule, doesn't have this activity, and a similar bile acid, another bile acid, iso-DCA, also doesn't have this activity. So this highlights a key point of my talk today, which is that small changes in structure amount to large changes in biological function when it comes to steroids and when it comes to bile acids. We then were able to show that when we colonized mice with Th17 inducing bacteria and fed either 3-oxo-LCA or iso-LCA, these molecules reduce Th17 cells in vivo. It's known that these two compounds are absent from germ-free mice. That means they're microbiome-dependent metabolites. But what wasn't known was whether bacteria actually produce these compounds. We then set forth a fairly basic biosynthetic hypothesis about how bacteria could be producing these molecules from chenodeoxycholic acid and then bacterially produced lipocholic acid. We proposed a relatively simple biosynthetic pathway where two enzymes, a 3-alpha hydroxysteroid dehydrogenase could oxidize at the 3 position to the ketone, and then a 3-beta HSDH could come in and do the reduction, inverting the stereochemistry. Again, these are simple changes from a biochemistry point of view, but LCA is not active in this sense, and iso-LCA and 3-oxo-LCA are, so it's important to figure out what enzymes are responsible for this transformation. We chose LCA then based on this hypothesis for our screening assays. And what Lena did in the lab is she took stool from 2 human donors. She recovered individual colonies on plates and then made an assay plates of about 1,000 total individual colonies. She then added LCA in liquid culture, our putative substrate, and then using UPLC-MS, we looked for new peaks that would indicate transformation to other LCA derivatives. We then recovered positive hits and restreaked and sequenced to get their function and identity, verify their function and identity. Using this process, Lena found over 40 different human gut bacterial species that produce either 3-oxo-LCA or iso-LCA from LCA. Here, just for the microbiologists in the audience, I highlight a few of them. Agarthala lenta and its actinobacteria relatives like Gordonobacter and Rholtobacter are good 3-oxo-LCA producers. Collencella intestinalis also makes this molecule. When we look at top producers of iso-LCA, we see mainly firmicutes, gram-positives, but we also saw that Bacteroides fragilis, a gram-negative organism, produces iso-LCA as well. I put our biosynthetic pathway, our proposal on the top, and I'll just walk you quickly through how we were able to elucidate the genes. We took known hydroxysteroid dehydrogenases from soil bacteria and other environmental sources, and we did bioinformatic comparisons with the genomes of producer strains. Then a classic conundrum that many in the microbiome field are facing, many of the organisms that we're interested in do not yet have genetic tools for their manipulation. So we use heterologous expression of putative genes in E. coli to elucidate some of these genes in bugs like Agarthala lenta. As we probably know, in the audience, Bacteroides fragilis does have genetic tools. So in that particular case, we were able to do genetic deletion in addition to heterologous expression to verify the gene responsible and find this new gene BF3538 that's a 3-beta HSDH. We worked with the Turnbull lab because one of our best producers was Agarthala lenta, and Peter very graciously provided us with a library of sequenced Agarthala lenta strains. And we were able to find, in collaboration with his group, that bacteria that lacked a homolog for the 3-alpha HSDH, here highlighted in red, also lacked the function, just as we predicted. But this also gives us a tool, because now we can go in vivo with these strains. And we colonized germ-free mice with the type strain, Agarthala lenta DSM2243, and we have to feed LCA because this LCA itself is a bacterial product, so we had to start with that. It's not present in germ-free mice. So when we give LCA as a substrate, we see that there are much higher levels of 3-oxo-LCA when we have a bug that has the the enzyme present, versus when we colonize with a strain that lacks the enzyme, we see lower levels. Here on the y-axis, picomole per milligram of wet mass approximately equates to about micromolar concentration. So we're at the roughly 30 to 40 micromolar levels here in fecal contents. We then endeavored to show that colonizing mice with bacteria that could produce this molecule affected Th17 cells in vivo. On the right, I show you on the x-axis, we have 3 quartiles of bile acid levels. We colonize mice with either high or low producers of 3-oxo-LCA. So quartile 1 has the lowest 3-oxo-LCA, quartile 4 has the highest. And we show on the y-axis that this is a molecule that suppresses Th17 cells. So as expected, when you have a high producer and high 3-oxo-LCA production by bacteria, you see low Th17 cells in an inflammatory mouse model, in this case, a Citrobacter infection. We then collaborated with Curtis Huttenhauer's lab and Clary Cleach's lab at the Broad to look at human data. We used IHMP data, and we looked at non-IBD patients as well as patients with Crohn's disease and ulcerative colitis. And now we are able to see that in dysbiotic Crohn's disease patients, so patients likely undergoing a flare, in the dark red here, we see lower levels of 3-oxo-LCA and iso-LCA. And now that we did the heavy wet lab lifting to isolate or identify the genes responsible, we also see lower levels of this homologs of this bacterial 3-alpha HSDH in these patients as well, indicating there was at least a correlation in human data between lower levels of this inflammatory metabolite and worse levels of Crohn's disease. In the interest of time, I just wanted to quickly highlight a third molecule. So this is yet another lithocholic acid derivative. This is iso-allo-LCA. And I apologize, bile acid nomenclature can be quite confusing. This is a different LCA derivative. And in previous work with the Hull lab, we'd shown that this molecule induces the differentiation of naive T cells into regulatory T cells. And so this is an anti-inflammatory effect as well. This molecule is shaped differently. Unlike canonical bile acids that have that bent structure that I showed you, this molecule actually has a flat structure because it has this hydrogen at its 5 position inverted, changes the shape, changes the function. We then returned to this screen and we didn't see any production of iso-allo-LCA from LCA. When we start one compound down in our biosynthetic pathway and we start with 3-oxo-LCA as a substrate, we were able to identify abundant Bacteroidetes species, kind of a theme of today. Bacteroidetes do a lot of biochemistry that convert 3-oxo-LCA to iso-allo-LCA. We proposed a slightly more complicated biosynthetic pathway here, where you start with 3-oxo-LCA on the left. A 5-beta reductase can make this alpha-beta unsaturated ketone. A 5-alpha reductase installs this key inverted stereochemistry at the 5 position. And then a 3-beta hydroxysteroid dehydrogenase would make iso-allo-LCA. We used so the first enzyme and the last enzyme have homologs in bacteria, and we use that to do blast searches in some of the genomes of our producer strains. And we were happy to find a gene cluster. In bacteria, oftentimes biosynthetic genes are clustered. They are in this case. And we were able to, with genetic deletions and biochemical heterologous expression, elucidate and confirm the function of this gene cluster as making iso-allo-LCA. Importantly, this is the first 5-alpha reductase in bacteria ever identified. There are 5-alpha reductases found in mammals, like in humans, 5-alpha reductases reduce testosterone, but none had ever been found in bacteria before this point. Then Wei in the lab was able to show that this 5-alpha reductase is important. Here is a phylogenetic tree of some producers and non-producers. The green dots show that you need the 5-alpha reductase to produce iso-allo-LCA. And then we were able to show now when we colonize mice with strains of bacteria, either a wild type strain, Parabacteroides myrtae, that has this cluster, or a knockout strain that we generated using genetic deletion that lacks this cluster, we're getting, and we have to co-colonize with this other organism that has a biosalt hydrolase. But when we do that, we're getting about 20 to 40 micromolar of iso-allo-LCA in vivo. So we can generate physiologically relevant concentrations of this compound in mice. Again, working with the Huttenhauer lab and Clary Cleach at the Broad, we are surprised in a good way to see even more striking correlations in the human data with iso-allo-LCA. Now we see really low levels of this molecule in Crohn's disease and ulcerative colitis patients, both in flare and non-flare states. On the x-axis here, you can see the percent zeros. This is patients that have none of this molecule. And you can see in the non-IBD patients, we only have 20% zeros, versus in the dysbiotic Crohn's, we have close to 90% zeros. So patients have none of this molecule, really showing a dramatic decrease in iso-allo-LCA in sick IBD patients. And now we've elucidated the gene responsible. We can see the levels of the 5-alpha reductase are similarly reduced in patients with Crohn's and ulcerative colitis. At the same time we were doing this work, Kenya Hanja was also independently investigating this molecule. He did it from a totally different point of view. He was looking at the microbiome of centenarians. These are people over the age of 100 compared to just elderly people in their 80s and in young subjects. He reasoned that these centenarians have been through a lot. They've been through 2 world wars, multiple pandemics. Maybe there's something special about their microbiome that makes them resistant to disease. He actually found that isoallo-LCA is significantly enriched in the microbiomes of these centenarians compared to elderly and young subjects. And he was then able to show that this molecule is an anti-infective that inhibits the growth of Clostridium difficile, and it doesn't affect other commensal bacteria. So we are excited about this result. And we think isoallo-LCA, not only is it anti-inflammatory, it also looks like it's anti-infective. It could be a beneficial molecule in some cases. So just to sum up this part of the talk, I've shown you 3 molecules that are anti-inflammatory. 2 of them, 3-oxo-LCA and iso-LCA, that inhibit Th17 cells, and isoallo-LCA that induces regulatory T cells. And we hope that either through us or through industry and pharmaceutical companies will investigate either the metabolites as therapeutics or the bacterial producers as potential probiotics for IBD treatment. All right, so very briefly, just at the very end of the talk, I'd like to talk briefly about a new project that's about to come out, we hope, in this summer, and that is also in this immune vein. And this is led by 2 postdocs, Jinsuk Park and Lena Yao, as well as a graduate student in my lab, Gabriel D'Agostino. And I told you at the beginning of this talk that the lab is really interested in how bacteria metabolize endogenous molecules. Well, one endogenous molecule is cholesterol. Cholesterol is provided both by the host and also in the diet. And we serendipitously found that when we colonized germ-free mice with B theta and we fed a high-fat diet, we found higher levels of this compound, cholesterol sulfate, in the fecal contents of these animals. Now, canonically speaking, this is a host-produced metabolite. That's how it's understood in the literature. It's made by this host enzyme SALT2B1. So we looked at expression of SALT2B1 in the mouse distal ileum and liver between B theta colonized and germ-free mice, and we didn't find any differences. So this really raised the question, can bacteria, gut bacteria, actually perform sulfonation? It's known that certain environmental bacteria and certain pathogens like Mycobacterium tuberculosis can perform this reaction, but it hadn't been characterized in commensal gut bacteria previously. Long story short, we were able to identify a gene cluster in Bacteroides theta. Interestingly, we didn't identify the gene cluster by looking for the sulfotransferase. That sulfotransferase actually has low homology to known bacterial and eukaryotic salts. We did it by looking for the genes necessary for cofactor synthesis, these cis genes. So once we found the cis genes, we saw a putative, a hypothetical enzyme next to it that we then were able to downstream elucidate as the salt. So I won't take you through all the details of the pathway that we elucidated using both genetics and biochemistry. What I will say is a couple things. One, that this is a pathway that requires the production of this key cofactor, PAPS, which then transfers this orange group, this sulfate group, to a substrate like cholesterol. The other thing I want to say is that I believe that the Johnson lab, Professor Johnson's lab, has also similarly discovered that bacteria produce cholesterol sulfate through independent means. And I think that's really fantastic news. One of the crises we have in our field is lack of reproducibility in science. And I think when 2 labs independently find a result, that gives us confidence and really helps the microbiome field show that these results are real. So we were able to show that this salt is widespread in gut Bacteroidetes species. It's really in Bacteroidetes and not so much in other phyla. We were able to show that when we colonized mice with a we made a cluster deletion strain of B theta. The wild type mice have higher levels of cholesterol sulfate than the deleted strain. And we were able to show that we did a little bit of investigation of what types of substrates are accepted. And we found, first off, this is not a promiscuous enzyme. It doesn't sulfate cyclohexanol or other alcohols, for example. It really preferentially sulfates steroids, that have a flat structure. Things like cholesterol, things like dehydroepiandrosterone and pregnenolone, these sex hormones, calcifediol, which is a vitamin D derivative, beta-sitosterol, which is a plant steroid, but they have to be flat. So this enzyme does not sulfonate bile acids, for example. But then in just the last 2 minutes, I'll show you some new results that come back to this key theme of the talk, which is about immune function. So what is the function of cholesterol sulfate? What's known in the literature? Well, it's known as a protease inhibitor, and it helps to modulate membrane permeability. It's also immunomodulatory. It inhibits T cell receptor signaling, and in one paper here at the bottom, it was shown to inhibit leukocyte migration in the mouse eye. So we wondered whether or not this molecule and bacterial production of CHS could affect immune function in the gut. We first started by doing an in vitro study in which we did a trans-well chemotaxis assay, in which we looked at the effect of cholesterol sulfate on leukocyte migration. Here, lower numbers on the y-axis mean inhibition of migration. So you can see at physiologically relevant concentrations like 10 micromolar, we see that cholesterol sulfate inhibits this migration while cholesterol does not. We then turned, with the help of the Hull lab, into an in vivo model. And in this case, we're colonizing mice with either B theta wild type or the cluster deleted mutant. And we are transferring naive CD4 plus T cells into these animals and tracking where they go. And it's known that naive CD4 plus T cells home and migrate to secondary lymph nodes, like mesenteric lymph nodes, in response to CCL21, this chemokine. And we were able to show in this work that when you colonize mice with the wild type strain that has this BT salt gene cluster, you have lower migration of these CD4 plus T cells to mesenteric lymph nodes compared to when you colonize with the cluster deleted strain. We then finally, to sort of wrap everything up, we also were able to show that dysbiotic Crohn's disease patients also have lower levels of this gene cluster than compared to non-IBD controls. What we don't know yet, we've shown that at least the presence of the gene cluster affects leukocyte or T cell migration. We don't know whether or not that has anti-inflammatory effects. We need to use anti-inflammatory mouse models to do that. So that's a next step that we're working toward. Okay, so this research really takes a village. Just in the interest of time, I'll quickly thank our main collaborators in the Hull lab, as well as help from the Huttenhauer lab, Randy Longman, who provided fecal samples, Peter Turnbaugh and Romnick, who provided strains, and Clary Cleach, who helped with metabolomics. And just really quickly, the Devlin Lab is looking for postdocs. If you are in particular a person with a background in cell biology, microbiology, or molecular biology, and you're interested in learning some chemical biology and studying gut microbes, I encourage you to get in touch with me. So thank you very much for your time and for the invitation. If we have any time left at all, I'll take questions.
Thank you so much for an amazing talk. We'll take questions. So feel free to ask in the chat and to raise your hand here. I guess I'll get started. So you've shown that the derivative of lidocholic acids are affecting directly Th17 cells. I think you showed quickly the mechanisms or the receptors. Can you elaborate a little bit on that and how it's specifically affecting Th17 cells?
Right, yes, I spoke very, very quickly. I apologize for that. So we've shown that first June showed that digoxin, and now these molecules 3-oxy-LCA and iso-LCA, they affect the cells by antagonizing this key nuclear hormone receptor, ROR gamma, which is known to control the differentiation and function of these cells. So we've shown direct binding of these molecules to ROR gamma, and we hypothesize that that's the mechanism.
Awesome, thank you. Question here in the back.
Thanks, Lynn. That was a great talk. I wonder, like, do you think that it is surprising that so many compounds are at lower levels in IBD? That, is that reflective of just an overall sort of loss of microbial metabolites?
Right. So this is a good question. Peter, I would ask if you were one of our reviewers, but I know you couldn't have been because you were on the paper. We worked with a little bit with the Hutton Hauer lab to try and answer that question, because indeed, I joke, but a reviewer brought that up. And we show that this is not, when we control for phyla level changes. So there are shifts, major shifts in Bacteroidetes and Firmicutes that happen during inflammatory bowel diseases. So couldn't it just be that, the depletion of one or the other could also result in depletion of the molecules? But when the Hutton Hauer lab controlled for those phylum level and even I think genus level changes in bacteria, we still have the effect. We still have the correlation. So yes, there's certainly large overall changes that occur, but I mean, it's only so much we can do with correlational studies, of course, but it does seem like even when you control for those factors, we still see these differences.
All right, thank you so much. If we don't have further questions, we're gonna wrap up here this first session. Thank you again. And actually, I would like to thank all the speakers for this fantastic session. And so we'll have a short break now, and we'll start back in an hour at 1 p.m. Thank you, everyone.
Okay, well, welcome back, everybody, from lunch. I hope your microbiome is very active, metabolizing bile acids and all the lipids that you ingested in the last hour. Really looking forward to this session. It was really a great morning of talks, but especially looking forward to this one. So our first speaker of this session is Dr. Ines Thiele. She's a professor and principal investigator of the Molecular Systems Physiology Group at the National University of Ireland in Galway. Galway is the California of Ireland, so she picked well. Her research aims to improve the understanding of how diet influences human health. She earned her PhD in bioinformatics from the University of California, San Diego, and held a faculty appointment at the University of Iceland and the University of Luxembourg prior to her current role. She's an author of over 100 internationally published scientific papers and reviewer for multiple journals and funding agencies. And in 2020, she was named highly cited researcher by Clarivate and received the NUI Galway President's Award in Research Excellence. And so today, Ines will present a talk entitled A Computational Modeling Approach to Host Microbiome Co-Metabolism.
Okay, thank you very much for the introduction and for the invitation to speak today. I lived in San Diego for many years, and I would say Galway is a complete opposite. San Diego was always sunny. Here we have always rain and sometimes sun. So, but it's great anyway to be here. And so my talk will be about computational modeling to host microbiome metabolism. And the interest really originated some years ago when I got interested in diet and nutrition and realized that it's not only us that are eating, but also the microbes that are living within us. And so since I am a bioinformatician / systems biologist, I wondered whether we could apply our approaches, that is genome scale metabolic models and computational modeling, to further understand the diet, microbiome, and health axis. So as you know, there are many microbes living in and within us. And we use a tool that's called genome scale metabolic networks to better understand a target organism, and particularly a metabolism of a target organism using in silico approaches. So traditionally, when a genome scale metabolic network was reconstructed, we started with a genome annotation as illustrated on the left-hand side, and we complemented the information that we could retrieve from the genome annotation with extensive literature review. And we put both information types in a structured manner into the computer. And since we collected a lot of information about the biochemistry and physiology of the target organism, we really refer to the resulting database, metabolic database, not only as a database, but a knowledge base. So you can actually look up what are the references for the metabolic biotransformations that we capture in the database itself. But importantly, this database, also a knowledge base, serves as a computational model. And concurrently, we also develop metabolic maps for the different organisms. For some of the model organisms, it's available, the microbiome model organisms, and then also for human metabolism as depicted with this icon here. And more recently, we are now working on a microbiome-wide metabolic map that will allow for the mapping of predictions, but also for omics data. So overall, these genome scale metabolic networks represent the integrative knowledge about chemistry, genomics, and physiology of the target organism. Now, once we have this computational model, we can actually predict emerging properties of the model. And this is done, for example, by altering the nutrient supply or the diet to the target organism, and then predicting how, for example, the biomass yield would change. But alternatively, we could also change the flux through a particular reaction corresponding then, for example, to a gene knockout or a genetic modification or mutation. And this is achieved by tuning the flow through the tubes as illustrated here, similar to a valve system. And importantly here, we can not only predict what would happen locally, so in the surrounding of the reactions that we manipulated, but also globally. So you can imagine if you had a knockout in the oxidative phosphorylation, so what would be the consequences of the biomass yield if you had such knockout? So this is really powerful. Unfortunately, this approach of reconstructing that I just showed you a slide early on has been traditionally very slow. The systems biology community has developed methods to automate the reconstruction process, but it remains still a semi-automated process because you do need as much as possible experimental data or literature-derived data. So how do we get around this issue when we want to reconstruct the species found or strains found in the human gut microbiome, which might be hundreds or thousands of different strains depending on condition? And we derived a semi-automated tool a few years ago, which resulted in Agora-1, which is a compendium of metabolic reconstructions for the human gut microbiota in specific. And that particular resource captured 14 different phyla, more than 773 microbes, and overall there were 3,800 unique reactions that we covered if you were to look at the combined set of reactions. This Agora-1 has been used widely by the community for approaches that we actually did not anticipate it in the first instance. And so that's actually very gratifying because Nevertheless, a lot of work went for many people into the reconstruction process here. And it's great to see that these reconstructions are really used by the broader community. During the time course, we got interested in drug metabolism, mostly driven by the fact that we analyzed metagenomic data using Agora1 from elderly people, either from Parkinson's studies or Alzheimer's studies, and realized that many of these elderly individuals not only vary in their dietary intake, but also in their drug administration. And so we searched the literature a little bit and found a fantastic review that has been published a few years back that describes the direct and indirect mechanisms by which the gut microbiota can modify drug activity and toxicity. And so a well-known mechanism is, of course, the activation of a prodrug. And this is often exploited by the pharmaceutical industry when they deliver, for example, colonic drugs. But then also the gut microbiota can inactivate active molecules or even toxify them. And of course, they can have an antimicrobial effect, the drugs themselves on the microbes, or promote selective microbial growth. But at the same time, the microbiota can also produce certain metabolites, such as paracresol, that then interact with host metabolism and confer with drug metabolism. So we set out to query the literature and try to identify what is known about microbial drug metabolism, and then expand Agora1 that I just introduced into Agora2, which now covers many more strains than before. And so we have over 7,000 strains now capturing 24 phyla. We consistently incorporated more information about biochemistry of these strains and species, but also drug metabolism. So great emphasis was on drug metabolism. One aspect that I'd like to highlight here is not only for drug metabolism, but also for general metabolism, is that we conducted extensive manual comparative genomic analysis for more than 5,000 strains across 35 different subsystems. This is important as many of these strains have not been, or species have not been well characterized. So not a lot of experimental data is available that could be used for improving and curating the metabolic reconstruction. So having good annotations is key. So we formulated the drug metabolites and drug reactions in a manual manner, so this has not been automated. At the time when we conducted the work, there were 15 known drug metabolizing enzymes encoded by 25 different microbiome genes. And those include activation, inactivation, degradation, and detoxification for 98 prescription drugs. So for those ones, we can now describe what is happening in certain strains and species of the gut microbiota. And here's two examples shown. One is a cancer drug, which is given as a prodrug, the 5-fluorocytosine, that's then activated to 5-fluorouracil. And the second example is a Parkinson drug, levodopa, that is converted either to dopamine or further to M-tyramine. And of course, the seminal work by Peter Turnberg is really responsible for many of these drug-related enzymes and genes, in particular in Igatella lenta. As I mentioned beforehand, we developed a semi-automated reconstruction pipeline building onto pipelines that have been generated by the community. So we start with draft reconstructions. In our case, they're generated by Modest Seed and K-Base, but there are other resources as well where you can obtain them. we require, our tool requires taxonomic information for each of the species and strains, where available experimental data and comparative genomic data can be included. And then we have an extensive pipeline that converts the draft reconstructions into our in-house name scale, and that has an advantage that we then can combine that with human metabolic modeling, as I will illustrate to you later. We incorporate information about the gram status, the experimental data and genome analysis data, perform gap filling, remove artifacts from the reconstruction process, and run an extensive test suite. If the reconstruction passes the test suite, it will pass, otherwise it will be debugged in a manual manner, which is then any of these debugging tests are then propagated throughout the microbiome reconstructions as appropriate. And so this cycle is run iteratively. And at the end, you have refined metabolic reconstructions that are, to the best of current knowledge, representative to the metabolism of the reconstructed organisms. Here's an overview about the reactions and metabolites of the different phyla and the corresponding reconstructions. And as you would expect, reconstructions within the phyla of the proteobacteria have a wide spread of number of reactions as well as metabolite, but they're also the largest ones that are available. This has not only to do because of higher density of knowledge for representatives of proteobacteria, but also because they have larger genomes. And then, of course, we have other representatives that are much, much smaller. As to the drug metabolism, as there was a focus on Agora 2, we tried to identify independent data set and independent data against which we could validate predictions of drug metabolizing capacities. Importantly, this data has not been used during the reconstruction and refinement process that I just showed you. Overall, we were able to find experimental data for almost 170 species of strains, corresponding to 253 drug micropairs. And you can see here the predictions in silico as well as in vitro, and overall the accuracy is quite good, so is the sensitivity and the specificity of the predictions. Considering that these data has never been, the reconstructions never been seen, the data. So the interesting part is now, of course, where we have the discrepancies. In 31 of the cases in vitro, the organisms can metabolize a drug, while in silico we can't. Reasons for that can be either different culturing conditions, so we don't model the right conditions, or we miss the pathways with a comparative genomics approach, and the reasons are manifold. And it's definitely worthwhile to further refine those ones based on the experimental data. But at the same time, we say in 19 of the cases that there's a drug metabolizing capacity, while in vitro it could not be observed. Again, this could be due to differences in medium composition, but also regulation could play a role that we do not capture in our metabolic reconstructions. Here's a general overview of the 15 drug metabolizing enzymes and the number of strains that can within the resource carry out those functions, and then which drugs are actually modified by the different enzymes. And as you can see, there are many certain enzymes that appear in many of the strains. And this is due to the fact that these enzymes, like the cytidine deaminase, is of course a central metabolic enzyme having other functions in metabolism than only the deamination of drugs. And similarly, the case for the cytosine deaminase or the alkaline phosphatase. In contrast, the digoxine reductase and also the dopamine dehydroxylase, they're very specific to certain strains and species. And in fact, there have been so far only identified, to our knowledge, in Igatella lenta by Peter Thornburg and collaborators. So now that we have this resource of gut microbiome reconstructions, we can actually move to the next step and ask, well, what are these microbes doing in a community? And do different microbiome compositions found in individuals correspond to different drug metabolizing capacities? To address this question, as a proof of concept, we used a published study of a Japanese cohort of 365 colorectal cancer patients and 251 healthy controls. Therefore, we used our microbiome modeling toolbox. From the experimental data, we retrieved the species level abundance. Those ones are metagenomic or shotgun sequencing data. We mapped them onto pan-species models of Agora2, and in fact, we could on average map 97% of the named species in a given sample. So that's a quite high coverage and gives us the confidence that we actually can say something about the metabolic activity of the corresponding community. We created then community models for each of the individuals. So one sample corresponds to one in silico community model. We administered an average Japanese diet and added into the diet or to the diet a given drug metabolite. And then we performed our simulation similar to the tuning. Here it's now the presence and absence of a particular drug. And we asked the question, can certain drug derivatives be synthesized by a community? Yes and no. And the results are shown here. So that's binary. Blue means that the community could in silico conduct a conversion, and red means no conversion of the drug was possible. And so just to orientate you, the y-axis are representatives of the different drug categories. And then on the x-axis, we have the 600 odd microbiome computational models. And you can see most of the drug conversions could take place. Qualitatively, there were no differences. However, quantitatively, there were quite some differences. However, we found that certain conversions could only be carried out by a subset of the microbiome community models. In fact, basalacide was only converted in 42% of the cases, and the conversion of both levodopa and digoxin was only possible in those microbiomes that contained Igatella lenta. Furthermore, we could also ask now using the quantitative predictions whether there's any correlation between the secretion potential, that is the quantitative predictions of drug derivatives, and age or BMI, and the results are shown here, and those ones are statistically significant. And this is quite interesting because that could mean that indeed, as you, for example, age or you have a higher BMI, that you actually have a lower or higher potential of activating, deactivating, or detoxifying the corresponding drug. On the other hand, we worked on human metabolism. That has been an effort of the systems biology community since, well, more than 15 years by now, I think. And it started with a generic model of the human metabolism now in its third iteration, published in 2018, capturing more than 3,000 genes and 13,000 reactions. The problem on the generic reconstruction is that it does not capture the different organs and how the organs in the human body cooperate. So we set out and developed the first comprehensive organ-resolved whole body metabolic model, starting from Recon 3D, a lot of proteomic and metabolomic data, and again, literature resources. And now we have a metabolic map for each of the organs within a whole body model. And here's some statistics about the whole body model. Now we have more than 80,000 reactions and 56,000 metabolites. Those ones are non-unique, and the organs are connected via 13 different biofluid compartments. Now, in terms of host microbiome co-metabolism, the really exciting part is that we can actually add on or impute into the computational model in the large intestine a microbiome community model, which is personalized based on a given metagenomic sample. And we then can ask the question whether there are any consequences of having a different composition and a different metabolic activity of the microbiota in different human organs in silico. And so, for example, for the brain. And we used a similar pipeline for doing so. And here we're predicting the effect of having different microbiome compositions generated from the Human Microbiome Project, which contained over 100 healthy individuals, the effect on neurotransmitter brain production capacities. And one of the really exciting aspects that one can do in silico and not in vivo is that we actually can compare germ-free versus microbiome-associated human metabolism. And these results, predictions are, at least qualitatively, as one would expect, without the microbiota, we have limited production of dopamine, serotonin, and adrenaline. And this is consistent with our knowledge that the microbiota itself is a reservoir for neurotransmitters, but also it produces actively the precursors for the host to produce then the neurotransmitters. So this prediction is consistent. Now, I had promised that I would go into virus infection, but I'm not quite sure on how I'm doing with the time. Can I get some feedback on that? Okay, so then I will quickly show you what we have been doing on modeling virus infection, that is rather COVID infection. And I will keep it brief. We took the whole body model of human metabolism. We formulated a COVID or SARS-CoV-2 specific metabolic reactions, basically representing the virus replication in the lung and then its exhalation, and also in some peripheral organs, but then the virus is not exhaled, to kind of assess, are there any differences in metabolic activity on the host site upon virus replication. Importantly here, we do not only focus on spike, but we also accounted for the different numbers of protein copies of the other structured proteins, as well as capturing the non-structured proteins of the virus when accounting for the viral replication within the host. And we again had simulated diets, and we performed our constraint-based modeling approach. And one thing that we observed is that lung metabolism is predicted to change substantially here, as illustrated on the human metabolic map, specifically for the lung. And you can see the changes either up or down in blue and red respectively. So quite some changes occur during the viral replication in lung tissue, at least in silico. And that is, again, consistent with what we would expect about cellular hijacking of viruses, as well as SARS-CoV-2 infection. Now, one thing that we observed during our simulations is that actually some of the amino acids were limiting the replication and the shedding in silico of the virus, and in particular, isoleucine and threonine uptake were limiting this. And we wondered if diet could actually have an effect. And as for lung replication, we did not see a lot of differences between the different diets that we investigated in silico. However, we found that vegan and vegetarian diet actually resided in the lowest shedding rate in silico. However, for liver metabolism and also adipocyte metabolism, the replication of the virus was very much affected depending on which diet was administered in silico. And so this is, of course, very hypothetical, but indicates that maybe some of the liver damage that might be observed could be also attributed to differences in diet composition. Now, we were curious to understand why isoleucine and threonine would have an effect both on the lung shedding rate as well as through the diet. And we observed using the different variants of concerns defined by the WHO that actually the dependency of the virus particle decreased with silico virus shedding rate as a function of isoleucine, and it was in return to increasing its dependency of threonine. And that occurred in part in single mutations in structural proteins, but since these structural proteins are highly abundant in the virus particle, it actually resulted in a higher requirement and hence had an influence on host metabolism. So overall, this proof of concept study for virus replication using the host metabolic model, whole body metabolic model, identified isoleucine as a potential limiting factor for replication rate of SARS-CoV-2. What that would mean in real life, I have no idea. It is very difficult to correlate these results with epidemiological data that is available. So our vision here goes over all that, having now this human metabolic model available, which incorporates, will incorporate and can incorporate genetic information, hopefully at one point genomic information, that we can then make predictions about changes in metabolic fluxes across different organs. We can incorporate changes in food items and those dietary components, as well as personalize the whole body model on the level of metagenomic data using either 16S RNA or metagenomic data. And we can either predict the metabolome on the level of blood, CSF, or urine, or use a metabolomic data to further personalize the host model. With that, I would like to thank you for your attention. The many people that have been working on the different aspects of this work that I showed you, both at the University of Ireland and former group members, which were mostly located at the University of Luxembourg, our collaborators, the patients that gave us samples, and of course, the funding agency. Thank you very much.
Thank you so much. That was really a wonderful tour de force. I'll take questions from the room first before moving to the online audience. Any questions online? Peter?
Yeah, great talk. I was curious about this dependence on isoleucine and other amino acids. Is that coming from sort of mutations in the amino acids that make up the viral protein itself, or there's some other interaction with the cell?
No, it is due to single mutations that result in changes in from isoleucine to threonine in most of the cases. And really the change in requirement of a single change or mutation results really by the fact that there are so many copies of the structured protein incorporated in the virus particle.
Sergio, I see your hand is raised. Go ahead.
Yes, thank you. Hi, Ines. A great talk. I enjoyed it. I think your whole body metabolic modeling is really impressive and far-reaching. My question is, how do you feed the parameters for the metabolic fluxes that you will need for the models to learn? Are there enough in vitro or in vivo data to infer these parameters?
So the approach that we're using is constraint-based modeling, and we assume the system to be at a steady state, which means that we need less parameters than if you perform ODE-based modeling. And so the assumption here is the steady state. As far as we compute, we are normally going on a per day basis. So the assumption of metabolism being at a steady state, I think, is valid. And the second aspect of constraint-based modeling is that we assume that the system tries to optimize a particular state. And this is, of course, a very big assumption. And for example, in the virus example that I showed you, I optimized for maximum virus production. Now, there might be other optimization criteria that are in place, but by doing so, I can identify with limited experimental data potential flux values for each of the reactions throughout the network. Now, having more data available would mean that I could further constrain, and hence the word constraint-based modeling, the set of feasible fluxes such that we would be only limited to what's biologically relevant. Does that answer your question?
Yes, thank you.
Great. Okay, thank you so much. Our next speaker is Dr. Paul Blaney. Dr. Blaney is an associate professor at MIT and a core member of the Broad Institute of MIT and Harvard. He earned a PhD in physical chemistry from Harvard and did his postdoc studies at Stanford University. And there he developed novel optofluidic methods to perform single-cell microbial sequencing. Currently, the Blaney Lab investigates DNA-protein interactions, evolutionary processes, and functional differences between cells using microanalysis systems. And the title of his talk is High Throughput Analysis of Microbial Communities with Droplet in Microwell Array Technology. Dr. Blaney.
That's right. Thank you so much. Let me share my screen and just let me know if there's any technical issue here. I want to thank everyone for the opportunity to present today. I appreciate it very much. And I'll be focusing on a technology platform developed a few years ago in my group and how we're applying that for analysis of microbial communities. I'll also include some other infectious disease related applications to show you the versatility, which, as a surprise to us, turned out to be one of the great strengths of this platform. I've got a few disclosures to make on funding and relationships and financials interests in industry. I think the most salient here is the last one, Concerto Bio. So this is a new company that some group members spun out of my lab, applying the technology platform I'm going to focus on today. And Concerto is focused on the discovery and development of therapeutic bacterial consortia. So keep an eye on them. I'm very excited about what they're doing. So the story here really starts with our interest in developing combination antibiotic therapies. And Inés's talk is just a perfect introduction, because it's really, I think, our growing knowledge of these amazing biological networks that inspires this, right? Because it's, for me, it's hard to look at, a metabolic diagram, for example, and not start thinking about, combinatorial interventions that could push, to a desired therapeutic outcome. And my group typically thinks more in terms of drug interventions, but dietary interventions or combinations of dietary and drug interventions are certainly relevant. Now, combination therapies are already quite broadly used in oncology and infectious disease. My observation is many of these are combinations of drugs that were known previously as single agents, and so that's great, and these are important in the clinic. But what we're particularly excited about is phenotypic screening for empirical discovery of combination interactions based on new mechanisms, and really doing discovery at the level of combination and building technology that enables that kind of activity to be practiced on a routine basis. And so we were attracted to, as technologists, as biological engineers, to droplet microfluidic technology as a basis for this kind of approach. And that was really, I think, driven very simplistically by the idea of that one might be able to achieve really substantial scale using a droplet approach, and be able to tackle these large combinatorial screening spaces head on. And so very quickly, we learned about the field about the problem with this approach that was broadly recognized in the field, but we didn't know about that at first, which is that drug molecules leak like crazy out of droplets. And so you should be seeing in the movie the dye molecule resirufin that has drug-like solubility equilibrate within each set of 7 droplets here over the time course. And that illustrates the transport of a small molecule from droplet to droplet. And that's a tough basis if you want to run a large-scale drug interaction assays at the droplet level. Now, students in the lab, they put their engineering hat on and came up with an engineering solution, which was to corral sets of droplets in sealed microwells in an array format. And so that's also represented here. The red dotted circles show microwells designed to accommodate sets of 7 droplets, and the microwell in the middle happened to be loaded with 7 empty droplets. And what you can see over the time course is that those droplets remain dark, indicating that they are chemically compartmentalized from the adjacent microwells, and dye is not being transported into that microwell. So with this chemical containment at the microwell level, that's going to be our assay unit for our screening activities, and is the basis of the platform. So this is, of course, compatible with phenotypic cell-based screening, so we can hopefully do some interaction discovery across the full complexity of cellular biological networks. A feature, and you'll appreciate it on the next slide, or 2 slides from now, I think, is that the amount of work we have to do to set up these assays, it actually scales not with the combinatorial space that we're exploring, but actually just the number of inputs from which we're assembling those combinations. So that's a big advantage with this approach. And then, of course, as a microfluidic platform, this has very low compound consumption, and for drug screening in particular, that's an important aspect here. Since for combination screening, we're screening many combinations with each given compound from the library, and we need to manage the compound consumption. So we're building these arrays and doing a lot of this multicolored imaging. You'll see how we use that to identify the compounds that are present in each assay point. But before we get there, I want to just emphasize the microfluidic aspect for those who don't think about that every day. And so simple point, just how extremely small those are. So the natural scale of oceans, I like to think of as cubic kilometers. So that's trillions of liters, or trillion times bigger than our everyday sort of kitchen and meal volume scale. I bet there are a couple liter water bottles in the room there, and we actually have to go down by a similar factor to reach, in turn, the microfluidic scale, which, depending on the specifics, is going to operate at the scale of a trillionth to a billionth of a liter. So this is really quite an extremely small scale, and that was a big part of the attraction that we felt for this kind of a approach for really trying to achieve high throughput for these sorts of drug screening and microbial interaction assays. Okay, so here's a example of the workflow in a little movie that shows practically what that means in the lab. And this is described in terms of the drug screening, but it's really similar when we're looking at microbial communities or some of the diagnostic applications I'll come to at the end. Name of the game is to color code each of the inputs with a mixture of fluorescent dye molecules up front, and then emulsify individually each of those inputs. So this is the part of the work for setup that scales with the number of inputs. When we're ready to screen, we combine all those emulsions. So we have a rainbow mixed emulsion that we can load into the microwell array. Now, droplets randomly populate the microwells. In this case, I'm illustrating microwells designed to hold exactly 2 droplets. And then we can image the microwell array, get that beautiful multicolor image so that we can re-identify in each microwell which compounds in this example are present. Once we know what's in each microwell array, we can merge the droplet set to formulate the assay. And then in this example, this is describing a microbial growth inhibition assay. So there are fluorescently labeled live bacteria. And after an incubation period, we can come back and image that bacterial channel to measure the degree of growth in each microwell. So we spent some effort validating the system for these growth inhibition assays and showed that we were able to recapitulate the growth kinetics of a shake flask in this chip-based system. Also that we could accurately measure IC50s for a panel of drugs. And we also did some analysis to figure out how many microwells we needed to collect statistics over to get a overall result with a given degree of reliability. And that all looked pretty good, and we went off to the races with screening. So our first screening project was to conduct a sensitizer screen, asking which compounds from the Broad's drug repurposing collection could sensitize E. coli to the action of a panel of 10 antibiotics. So this was a fairly complex combinatorial screen with some scale behind it. Nonetheless, a couple of grad students in the lab the same two who developed the platform initially, were able to complete this in 10 days without any automation equipment, really just operating the platform manually. And you're seeing the summary of the results from the screen here in terms of the heat map, which has a statistic for the compound antibiotic interaction, a synergy score, and then a p-value on the vertical axis. And so requiring hits to surpass cutoffs on both axes, we had something like 20 hits by fairly stringent criteria. And so, of course, we went on to validate those and found when comparing with a conventional liquid culture checkerboard assay, a pretty high validation rate of our hits. And among those, some interesting ones. So, this being a repurposing collection, many of these were compounds in development against human targets, really without any annotation for activity in microbes. Nonetheless, we had a number that validated with activity sensitizing E. coli to a number of the antibiotics. And interestingly, these hits did seem to actually concentrate in antibiotics that don't have clinically useful activity against gram negatives. And so, we looked at this really as reactivating some of these drugs against gram negatives and thinking a lot about antibiotic entry into the bugs. Now we've since expanded, and I'm not going to go into it for limitations of time today, this effort, and we're now screening a larger compound libraries, including novel chemical matter, against a larger panel of antibiotics and across a larger strain panel, still focused in gram negatives. But I want to shift gears a little bit now and talk about expanding the application of the platform into microbial community analysis. And so this means a couple of things. It means we need to be able to handle a wider range of bacteria, and not just a small set of screening strains. And you're seeing here from one of our studies the diversity and droplet level colony morphology across a set that we had engineered to be fluorescent. And fluorescence we still like in this context because we can track specifically the growth of one member in the context of a microbial community. And then, of course, permute the label across the community to really characterize what's happening there. But of course, there are other optical readouts that are really well suited to the chip. For example, the resazurin assay, which allows us to follow the activity of a community as a whole. And so in one study, we focused in on pairwise interactions between a set of soil bacteria against a large variety of environmental contexts, 39 different carbon sources in this case. And so here we were interested to carry out a broader survey just of, take a bunch of microbes, what are the statistics of interactions and how sensitive are those to the environment? And so we found there are actually a whole spectrum of interaction types, everything from direct competitive interactions to very strong mutualisms, with a whole variety of effect sizes, and that these were quite strongly dependent on the chemical environment. The highlights here were positive interactions were actually quite common, and particularly so in the case where we paired one organism that did not grow very well in a given environment with another that did grow well. And in those cases, it was almost always the result that the poor grower was facilitated. And this was like in up to 85% of cases in the most extreme examples of that scenario. So we think there are pretty strong implications there for designing consortia and bringing environmental microbes into laboratory cultivation. So we've been further developing the platform as well Here I'm showing going to larger microwells for more complex communities. And in this study, what we did is we focused on one member. This was an Herbispirillum, which is a model plant symbiont, and asked the question, okay, are there other organisms, other consortia that can help support Herbispirillum growth across chemical variability, so different carbon sources again, but then also biotic variability, so make Herbispirillum growth robust to the presence of additional species. And so here we looked at community complexities all the way up to 16 different microbes, microbial species, with that variability in carbon source. And we're able to identify a couple different consortia that seem to confer robust growth on Herbispirillum across those dimensions of variability. So moving ahead here, I mentioned we've also been applying this platform for microbial diagnostics. And our first example was for viral nucleic acid testing. And so this is a project we started pre-pandemic. It was started by two postdocs, one in my group and one from Pardis Sabeti's lab, with a strong input from a graduate student jointly advised between my group and Deb Hung's group. And the idea here was to take these powerful CRISPR-based nucleic acid detection chemistries, Sherlock is the version developed at the Broad, and multiplex them. And here we had a high ambition for that. So not just a multiplex across many different viral nucleic acid targets, but also to simultaneously multiplex across many different test samples. And so you're seeing in the bottom, that viral panel, which was more than 150 targets, and our original panel did include some coronaviruses. The pandemic emerged while we were in review, so we added a CoV-2 test to the panel that was published. And the heat map is showing results mostly across synthetic targets there with a pretty good specificity. And I'll say, this result is really only after 2 rounds of optimizing that panel, which isn't very much for such a complex multiplexed test. And yet we're showing pretty good performance there. And that is really a testament to the power of the, and specificity in particular, of these CRISPR-based detection chemistries. Now, more recently, we adapted this for application to bacterial pathogens. And so in this study, just came out a few months ago, we have a simplified front-end workflow. So it's just a single one-pot PCR. We're producing a top A amplicon that gives us a nice balance of coverage across the bacterial pathogens. We looked at 52 that we wanted to cover, but then still with enough sequence variability within that amplicon where we could address CRISPR guide RNAs and distinguish amongst those 52. In this paper, we're also showing for the first time a microfluidics free or a droplet free day of test workflow where we can take an array that's been preloaded with all the assay content and then simply just apply a aqueous test sample to that and then read out. In this case, we demonstrated some mobile phone imaging, imagining deployments closer to the point of care and in lower resourced settings. So we're pretty excited about some of these new results. And I'm going to wrap up just with this last example. So this is our first foray into assays of mammalian cells on the system. And here we've been focused on looking at primary human immune cells that we can put into the system, treat combinatorially with immune ligands, and then recover the cells from the array for single cell RNA-seq readout to look not at the baseline transcriptional state of these cells, but at their responses to the stimulations that they received in the chip. And so we're applying this in connection with some work we've had collaboratively with Neha Cohen and others on a host side view of bacterial sepsis and viral sepsis. And so that's been great. Moving forward, I'm really excited and hearing the talk. Maybe some of you are thinking about really high order combinatorial experimental designs where you've got human cells and different microbes and different immune ligands and maybe some different drugs as well, and trying to sort through all the complexity in that sort of system. So I'm going to stop there and just briefly summarize. For those of you who've seen other microfluidic droplet systems, I think one of the things that distinguishes our approach is this is really a parallel processing droplet system, and that gives it some unique characteristics, particularly in terms of scalability and reliability. This is a workhorse in our lab. We run it every day, particularly these days for the antibiotic sensitizer screening. And it's really just, as you've seen, worked well for us, even as we pivot from assay to assay and application area to application area. One limitation I will point out in the microbial context is, these are small population sizes of bacteria, and we typically just, run a single batch culture to our endpoint. And so this is not, in its current version at least, suitable for long-term studies of evolutionary dynamics. But it is certainly useful for a lot of things. All right, I'll stop there. Thank you so much for your attention, and I'm happy to take as many questions as there's time for.
Wonderful. So we'll take questions from the room first. Paul, maybe I could start. I'm really intrigued by the kind of immune assays that you're using in this with this technology. How complex are those kind of immune cell combinations, or what kinds of primary immune cells are you starting with for these kinds of assays?
So we're starting very simply. Our proof of concept work was carried out on PBMC and with a single stimulation in the chip. And this was designed so that we could compare directly to a parallel set of stimulations in just liquid culture in a plate, so we could understand where we are technically. Now, I think the unique capability of the system is really going to shine when we get to high order stimulations, which is of course very relevant to the in vivo context. I think there's no immune cell in the body that's only receiving a signal from one cytokine or microbial metabolite.
Yeah, great. Do we have any other questions in the room? Maybe I can do a quick follow-up then, just because you've piqued my interest. How reproducible is that if you have multiple donors providing those PBMCs? Because we see a lot of variability in the response of PBMC pools to stimuli.
Oh, great question. No, and this is so big in our sepsis work. So we always look at multiple donors, and we always see differences. Yeah, it's. I'll take the liberty to extend the answer a little bit in another direction. So, one of the, I didn't emphasize it, but one of the characteristics of the random or consequence of one of the random assembly of the droplets into the microwells, so we get statistical coverage of the combination space. And so there's a nice analogy there to shotgun sequencing, right? So when you do a shotgun sequencing experiment, you can specify the average coverage level, but then you have variability around that because of the average sampling, the statistics of sampling sequences. So it's just like that for us, except with our microwells sampling from the combinatorial space. And so what that means is we set that average sampling level quite high to make sure we achieve a minimum sampling level for a large fraction of that space. And so we have great statistics typically for each of the combinations that we're testing. For drug screening, for example, we'll typically make that average micro assay replication level 10 or 15. And so even from a single chip run, we have those statistics and we do understand quite well where we are from a technical reproducibility standpoint. And so, thinking about, comparing results from multiple donors, because we have those other internal statistics, we're very well positioned to talk about those differences.
That's great. Great. Ben.
Hi, Paul. Thanks so much for your talk. Followed Sherry's work out of your lab. It was really awesome. I was wondering about, so you mentioned your limitation that you're kind of at a pretty small number of interactions. I wonder kind of what you all are thinking about in kind of relation to the microbiome field and kind of looking at communities of larger numbers of microbes and things like that within your platform, and just kind of thinking about how to capture some of the community dynamics within single drops or within merge drops or something like that.
Yeah, great question. So, certainly I think we can go to fairly complex communities, although this is still at the micro scale, and so the total population sizes won't be that large. Really thinking about the analogy to the real anatomy and physiology, the spatial structure, I think, is a big factor there that needs to be considered as well, and good opportunities to think about how to map our system onto the spatial structure that would be present natively. And so I'm not sure the right way to think about it. So Jared Kay, also part of that early cohort developing and applying this technology, he was very enamored with this idea that, okay, the droplet is somehow the right experimental model for physical micro niches that are going to exist in a lot of microbial growth scenarios in the wild. So, I think in the microbiome, it's worth thinking about that, right? I mean, is that like one little locus of microbes in the gut? How does that map? And do we need to start thinking about ways to actually let the droplets talk to each other a little more rather than keep them fully isolated, which was our initial drive thinking about some of the compound screening. So great question. And I love the idea of pushing this platform in that direction.
Great. Thank you so much. That was really great. So rounding out our session, this afternoon is Dr. Ophelia Venturelli. Dr. Venturelli is an assistant professor for the departments of biochemistry, bacteriology, chemical and biological engineering, and biomedical engineering at the University of Wisconsin-Madison. She received her PhD from Caltech, and her lab currently investigates and engineers the spatiotemporal behaviors of microbial communities. The title of her talk will be Predicting and Designing the Dynamics and Functions of the Human Gut Communities.
Thanks very much. Just going to share my screen. I wanted to thank the organizers for the opportunity to speak today and tell you about what we're working on. And yeah, can everyone hear me?
Yes.
Okay, great. Yeah, so microbes have evolved in communities in nature, and we've studied them until recently, primarily as like single organisms in the lab. And I think to actually understand their functions, we have to understand them in the context of other species because they evolved to interact and perform functions within the communities themselves. And so the other important aspect of microbial communities is that they can expand the functions of individual microbes. So through the interactions with other species, new metabolisms are enabled, new molecules are produced, and they can be more productive and resilient as a community. And then finally, the community functions can be harnessed for a variety of applications, and in particular, human health is one that we're particularly interested in. So the gut microbiome is a really dense and diverse ecosystem and provides many beneficial functions for the host. And in a homeostasis environment, the gut microbiome has, kind of a positive impact on many aspects of human physiology and health. In response to environmental disturbances, antibiotics, or even malfunctions of the host, the microbiome can be shifted to an alternative state because the environment changes, and that leads to changes in the composition and the functions of the communities. And the microbiome changes because they're engineers of their environment. They actually can reinforce this unhealthy state, which we call dysbiosis. So I think there's an opportunity in microbiome engineering and thinking about predicting the microbiome to think about how we can shift and steer communities to desired states. How do we enhance the production of beneficial metabolites and degrade metabolites that are toxic for human health? And the microbiome is producing these wide range of metabolites, and many of which we've actually begun to understand their mechanisms, or molecular mechanisms of interaction, of how they interact with the host and how they affect the ecosystem. And so these molecules are critical functions of the community. So when I talk about functions, I typically mean these metabolites that are being produced at the community level. And this is a bidirectional interaction. So the microbiome is producing these metabolites which impact the host, and the host changes the environment to impact the microbiome, and it's a feedback process. So they're complex feedback loops that are driving the system's behaviors. So central to understanding this really complex system are the interactions between microbes. Microbes are, interacting with each other continuously. They're changing as a function of time and space, and the molecular mechanisms of interaction can be quite diverse in how microbes interact with each other in nature. And so in order to understand how we can manipulate this system and steer microbiomes to healthy states and optimize the functions of these communities, we have to understand and map these interactions, which is why the microbiome is so complex, or one of the main reasons. So our lab is interested in kind of a bottom-up approach where we take microbes that have been isolated from different individuals, and we have this toolbox of microbes, and we can build microbiomes of variety of ranging complexity, and introduce them into different experimental, platforms ranging from low throughput mouse experiments and bioreactors to ultra high throughput approaches such as droplet microfluidics. And so we also use liquid handling robots, which, so all of the microbes we culture are anaerobic, so they're sensitive to oxygen. So we culture them in glove bags, and in order to enable high throughput characterization, we can use liquid handling robots to create these combinations of different communities that we want to study, or different environments that we want to study. This data we use to build computational models. Our models are informed by the experimental measurements, and the models can decipher interactions between microbes and also predict and design communities from the bottom up. So that's ultimately, we use the models as a tool to drive biological discovery, and also for designing communities with optimized functions that we can understand more deeply using, hypothesis-driven techniques. So one of the big questions that we've been interested in is mapping these function landscapes. So we can think of the presence, absence of different species in a community as generating different levels of a given function. This could be a metabolite, a health relevant metabolite, or set of metabolites that we want to study. And this space actually grows exponentially with the number of species in the community. So varying the presence, absence of different species, we have a very large space that is difficult to explore, especially when we talk about, microbiomes of hundreds of species. We want to be able to explore all the different functions that these communities can perform. So it's a very large landscape that we have to efficiently navigate. And so our lab is thinking about different ways, both experimental and also computational, and all of the computational approaches we use are informed by our experimental data. But how do we navigate this space? One, to understand the interactions within these communities so that we can design more kind of ecology-inspired interventions, but also how do we optimize functions of interest? So if we want to optimize, the production of beneficial metabolites and use these communities as therapies, how would we actually identify these unique communities among all the possible communities we could study? So I'm going to talk about a few kind of vignettes from the lab. And the first is actually focused on leveraging this computational modeling technique integrated with our experimental data to understand and design synthetic microbial communities. So one of the metabolites that I'm sure many of you have heard of is called butyrate. Butyrate is a beneficial metabolite in the gut. It promotes homeostasis and has many interactions with the host. And this pathway is actually a specialized function. So only a subset of the microbes in the gut can produce butyrate. So they're a unique group of species, and their ecology is really interesting and really important for understanding how we can manipulate butyrate concentrations in a predictable way. When we look at individuals, they vary widely in the amount of butyrate concentration across different people. And then if there are dietary interventions such as consuming resistant starch, and that can lead to also unpredictable effects on whether butyrate concentration goes up or down in a given individual. So we'd like to be able to predict butyrate concentration within the gut and also be able to design communities that can steer kind of low butyrate producing states, microbiomes, to high butyrate producing microbiomes. So that's our ultimate goal. And we sought to actually understand the interactions within a community that lead to different production levels of butyrate within our system as a starting point. So central to this approach, because microbiomes are so complex and there's so many interactions and unknown mechanisms that are taking place, are computational models. And one of the models that we have used and others have used widely is called the generalized lockable Terra model. It's a set of ordinary differential equations that describes the interactions between species and the growth dynamics of individual species. And the growth dynamics are governed by the single organism's growth rate and its interactions with all pairs of species. So it's a pairwise representation of a microbial community. This model, one of the advantages is that we can look at time series data or even just, sparsely sampled time series data, and we can infer the parameters of this model because we can observe all the species in our data set. And so we can infer these parameters quite robustly from experimental data. So this model, I'm going to talk a little bit about in other models, but I just wanted to kind of introduce the parameters because I'll mention this AIJ parameter a couple times, which means the impact of species I on species J. So we were interested in leveraging computational models combined with experimental data to understand these interactions that shape butyrate production in our system. And so we combined a generalized lockable Terra model, which describes the growth dynamics of the community, with a statistical model that takes the endpoint species abundance from the model and maps that to butyrate concentration. So this is a very interpretable model, which is one major advantage. And the disadvantage is that it's a little bit more rigid, and I'll talk more about kind of the flexibility question later on. But we were interested in using this highly interpretable model to capture these interactions within our system. So we also leverage a design, test, learn cycle. This is an engineering inspired approach where we can take a limited set of measurements from our system, experimental data, build a preliminary model, use that model to guide the next stage of experiments, which select experiments based on their information content. So how much information can we gain from an individual experiment? And then we use the experimental data from that next experiment to update our model so that we have a more predictive model. And we can do this iteratively until we are satisfied with our model's predictive performance. And we used a 25-member synthetic human gut community that contains 5 butyrate producers. So these are 5 highly prevalent butyrate producers. This gut community was designed to kind of represent the healthy human gut and also represents kind of the metabolic and functional diversity of many species that we find across individuals that are very prevalent across individuals. But we also wanted to think about kind of this trophic web of how microbes kind of interact and filling all of those metabolic niches to the best of our ability. So using a series of design, test, learn cycles, we were able to generate a model that could make predictions about the concentration of over 1 million consortia. All of these communities contain all 5 butyrate producers in them. Our guts have multiple butyrate producers, so we wanted to consider kind of the functional redundancy case of where we have many butyrate producers present. So every data point on this scatter plot represents a different community that was predicted based on our model. On the x-axis is the predicted total butyrate producer abundance, so how much butyrate producers we have in the community, and the y-axis is the butyrate concentration that we predict. And we observe this bimodality depending on the presence and absence of D-sulfovibrio piger, which is a hydrogen sulfide producing bacteria in the human gut and has a very unique and interesting metabolic niche. And so we were interested in taking this model that was only trained on kind of lower order communities, so communities that had like 3 to 5 species and 2s and 1s. So this was a design, test, learn cycle. We wanted to use this to make a prediction of whether we could decipher this effect of D-sulfovibrio piger, because it's such a prominent effect in our prediction landscape. And we found that if we design communities that either maximize or minimize butyrate or kind of randomly sample this space, that D piger always shows up as an inhibitor of butyrate production. So it doesn't matter how we sample this design space. We did some further experiments because we hypothesized that hydrogen sulfide was inhibiting butyrate production in our system because D piger has this unique metabolic capability. And we found that butyrate production is inhibited by hydrogen sulfide. And this is something that's an ongoing project. We're trying to dig into the molecular and ecological mechanisms that are taking place with this really interesting interaction. But we found that D-sulfovibrio piger does inhibit butyrate production through hydrogen sulfide. And there's some interesting connections clinically, potentially, between hydrogen sulfide and butyrate production that we're exploring now. So using all this data that we collected, we had gone through several design, test, learn cycles. We could build a model that encompassed most of the information we had collected and use that model to predict communities that we had never measured before or the model's never seen before. And we found that this final model was highly predictive of butyrate concentration across a wide range of community contexts. We have a driver species of butyrate, which is Acaciae, and Acaciae kind of determines kind of this high or low butyrate production. It's a major driver. And one of the interesting things we learned from this whole DTL cycle, design, test, learn cycle, is that we couldn't accurately predict high richness communities, so communities that had over 10 species, with a model that was trained on low richness communities. So this suggests that there are many higher order interactions or significant higher order interactions that are driving butyrate production in our system for these high complexity communities or high richness communities. Our model does not capture higher order interactions. So that's something that I'll address in the next kind of vignette. But another interesting aspect of this data was that, typically we think of, taking functional organisms that have a benefit, say a probiotic that has some sort of benefit to, human health, mix them together, create a mixture of species that can provide a kind of maximal benefit by combining them. So if we mix all 5 species together, all 5 butyrate producers, we get 40 millimolar butyrate concentration. If we vary the presence and absence of other species that don't produce butyrate in our system, we can increase butyrate production by up to like 45%. And this is just what we've measured so far. We haven't tried to actually optimize this further, which is something we're interested in doing in the future. So this suggests that microbial interactions can be engineered to enhance beneficial functions, not just between organisms that have the function of interest, but between organisms that are interacting with these functional organisms. So we like to think of these communities as like support communities. They're supporting a beneficial function, and their interactions are leading to a more kind of an optimized level of activity, which was interesting from a microbial engineering perspective. So using all the data that we collected, which was many measurements of microbial communities and our computational models, we were able to identify kind of an interplay of resource competition, pH modification, and cross-feeding, and hydrogen sulfide that all interact to determine butyrate production in our system. So it's pretty complex. And one of the other aspects that was clear based on the data and the model was that pH and competition, so resource competition, have a higher order interaction on butyrate production. So if you have an organism that can buffer the pH and have very strong resource competition, we can maximize butyrate production. And we demonstrated this experimentally in addition to our predictions. So this again suggests that we need to think about flexible modeling frameworks that can capture these higher order interactions and also begin to maybe have better kind of computational efficiency to be able to explore many different types of communities. And so another kind of limitation with what we had explored was that we were looking at a single metabolite, and we know that microbiome metabolite. So we expanded our set to look at 4 health relevant metabolites. These are fermentation end products, acetate, lactate, succinate, and butyrate, that all have been linked in some way to human health and disease. And so they're health relevant. They also influence the ecosystem, the gut ecosystem. So by, through cross-feeding and through, maybe toxic kind of effects on gut, on the gut community growth as well, and metabolism. And so in order to kind of capture all this complexity with these interactions, we turn to recurrent neural networks. And this model is called the long short-term memory model. So it's a machine learning model, and it's highly flexible. So it can approximate any function. It's scalable to large communities. It can learn complex interactions between system variables and feedback loops. It can capture kind of the temporal behavior of these communities. And so we were interested in exploring and leveraging this modeling framework to kind of capture the relationships of that between microbes and metabolites in this complex system. So we took this 25-member community, and we had some initial data that we had from our previous study, and we used that to train a recurrent neural network model and use that model to predict every possible community. So now we can look at all 26 million communities very efficiently, and we can train our model very efficiently on experimental data. We are predicting 4 metabolites, and we're also looking at microbe, and micrometabolite interactions with interpretation tools. So machine learning models have a reputation for being black box, and they are black box, but we have tools for interpreting the relationships between variables in our system. So we can actually get biological insight from these models. And so using this recurrent neural network, we can now explore the relationships between all of these different variables, all these different metabolites that we're looking at within this community. And every data point represents a different consortia. Our model can capture the trade-offs between different metabolites, such as lactate and butyrate. This is due to the metabolic networks, structure of the networks. So the model learns this very efficiently. It also predicts that most communities have typical behaviors. And then there are a few key driver species, such as Acaciae, which determine kind of high versus low butyrate production in our system. And we wanted to challenge our model to try to maximize and minimize all combinations of metabolites. We call this corner cases. These communities are very infrequent in the function landscape. And so it challenges our model, but also we're kind of interested in understanding what leads to very extreme metabolite production profiles. We could study kind of the typical behavior, but the communities do tend to this typical attractor state. And so if we choose a community randomly, we're most likely going to be measuring similar functions. So we wanted to challenge our model. We demonstrated that our model could outperform other models that we've developed, and is highly flexible, can capture these like complex relationships between all these variables and be highly predictive. And so this scatter plot's just showing that for most variables in our system, the LSTM model outperforms our composite model, which is the GLV statistical model, or replacing the statistical model with different types of models, such as random forest and feed-forward networks. So LSTM has the best performance overall across most variables in our system. And then using this model that was trained on this large experimental data set, we can infer microbe metabolite and microbe interactions within this system. The LSTM model, every interaction network for each community is unique. But we can take the average across all communities, and we get this kind of global interaction network. And what we identified from this is that Firmicutes, actinobacteria, and protobacteria within our community, they tend to specifically impact metabolites in a unique way, whereas Bacteroides tend to shape the community growth dynamics. So they're not contributing specifically to these metabolites that we measure. They might be contributing to many other types of metabolites that we're not measuring. But in this system, Bacteroides are driving community growth and community assembly and not impacting uniquely the metabolite production. The other interesting aspect of this model elucidated that acetate and succinate are influenced more by interactions between species, and butyrate and lactate are primarily driven by action of individual species. And so we're able to extract some interesting biological insights from this model. So I think I only have a few more minutes. Maybe I can go a little bit over, but I wanted to possibly skip ahead. I'm sorry, this is just kind of showing that the LSTM can predict community dynamics and also be used to identify kind of design rules, presence, absence of different species that contribute to different dynamic profiles. But I wanted to mention kind of a newer project that was built on some preliminary work we did a while back, looking at using droplet microfluidics as a tool to both study communities and single cells. And this particular work has been published previously, and we're working on kind of using sequencing as a readout instead of using fluorescence as a readout, so it can be more generalizable. And that's kind of ongoing work that hopefully I'll talk about in the future. But one thing that we learned from this is that communities can be studied in a very quantitative way within these droplets. We can elucidate, quantitative relationships between these microbes. We can identify the effects of like antibiotics on shaping a microbial interaction network. And we can model the growth dynamics using stochastic models. And so we were really motivated to kind of use this droplet microfluidic system as a way of studying both communities and single cells. And more recently, we have been leveraging the power, kind of the ultra high throughput aspect of droplet microfluidics to look at genetic heterogeneity within microbiomes. So there's many examples of heterogeneity, both at the phenotypic level and at the genetic level within communities. And this heterogeneity actually drives differences in community functions. So it's really important for us to understand this, but typically it's hard to do. These just some examples are phase variation, mobile genetic elements that move, from one species to another, or specialized metabolic activities that might be found in one strain, but not another strain of the same species. So we need to be able to develop more kind of easier, kind of more accessible methods for studying this genetic heterogeneity. And so a talented postdoc in the lab has developed a method that allows us to do targeted sequencing of specific loci within thousands. We create millions, we can encapsulate millions of cells, but we typically sequence on the order of 1,000 to 5,000 cells. And we can target specific loci and then multiplex this, so we can look at many loci within individual cells within a community. And I won't be able to go into the details here, but just at a high level, we've applied this to study phase variation, which is a really interesting and important phenomenon that determines kind of adaptation of microbes to the host environment. and has kind of protective effects. It's kind of a polysaccharide shell that's created around the cells, and it's controlled through reversible genetic change that's very rapid compared to, say, mutations or insertions or deletions that occur over longer timescales. And we've been able to study this within B. fragilis and map all of the phase variable states within a population. And this is kind of like, we've been able to kind of study the temporal dynamics and model this phenomenon using the data that we collect from using DOTA-seq, which is this targeted single cell sequencing method. And so we can actually begin to understand the diversity within a single population or microbiome community, how phase variation kind of changes as a function of time, and also the extent of phase variation, all within one experiment. So it's very rapid and very, very efficient. And we've also been studying antibiotic resistance genes and how they can move through a microbial community, how they change as a function of time. And this kind of data is really not available through any other method. So we can look at both community composition changes temporally, but at the same time, within those individual cells, be able to map the individual genes of interest. In our case, we're looking at antibiotic resistance genes and how they change as a function of time within those single cells. So in summary, designing high throughput experiments and computational models together is a powerful way to explore structure function landscapes of microbiomes. I've shown that microbial interactions can be engineered to support communities for functions of interest. And I think this overall goal of being able to predict and understand interactions within the microbiome moves us toward a personalized, and precise microbiome intervention. So it really lays the foundation for these kind of next generation approaches for medicine. And finally, highly parallelized measurements of microbes can reveal both microbial interactions and also single cell genetic heterogeneity and phenotypic heterogeneity. With that, I'd like to thank my group, which is a fantastic group of basic scientists and also engineers. Also wanted to mention we have several open postdoc positions right now in different areas for different projects. And I'd be happy to share more information, just feel free to contact me. And with that, I'm happy to take any questions.
So much, Ophelia. A question in the room.
That was super cool. Thank you. I had a couple questions about the first part of the talk. First of all, I was wondering if you have looked at in population metagenomic data sets to see if the interactions learned by your model or the predictions are reflected in human data or in low butyrate versus high butyrate in humans. And I was also wondering if you looked at whether different environmental conditions affect the predictions of your model, like if you change the carbon source or things like that.
Yeah, those are both really great questions. So the first question, we haven't done that, and I feel like it's really important. So something that, thanks for bringing that up again. It's been on my mind for a while, and something that we need to do. The second question, we have started to explore the effects of dietary fiber on community assembly and also on butyrate production. And so this is something that we've really been digging into, how it shapes interactions and whether we can kind of combine maybe dietary factors and communities to yield a more robust butyrate production when we actually try to invade a natural microbiome and alter its butyrate production state. So those are ongoing projects that we're working on. We do have one preprint of that looked at the effects of dietary fiber on community assembly, which I didn't get a chance to talk about. But yeah, so hopefully I'll be able to talk about that in the near future.
Thanks.
Any questions online? Okay. Do we have one more from Peter?
Bring you the microphone. Yeah, great talk. I was wondering, how much, do you think that the predictions would really change if you had a sink for the butyrate, like, mimicking an enterocyte?
Yeah, that's another great question, because butyrate concentration can build up in our communities and probably is, inhibiting the production of more butyrate in our system, whereas in the host, the host is absorbing the butyrate. So there's a kind of removal of butyrate from the system. Yeah, that's a great question. I think the best way to kind of study that would be, possibly some sort of co-culture system, which is, challenging, but something that we're working on, to kind of co-culture communities with host epithelial cells and look at, the interaction between the community and the host. We can also do, simpler things where we can transfer the communities to a new environments is just kind of like a poor man's chemostat, just basically transferring the community to fresh media so that the butyrate concentration doesn't build up to too high of a level. So that's also something we can do, that we haven't fully explored, that the effect of this passaging on community assembly and butyrate production. But yeah, that's a very important point. I would expect that that we know that certain organisms are inhibited by butyrate, so their growth is inhibited by butyrate. So it's very possible that some of the interactions that we're measuring in our system are due to butyrate specific effects, because butyrate concentration builds up. So in that sense, it could be affecting community assembly and function. Yeah.
Paul, I saw your hand was raised. You want to unmute and ask your question?
Yeah, absolutely. Yeah, thanks for a great talk, Ophelia. I was captivated by the DotaSeq workflow. Could you just say a little more about that?
Sure. Yeah. So essentially, we can encapsulate. So one of the major advantages of DotaSeq is that we can separate like lysis and PCR amplification, so that we don't have to optimize both of those in the same environment, which makes it really hard. And we've tried this in the past, because like the conditions that are good for lysis of bacteria are not good for PCR. So that's one major advantage. The other aspect is kind of the way we design like these barcoded oligos, so we can barcode individual cells. So it's worked across a variety of different systems. So we're pretty excited about applying it more broadly, and also having other labs adopt it, because hopefully it'll be easy to use by the scientific community. So yeah, hopefully we'll have a preprint out on that at some point soon, and I'm happy to share it with you. So yeah.
Cool. Yeah, looking forward to finding out more about that.
Cool.
Great, thank you. And thank you to all of our speakers. This was really a spectacular session. We'll take a short break and we'll reconvene at 3 p.m. local time. Thanks so much.
We are ready to get rolling whenever you are.
Wonderful. Can you see me? Okay, wonderful. And all right, so welcome back, everybody. And it is now my pleasure to introduce Dr. Breck Dorikop. Breck is an assistant professor in the Department of Immunology and Microbiology at the University of Colorado. And he earned his PhD in microbiology from the University of Washington, where he studied quorum sensing gene regulation in gram-negative Burkholderia species. He completed a postdoctoral training at the University of Texas Southwestern Medical Center with Dr. Laura Hooper, focusing on bacteriophage interactions in opportunistic pathogens of the intestinal microbiota. His group currently uses a combination of molecular genetics, biochemistry, computational biology, and animal models to study bacterial viruses and other forms of mobile DNA in the context of host microbe interactions. The title of his talk today will be Enterococcal Phase-Host Interactions and Mechanisms of Resistance. Breck, please go ahead.
All right, great. Thank you, Sergio. Let me get my stuff up here. There we go. Everybody should be able to see that. All right, great. Well, thank you all for the invitation to speak today, and it's just really been a wonderful opportunity to hear about some of the more cutting edge microbiome research that's going on around the United States and in other parts of the world. So this has just really been fantastic. So as Sergio mentioned, I'm a newly minted associate professor now at the University of Colorado Anschutz Medical Campus. And today I'm going to tell you about some of the work that we've been doing focusing on enterococcal phage host interactions and mechanisms of bacteriophage resistance in these important opportunistic pathogens. All right, so the enterococci are gram-positive commensal bacteria that are found primarily in animal gastrointestinal tracts. However, they are found in other sites of colonization, such as the oral mucosa, vaginal mucosa, and are even found in various other environments where mixed microbial communities reside. Now, they're typically benign commensals in individuals that are healthy, but they can become pretty significant, serious opportunistic pathogens when individuals become immunocompromised or when they undergo extensive broad-spectrum antibiotic therapies. And this can really lead to a lot of problems, including bloodstream infections. They can colonize wounds, and they're a major problem in individuals that are receiving broad-spectrum antibiotic therapy, resulting in urinary tract infections. And in rare cases, they can actually enter the blood and colonize the heart and begin growing vegetative clusters on the heart and cause endocarditis. And so this is, obviously a very serious problem. And so they're antibiotic resistant bacteria of concern as well, due to the fact that they're the fifth most common nosocomial pathogen that's isolated from a hospital setting. And this is primarily Enterococcus faecalis and Enterococcus faecium. And they're resistant to vancomycin in a lot of cases. And even now more regularly, they've become resistant to many of the contemporary drugs that have been identified that are useful against vancomycin resistant enterococci. So to put this into context, at the University of Colorado Hospital here in Aurora, 70 to 80% of Enterococcus healthcare associated infections are antibiotic resistant. The vast majority of these are vancomycin resistant E. faecium that are also resistant to beta-lactams such as ampicillin. And what really makes them prolific in their ability to acquire and retain antibiotic resistance traits is the fact that they have a large repertoire of mobile DNA that allows them to horizontally acquire these types of traits, contributing to their opportunistic pathogenic lifestyle. So as many of you are familiar with, there's been a resurgence in interest in using bacteriophages and antibacterial therapies, in Western medicine. And probably more recently in the last 5 years, there's been a number of high profile cases showing that in individual emergency IND cases cleared by the FDA, that bacteriophages can be used successfully in combination with antibiotic therapy to ameliorate multi-drug resistant bacterial infections in afflicted individuals. And so just to give you a little bit of a primer on bacteriophages, these are obviously viruses that infect bacteria. They're very abundant and diverse, and they're also genetically tractable. And they're considered safe due to the fact that they only infect bacterial cells, and they're often self-resolving, meaning that when the target organism has been depleted, the bacteriophages themselves no longer remain in the system. So my lab's been very interested in studying the molecular aspects of how bacteria evolve resistance to phage infection, as we view this as a pretty critical parameter in the successful use of phages therapeutically in medicine. And so there's a number of different strategies that bacteria use to evade phage predation. And today I'm going to tell you two short stories. One, in the first part of this talk, it'll focus primarily on cell wall polysaccharide biosynthesis. and enzymes that are critical for phage infection. And when resistance emerges in these genes, it comes with fitness trade-offs. And the second part is unpublished work where we've identified mobile elements that carry resistance genes to phage infection through acquisition of mobilizable plasmids. So the first part of this work is really spearheaded by two individuals in my lab, Anushala Chatterjee, who is a former postdoc, and Sydney Johnson, who's a senior graduate student who's actually defending her PhD next month, and a technician in the laboratory, Sarah McBride. So when I first started my group in Colorado, we amassed a large collection of phages that could infect Enterococcus faecalis strains. And we simply screened these strains for sensitivity to these phage panels. So what I'm showing you here is basically a map of how these phages infect different strains in our collection of E. faecalis, and whether they infect these cells very well, so they're highly virulent, as shown by clearing on a plate, or whether they have weak infection as indicated by these pink squares. So what you can appreciate here is that there's a diverse range of host specificity within this collection of phages for different strains within our collection. So what we did is we took a few different phages that had very distinct, unique genomic signatures and different morphologies, and had different host specificities, and we simply evolved resistance to these phages and asked a very simple question: well, what are the mutations that arise in isolates that are able to withstand phage infection for prolonged periods of time in the laboratory? What we discovered was that in all cases, regardless of the phage and the bacterial strain that was used to evolve resistance, all mutations mapped to one very specific locus in the E. faecalis genome. And this is the locus known as the enterococcal polysaccharide antigen, or EPA. And specifically, what I'm showing you here within this locus are sites where mutations were found, as indicated with these asterisks, in variable genes that are downstream of the core polysaccharide biosynthetic locus of EPA that are involved in the production of teichoic acids that are decorated onto this molecule. And so EPA is a protrusion of polysaccharide composition that is attached to the bacterial cell wall. And specifically, it's composed of a ramen backbone that's produced by a number of genes that are involved in its biosynthesis, shown here in gray. And then teichoic acid decorations are then attached to this ramen backbone by genes that are shown here in black. And this is where we found the vast majority of the mutations were in genes that are involved in the decoration of the teichoic acids on the surface of this molecule. So specifically, I'm going to focus on one particular glycosyltransferase, and this is a gene we call EPAS. It's a glycosyltransferase that's responsible for covalently attaching GalNAc to the teichoic acid decoration on the surface of the ramen backbone. And you can see that we had a spontaneous mutation in EPAS that basically confers resistance to phage infection. If we make a clean deletion in this gene in frame, you can see that we recapitulate the phenotype. And more importantly, if we complement back in EPAS on a plasmid, we can restore infectivity of these strains by these particular phages. So this tells us very nicely that this single glycosyltransferase is critically important for promoting phage infection in E. faecalis. So we wanted to know a little bit more about the function of EPAS in the context of the enterococcal lifestyle. So as I mentioned before, Enterococcus is a commensal, and we have really good models to study its interaction with the host environment using gut colonization models. So we routinely in my lab use a model where we take wild type conventional C57 black 6 mice, and we expose them to Enterococcus in their drinking water. These mice become colonized with E. faecalis over the course of a couple weeks of using this as their sole primary source of drinking water. Importantly, this is a model that does not require perturbation via antibiotic therapy. So it's more of a, I guess, a natural route of colonization, you might say. And how we do this is after a couple weeks of exposure to these animals with Enterococcus in their water, we take them off the antibiotic water and allow them to wash out, and they'll become chronically colonized with E. faecalis, and we can take this out for many weeks and see stable engraftment of the organism within the intestine. And then we can, of course, ask very specific questions about the abundance and density of the organisms within this environment under different perturbations. So when we did this with either the wild type E. faecalis strain or our clean deletion EPAS mutant, you can clearly see that after the washout phase here, at 18 days post exposure to the water, you can see that the mutant colonizes the animals less well than the wild type strain. And importantly, if we actually challenge these animals with vancomycin, you can see that the wild type bacteria will bloom in the intestine following antibiotic introduction, whereas the mutants fail to do this as robustly and as to a high of a order of magnitude as what you see for the wild type. So what this tells us is that EPAS is important for expansion of the enterococci and their ability to resist infection or resist antibiotic therapy. And it's a critically important macromolecule for persistence within the intestine. We've also been interested in understanding the kinetics of how these mutants might arise in the intestine using phage as a potential therapeutic for the decolonization of intestinal VRE. And so in this model, what I'm showing you here is a mouse that's actually receiving receiving vancomycin ahead of introduction of E. faecalis into these animals for colonization. And then we're treating these with oral phage inoculations over the period of about 6 days and monitoring the abundance of the bacterial colonization in these animals. And what's notable is that in animals that are treated with phage, we can see very good decolonization that maps pretty closely to the total number of phage particles that are found within the stool of these animals that are collected over the time series. What was surprising to us was that when we did this in germ-free mice that lack a microbiota, we didn't really see any decolonization over the same time period. So what this told us is that there's something functionally different about how these bacteria are removed from this environment in a phage-dependent manner that's dependent on the microbiota. So we decided to dig into this in a little bit more detail. And so in this situation, now we're taking germ-free mice and we're doing fecal microbiota transplantations into these animals prior to, or just following the introduction of E. faecalis into the germ-free animals themselves. Then treating them with phage over the course of 1 week and assessing the overall density of the bacterial population in these animals, but also the level of resistance that emerges. What we saw was that in animals that receive the fecal transplant, in germ-free animals that do not receive a fecal transplant, you can see that following phage treatment, you get a pretty significant increase in the number of bacteria that are resistant to the phage itself coming out of the stool. Whereas when we do this in animals that receive a fecal transplant, you basically see no emergence of resistant colonies over the course of the therapeutic regimen. So this tells us that in the absence of the microbiota as a selective pressure, there's a high abundance of phage resistance that's emerging in these animals. It's becoming probably the dominant composition in the microbiota. However, when there's a complex community that's present in these animals, this is somehow suppressed. So we're still trying to get to the mechanistic basis for this. But what we think is going on, oh, and I should say this, that when we pull isolates out, they all have mutations in EPA, which we know are important for colonization and that do have colonization defects. And I'm showing you here an EPAS mutant has been pulled out of these. And we just screen a handful of colonies, but you can see that there are other mutations in the EPA genes themselves. So what we think is going on here is that in the absence of the microbiota, the phages will select for variants within the population because it's very, very high density, right? And these variants over time become the vast majority of the bugs that are within the microbiota, and they maintain dominance, and they just shift their phenotypic state. However, in a complex community where you have other bacteria that these organisms have to compete with and are likely less fit in the presence of, when you get depletion of the target organisms that are susceptible, those variants that are selected for are restricted from outgrowing just due to traditional colonization resistance. All right, so to finish this out, I'd like to move to a different topic now that's related to the work that I've previously presented, that focuses more on some of the molecular interactions of bacteriophages with distinct mobile elements in the enterococci. And specifically, this is work that's been done by Sydney Johnson in the lab, who had mentioned is graduating next month, and a newer PhD student, Josh Kirsch, who's done some of the bioinformatics and biochemistry for this project. Okay, so many of you have probably seen, the slew of, new papers that are being published probably on a almost seems like weekly basis on new types of antiphage systems that are being discovered in a myriad of different bacteria using metagenomics and other kind of functional assays. And so there's many different mechanisms that have been discovered that have really unique and interesting biology, and biology that also overlaps with some of the things that we know about antiviral mechanisms in eukaryotic cells that kind of speak to the potential nature of some of these more primitive antiviral systems in prokaryotes, potentially being the precursors to the systems that have developed in mammals and higher animals. There's many mechanisms that are still being characterized, and we really know very little about how phages escape some of these systems and overcome these resistance mechanisms to then flourish and find successful hosts to reproduce in. So this really came from a kind of very insightful observation that Sydney made when she was in the lab, when she was working on some of the earlier work that I showed you a few slides ahead, when we were doing this screen, looking at these panels of phages that we have in our lab that can infect different strains of E. faecalis. And she focused very specifically on this one phage that we call 47. And as you can see here, based on this, killing map here, that phage 47 cannot kill our canonical E. faecalis V583 VRE strain that we work with routinely in the laboratory, but it has specificity for this other host known as SF28073, as shown here. And so importantly, the one thing that she noticed about these bacteria is that they have very high congruence at the genomic level, like extremely high. We're talking 97% identity at the nucleic acid level of their genomes, suggesting that the genomes are, they're very closely related. But what was really determined to be different between these two organisms were the sets of plasmids that are endogenous to these bacteria. And so a hallmark feature of the enterococci are the fact that they do carry a wide array of both species-specific plasmids and also broad host range plasmids. And specifically species-specific plasmids that belong to a particular class of conjugative elements known as pheromone-responsive plasmids that can be basically conjugated within the species and in some cases outside of the species and even outside the genre. And Sydney took advantage of the large collection of enterococcal isolates that we have in the lab, and she made the hypothesis that if one of these plasmids is potentially driving this restriction mechanism of phage resistance in V583, then if she actually tries to use this phage to infect a plasmid cured derivative of V583, she should get successful plaquing on this organism. And so she set this assay up, and this is exactly what she saw. So you can see here, the phage at about 10 to the 8th PFU per mil, it has no ability to plaque on our lab type strain V583, but on the plasmid cured version of V583 called V19, she gets similar plaquing to what she sees for the cognate host SF28073. So because these are pheromone responsive plasmids, these plasmids can actually very easily be remated back into V19, specifically using various different types of selective methods that we use in the lab for plasmid mating. And she'd made 2 different derivatives, a V19 carrying this plasmid known as PTEF1, and then a V19 isolate that carried PTEF2. She reinfected these, and what you can see is that when V19 carries PTEF2, they're now fully resistant to phage infection. When they carry PTEF1, they're not. There is a third plasmid in this system. This is not a mobilizable plasmid as far as we can tell. And fortunately, the mechanism doesn't appear to be on that plasmid, so that was her good luck. Okay. So we wanted to get down to, well, what's the mechanism of this restriction? So the first thing we asked was, well, do the phages adsorb differently to the surface of these bacterial cells? As I showed you before, there's different carbohydrate moieties that dictate phage infection, and the EPA molecule itself is actually particularly important for phage adsorption to the cell prior to engagement with a canonical receptor for DNA entry. And so basically we just asked the simple question, do the phages adsorb equally well to these different strains? And is that potentially one of the reasons that, we don't see as much infection? And that's not the case. So you can see that phages adsorb to the surface of these bacterial cells equally well. So we know it's not a particular adsorption problem leading to potentially DNA entry into the cell. So the plasmid PTEF2 itself is quite large. It's about 58 kb, and it has roughly about 70 open reading frames on it, roughly. The vast majority of these open reading frames are hypotheticals, which doesn't, give us much to go on. But the one thing that we did know is that PTEF2 is actually very closely related to another pheromone responsive plasmid found in Enterococcus known as PCF10. And so here I'm just showing you an alignment of PCF10 with PTEF2. And these regions in blue here actually show regions of the plasmids that are between 95 and 97% identical at the nucleotide level. And we use this as a proxy to determine what genes to go after in terms of targeting these genes to identify the resistance mechanism. You can see that there's 2 variable regions of genes that are different between PCF10 and PTEF2. So Sydney set up a CRISPR-I screen where she basically knocked down these variable genes from PTEF2, and you can see here that she gets 2 hits in these open reading frames that we call 58 and 59. And importantly, if she goes in, she makes a clean deletion in this open reading frame known as 59, either in the V583 background itself on PTEF2 or in V19 that's carrying PTEF2, that you can actually restore phage infectivity. So it looks like this is the gene that's actually conferring the restricted mechanism. So what are these proteins? Well, so 58 actually encodes a serine recombinase that has similarity to resolvases that are involved in recombination. And then 59 has domains that are associated with type 4 restriction enzymes, specifically this GMRSD found in E. coli. And it's also has a nuclease domain in a different type of restriction enzyme known as SSPE. So it looks like we're dealing with a type 4 restriction enzyme here that is mobilizable on this plasmid. So as I showed you before, Sydney was actually using a lot of pretty low, I would say low titers to do these initial experiments. So she actually upped her titer and actually asked whether there were phages that could escape the restrictive mechanism. Occasionally she would get a handful of plaques. So she actually pulled some of these plaques, sequenced them, and it turns out that there's a single SNP in, I think, 5 different plaque isolates that she pulled from these. That's a G to A allele, G to A change that changes the amino acid from a leucine at 32nd position to a phenylalanine in this hypothetical protein called ORF65. Now, ORF65 has no known function, but it appears that this protein might be involved in blocking the restriction enzyme's activity. So what we did is we cloned this open reading frame, either with or without the mutation, and asked whether this was capable of overcoming the phage infection phenotype. And you can see that if the mutant version of the protein is expressed in cells alone without the plasmid at all, you can restore phage infectivity. And importantly, if we do a pull down, and this is pretty preliminary at the moment, but we're still working it out, you can see that the L32F ORF65 actually pulls down more of the restriction enzyme based on normalized spectra from mass spectrometry compared to the wild type. So this suggests that this is particularly occluding the function of this enzyme. So what we think is going on here is that EF0059 is mobilizable. It's a restriction enzyme that cleaves phage DNA, and that there's a phage protein that when modified to have a bulky phenylalanine on that protein can actually occlude probably the active site or some other aspect of the restriction enzyme, rendering it non-functional. All right, and so the final thing I'd like to say is just kind of looking forward. So I mentioned this resolvase, right? This resolvase is very specifically associated with this restriction enzyme, and they're both on the same transcript. When we actually dig into the genomics of this enzyme, we can see that this enzyme always tracks specifically with different types of anti-phage systems on Ephicalis plasmids. So we think that there's a very specific reason that this resolvase is actually associated with these anti-phage systems. And importantly, they're always, in most cases, flanked by transposases. So what this tells us is that these are probably mobilizable cassettes that can move around amongst plasmids to diversify the repertoire of anti-phage systems to kind of hedge your bets depending on what environment you're moving into as this bacterium. So with that, I'd like to thank you for your attention. And I'd, of course, like to thank my laboratory for all of their hard work. And of course, all of my collaborators, very specifically Kelly Palmer, Paul Carlson, and John Whitney, who contributed to some of the work that I showed you today. And then finally, the funding that makes all of this possible. Thank you.
Thank you very much, Greg. That was terrific. And we have, I will need assistance here if we have any questions from the room first.
Just let me.
Yes, Sergio, one question.
All right, please go ahead.
Great talk. I was wondering for the hypothesis that competition is limiting phage resistance in Ephicalis, whether you think it matters what particular members are present, or do you think even having one member would reduce the amount of resistant bacteria that are emerging?
Yeah, I think that's a really great question. You know, I think so. Those are things that we need to do, and we just haven't done them. So I think, you know, it may have to do with the composition of the community that's present. I do think that it's probably going to take more than just one single organism to really outcompete Enterococcus based on what I know about doing co-colonizations and things like that, that these organisms are usually pretty robust in their ability to coexist with other bacteria. Now, whether there would be more nuanced changes in those colonization levels, dependent on the organism that's there, is yet to be seen. But these organisms are not particularly good at utilizing complex carbohydrates and other things that are part of the microbiota, the nutrient sources in the microbiota. And so typically I think they would get outcompeted probably pretty rapidly by a more diverse community. And that is even the case for wild type enterococci. They can be depleted pretty well by just a traditional, fecal transplant. From a healthy, microbiota donor.
Great, thank you. Any other questions from the room or the chat? Breck, all the analysis that you've done is with individual species. Do you think that the resistance or susceptibility to phage infection can be modified by other components of the microbiota? And if so, how would you approach it experimentally?
So let me see if you're saying like if we were to, well, I guess I'm not really sure. You're talking about the first situation, right? Like if we're just using, phage infection to decolonize? Right.
So if you have, you can transfer presumably resistance or susceptibility from one species to another, and how do you locate them if you have a complex mixture of organisms or a complex community?
Yeah, I mean, I guess I think the way I look at it is you're going to know the individuals that have that have been densely colonized, right, by Enterococcus, that are undergoing broad spectrum antibiotic therapy, or they have other underlying issues, right? That's usually where these organisms manifest themselves as high density colonizers in the intestine, that make up greater than 90% of the microbiota. So in that situation, I think what you would want to do is you would probably want to use phages in combination with probably a microbiota, like a fecal transplant, right? To push out any organisms that might overcome the phage therapy as kind of a second layer of treatment. So kind of thinking of it very similarly how we think about phage antibiotic synergy, right? Antibiotics work really good with phages in synergy or in combination, better than those two entities work alone. And I think you could potentially think of it as a combination type therapy like that. I don't know if that answers your question or not.
Yeah, no, that addresses it. Thank you. All right, so thank you so much, and we'll move on. Our next speaker is Dr. David Pride, and David received his PhD in microbiology and immunology from Vanderbilt, and his MD at New York University. He currently works as the Director of Clinical Molecular Microbiology Lab and Associate Director of the Microbiology Lab at the University of California, San Diego. He leads his lab where he researches interactions between phages and their host bacteria for phage therapy. His talk today will be potential roles for bacteriophages in shaping the human microbiome. Maybe please go ahead.
Great. Thank you so much. Let me just share my screen real quick. Okay. Thank you so much for your attention. It's really a pleasure to be here. And of course, thank you everyone for staying to closer towards the end of the day to see this talk. So I'm going to talk a little bit today about some of the work that we've been doing, really to try to understand the role of viruses in general, but really more specifically bacteriophages in shaping the human microbiome. So I think, as we have heard quite a bit today, we really are an organism that is full of microbes. There are tons of bacteria, as we've heard, that inhabit different parts of the human body. what we tend to focus on is the fact that not only do we have many bacteria that inhabit the human body, but we estimate that there's at least 10 times as many viruses that inhabit the human body. And wherever you're able to look in the human body and find bacteria, we believe you're able to find viruses. We even believe you're able to find viruses in places where there aren't robust bacterial communities. So we've been focusing on viruses rather than the sort of more cellular communities like the bacteria, but we focus on many of the same questions that people ask about bacterial communities. For example, like what bacteria constitute the microbiome, what is their role in different parts of the body, how do they respond to perturbations such as antibiotics, and ultimately, what is their role in health and disease? So we've been trying to answer or get at some of these sorts of questions, but taking a more virus-centric point of view. And just one sort of caveat, I tend to use the term virus and bacteriophage a bit interchangeable. So generally today, when I say virus, I'm really talking about bacteriophages. So my laboratory has been working really for probably the past 10 or so years now on some really fundamental questions about the human virome. We've started to get some answers for some of these questions, and we're still really working on others. Some of those questions really are, how do viruses differ between different individuals? How do they differ based on the body site that we're looking at? Ultimately, because I am a clinician, one of the things we really worry about is our propensity to share our viruses between individuals. So one of the things that my laboratory has really worked hard on is trying to understand, can the viruses that inhabit your microbiome be quote unquote contagious or not? And then lastly, we're really focused now on trying to understand how we can measure the effect of these viral communities on our resident cellular bacterial communities. So one of the studies that we performed was really trying to answer a lot of these questions that I sort of have already brought up. And that is, is there, for example, individual specificity, and is there body site specificity of the viral communities that you see in humans? And we did this study that we called the campus antibiotic study, even though the antibiotics really isn't relevant to what we're talking about today. And we looked at a bunch of different households here on the UC San Diego campus, and we enrolled 2 individuals, with the caveat being that each of those individuals had basically just moved in together. So they were basically college roommates. And one of the striking things about looking at their viromes, and we looked at their oral viromes and their gut viromes, what you're looking at right now is a representation of their gut viromes. And what we find is that we can tell who an individual is based on the composite of viruses that are present in their gut. And we can tell that not just today, but we can tell it, a couple of months from now as well. In other words, at every single time point where these individuals were housed together, we could tell exactly who they were just by looking at the viruses in their gut. So it's something that could be a wonderful forensic technique. Unfortunately, it's pretty cumbersome and relatively expensive, so it probably would never actually be a forensic technique, but it's nice to know that we can tell people apart just based on looking at the viruses that are present in their gut. But if we take that exact same data and we look at it in a slightly different context, one of the things that emerges is that we see these household patterns. And we see those household patterns really emerge quite quickly and be stable really throughout the study. And while there are 2 sort of panels here, I would probably just pay most attention to panel A. And as we had mentioned, you can tell who an individual is just based on the viruses in their gut. What we hadn't necessarily anticipated is that not only could we tell who that individual is, but we could tell who they live with just based on the viruses in their gut. So that was really surprising to us, and it was surprising how statistically significant that was and how quickly those patterns emerged. And we went on really to demonstrate that we can that we see this phenomenon because individuals share viruses with one another, and they share viruses relatively rapidly, and that sort of creates the patterns that we observe, and that some of those viruses that they share become fixed members of their virome communities as well. One of the other things that we really have been doing is we've been characterizing viromes really across a bunch of different human body sites. And while that in itself, it may not be the most interesting question, but in particular, because we were using a single technique, it allowed us to compare all of our viromes with one another. And as those who work in a virome field may realize, there are a bunch of different techniques by which you can purify out and sequence viromes, and comparing across techniques is going to lead to certain biases. Using even a single technique will lead to certain biases. However, because all of these viromes across saliva, dental plaque, feces, urine, breast milk, cerebrospinal fluid were all processed using the same technology, it really is more of an apples to apples comparison that we have. And we found that there are certain body sites that tend to have relatively distinctive features when we compare them with other body sites. So for example, in our analysis, when we looked at the distal gut with feces, we could find that the distinction was there are a lot of microviruses there, and we didn't find microviruses in a lot of other sample sites. When we looked at saliva, we found that a lot of siphoviruses, and we believe that that's why a lot of those viruses are just so persistent, and largely because we believe that a lot of those viruses tend to have more lysogenic lifestyles. In the urine, we actually found a pretty good amount of herpes viruses, in particular in people who had never been diagnosed with herpes viruses infections. We found a good amount of rhinoviruses in joint fluids and gallbladder fluids. And in breast milk, we found a lot of myoviruses, which do tend to be lytic, but aren't necessarily lytic, and that was really a distinctive feature of it. Interestingly, in the blood and the cerebrospinal fluid, we did not necessarily find a lot of distinctive patterns, and we believe that that's because the viruses that you have in your body aren't necessarily, or the viruses that you have in your blood and CSF get there largely through what we believe is transcytosis across barriers. So they end up being more of a compilation of different body sites, and that might be why we're not observing those really distinct patterns. So it kind of brings us to a question, now that we sort of characterize all of these viromes, we can see that they're shared, we can see that they're individual specific, how do we measure the impact, right? If most of what we can identify are bacteriophages, what are the bacteriophages doing? Probably the easiest answer that most people would arrive at is they're probably killing the bacteria that are there, or bringing in new gene functions that can shape the natural history of those communities. So we partnered with Dr. Emma Allen Vercoe at the University of Guelph, and she's got what we think is really a wonderful system that they call the RoboGut. It's a system that's actually now been around for a little while. And the distinct thing about the system is that you can basically put feces into it. And when that system reaches a steady state, which is about 24 to 30 days later, the microbial community looks a lot like that feces, which is really kind of amazing. It's one of the reasons that I personally stopped using the term uncultivable a long time ago, because many of the organisms we thought were uncultivable, we could actually find in these systems, because if you put them in the right context where you're, for example, controlling temperature well, controlling pH, controlling all the nutrients and the oxygen, et cetera, you end up with a different compilation of microbes than you do in a lot of other systems. But we're not really a bacteria lab. We're a bacteriophage laboratory. And our large question about it was, well, you know, if these systems are reproducing the bacterial microbiota really well, maybe they're reproducing the viral biota really well as well. And it's one of those things where these systems have been around a long time. It's sort of surprising that that question hadn't really been asked or at least explored with any depth. So years ago, we sort of looked at these systems and we just did a simple stain looking for virus-like particles. And we were surprised to find there were about 10 to the 9th virus-like particles in these systems, which is really the same magnitude that you find in feces. With the take-home message being that these systems are full of viruses, which likely are almost all bacteriophages because they're meant to grow the bacteria and thus probably other phages as well. And if we just look, we did really just simple profiling of these based on blast X hits to start. As a technology when we did the study was a little different than say the technology is now. And what we found is that these things don't look anything like feces on day 4, day 8, day 12. But by the time you hit day 24, when they hit steady state, boy, the profiles start to look a lot like that of feces. So we were really starting to get confidence that when these systems hit steady state, the viruses that really are very similar to the viruses that you would find in someone's fecal material. And we also look at these via a principal coordinates analysis. And that was really important for us because, you know, we looked at a bunch of different subjects. Here, there's 5 different human subjects. And we can see that in every case, the compilation of viruses that were reproducing in these roboguts always clusters with the actual feces of that individual. For this one, it's not in 3D, so you can't necessarily tell, but they are clustering separately as well. So we feel really comfortable that we can reproduce individual specific viral biota basically in these robogut systems. And why that's important for us is it means that we can simulate viral transfers or bacteriophage transfers between these systems. And it gets back to us specifically looking at the idea that you know, bacteriophages can be quote unquote contagious. And we had been looking for a way that we could kind of test that what might happen when we share bacteriophages, because we've already shown that that's something that happens probably in nature just about every day. Because we can reproduce these viromes in a test tube now, we can specifically purify out those viromes and we can transfer them to the bacterial biota that are reproduced from other individuals. And while it's certainly not a natural system, it gives us a sense as to what happens if I just take someone's microbiota and take viruses either from that same person or from a different person and put it into that system. What sort of perturbation are you going to see, or are you going to see a perturbation at all? So in this study, which unfortunately got affected significantly by the COVID pandemic, but did recently come out, we took 2 different subjects, which are D5 and D25, and we purified out all of the viruses from these D5 vessels. And then what we did was we grew up these D5 vessels. We perturbed D5 with its own phages, largely as a control. And we also perturbed this other subject, which was D25, with those viruses as well. The caveat here being that we perturbed with a heat-killed version of the viromes, and we perturb with the live viruses as well, with the idea in mind that heat-killed shouldn't do anything. And the live viruses certainly would have a much greater propensity to impact the communities because they might find hosts and start to kill them. We did some electron microscopy of the viruses that we purified out. The vast majority of what we could actually see certainly look like they're viruses, which isn't a surprise coming from the stool. When we sequence these, we see that they are much more complex than just all viruses. But it's nice to know that a large part of what we're transplanting between individuals are viruses. And when we look at just alpha diversity, what happens to the bacteria in these communities over time, we're looking here at the Shannon and the Simpson index. I would probably just pay attention to the Shannon just because they, in essence, are showing the exact same thing. When we look, for example, at D5 heat treated, we don't see much change over time. When we take D5 and we treat it with D5 viruses, so in other words, you're just taking the same individual, purifying out a bunch of phages, and then hitting it with what are probably pretty high levels of the phages that it already sees, we do see an increase in diversity over time. When we look at the intersubject transfers between from D5 to D25, we don't see a whole lot of change in diversity when the heat treated are used, but we see significant increases in diversity when we use the live viruses from subject D5. When we look at each of these vessels, the one thing that is that becomes fairly apparent when we start to look at how much change, in essence, or what's the magnitude of change we're observing in these vessels over time, we certainly see the greatest magnitude of change in subject D25 when subject D25 receives live viruses from subject D5. Coincidentally, the lowest magnitude of change happens when D25 gets the heat-treated viruses from subject D5. We can certainly see differences when we just look, for example, via principal coordinates in these different vessels when we treat these systems with these D5 either live or heat-treated viruses. So we can see that there's something different in the microbiota that we can observe really in both systems based on whether or not we use live or heat-treated viruses. And to some extent, if we just look at a very high level at the microbes that we see change, we do see some changes in subject D5 when subject D5 gets live viruses from, in essence, itself. And again, I don't view that as terribly surprising. You take an individual, purify out its phages, and then give it back a bunch of its phages, you're probably going to see some differences. But those differences are sort of overshadowed by what we see in subject D25 when it gets live viruses from subject D5. We see significant decreases in Bacteroidetes. We see significant increases in firmicutes. We don't see much change in Microbiome and proteobacteria, but we also see significant increases in actinobacteria. Again, strongly suggested that these live phages, in fact, when given at these levels, are perturbing these systems. And again, you know, this is something that could potentially occur on a daily basis, but it's unlikely that we are receiving sort of the high levels of phages that we transplanted between these different vessels. So we might want to go back and take a look with when we're transplanting, in essence, much lower abundances of these bacteriophages. Even when we take a look to see, hey, what differences are we observing in these vessels that are getting, the live viruses, we can certainly see For example, when subject D5 gets its own live viruses, we see changes in ruminococcus, and we see changes in Bacteroides as well. And then when we look at subject D25, which again is getting those inter-subject transfers, we see a number of different microbes that change, including Lactobacillus. So there's certainly, the unfortunate part about these experiments is that we don't know exactly what phages are causing what change. So to some extent, it is a little bit nebulous, but we do have a strong sense as to what microbes or what taxons are changing, and that will allow us to be able to, in essence, dilute out and identify the exact phages that are the source of the perturbations that you can observe. The other thing that we're really interested in are the effects of diet, specifically on virome contents. And we did a set of experiments where we took a group of mice and we transitioned their diet at about 16 weeks, just asking the question, what happens if you take a mouse, you put it on a normal diet, and then you transition them to a high-fat diet? What we find is that the virome really kind of mimics what you can see in the microbiome. We look specifically at the cellular biota, looking at the 16S for the bacteria. And the viromes, we get really good definition where we can see at 4, 5, and even 15 weeks, the viromes are somewhat similar. But when we transition to a high-fat diet, we see really stark changes in the virome. And by week 28, those viromes are really significantly different. When we ask the question, what is different in those viromes? Well, the one thing that we see, as we had mentioned, there's a lot of cyphoviruses in human feces. Those cyphoviruses go way down when we transition the diet, and they tend to be replaced by microviruses that we had sort of mentioned already. We also see some other eukaryotic viruses that sort of start to pop out in these mice as well. But for us, that really kind of raises a question, and it kind of gets to almost like a chicken and egg sort of question. What's happening first? Are the bacteria changing, and thus the viruses are just a reflection of that, because maybe they're more lysogenic or temperate bacteriophages that are present? Or can these phages themselves change and then drive the changes that we see in the microbiome? So it's a question that we've been trying to figure out the answer to, and it gets at almost a little bit of a larger question. If you guys remember that experiment by Jeff Gordon's group, where they had shown that if you take a mouse, I think he was using germ-free mice, and you feed them different diets. One gets a high-fat diet, one gets a sort of a normal chow diet. Of course, the ones that are on the high-fat diet will get fat, as we sort of shown in the last study. But if we take the fecal microbiota and we transplant them into mice, feed them a normal diet, those that got the high-fat microbiota tend to gain more weight, with the idea in mind being that the way the microbes are processing the nutrients that you're feeding them really can affect the obese and lean phenotypes in mice. One of the things that we tend to think about with that is, well, what's going on with the viruses? Because that set of experiments was done a long time ago now, but it obviously couldn't account for viruses at the time because we weren't really even interested in viromes at the time. And for us, the question is, are viruses sufficient to sort of be able to give us or reproduce some of those phenotypes? So we put together a set of experiments with a group of mice on a high-fat diet, a group of mice on a normal chow diet. We purified out all the viruses, and then from a dummy set of mice, and we just gavaged them over time and just asked the question, do we see phenotypic and do we see differences in their fecal microbiota. So for example, mice on normal chow, some of them got normal chow gavages of phages. Some got high-fat diet gavages. Mice who were on high-fat diet, some of them got normal chow gavages, others got high-fat diet gavages, etc. And what we found were actually highly significant differences in phenotype. So for example, those mice who were fed normal chow, who received high-fat diet, you can see gain more weight. And unfortunately, I don't have stars on here, but these data are really highly significant. Gavaging them with high-fat diet bacteriophages really causes significant change in the weight gain phenotype. The same thing for mice who got high-fat diet and were gavaged with high-fat diet phages. Those who got who were on a high-fat diet and got chow gavages gained less weight. Again, it's sort of indicating that these phages are somehow interacting with the bacteria, and that over time is helping determine these lean and obese phenotypes. And we can look at these. I will say that all of this data has been replicated. So we actually did what we call trial 1 and trial 2, or just replicate 1 and replicate 2. And we basically found the same data in both replicates. So in other words, we can see the weight gain phenotype replicable in each of these trials. And also we can tell the difference between which set of gavages you got just by looking at it on principal coordinates. And the differences again are statistically significant, and we can see both the gavages and the if we look at diet and gavage combined, both really are highly significant in terms of the data, indicating that there's definitely something going on in the gavages that these mice are getting. When we look at individual groups of these mice, so in other words, those that got a chow diet, what microbes are changing when we give them, for example, a high-fat diet. We certainly see changes in lactospiraceae, which is something that we had observed earlier, and we're kind of trying to work out and titrate down to see if we can figure out the individual viruses that may be responsible for these sorts of changes. The same thing if we look at mice on high-fat diet and we gavage them with chow diets, we see changes in these Myribaculaceae. You know, we've got sort of our work cut out for us in terms of understanding which microbes may be responsible for some of these reproducible phenotypes, but really the take-home message is that it appears that gavaging with these bacteriophages really is sufficient to recapitulate some of these lean and obese phenotypes. So just in summary, we know that most, if not all, human body surfaces harbor these viral communities, of which we have shown a bit. We know that most of them that we can identify are bacteriophages. They are shared and transmissible between close contacts. We can model both bacterial and viral communities in these chemostats or robogut systems. And we know now that we can utilize them to sort of help assess the role of bacteriophages in modifying bacterial communities. And lastly, there are specific differences that we can identify in the viromes of mice on high-fat and normal chow diets. And we now know that gavages with these bacteriophages are sufficient to reproduce at least some of the lean and obese phenotypes. So I'd just like to end by acknowledging all the folks in my laboratory who contributed to all of these research projects, as well as our funding sources and collaborators. And if there's time, I would take a question or so. Thank you.
Thank you so much, David. That was a terrific talk. I really enjoyed it. We may have time for one quick question in the interest of trying to keep up with our schedule. Kristen, is there any question in the room?
Yep, Sergio, one question from Ben.
Great, please go ahead.
Thanks for a great talk. I was wondering if you could just talk a little bit about kind of the kinetics and the kinetics of kind of transfer between people. And obviously you're kind of thinking about this in maybe a clinical setting with patients and things like that. But just kind of transfer of the microbiome and the virome and other things like that. So if you could talk about that for a little bit.
Yeah, I wish that was something that there was really a simple answer for. You know, as I recall, doing that particular study, again, this the campus study where we put people in a house together and examine their viromes. The one major regret we had about it is that for not all of them were we able to get sampled before they moved in, and that of course became a problem. We could easily identify, for example, one phage go to the next person, you know, at I know we did 8 weeks and a time point before that, but it looked like a lot of the transfers were occurring probably within a matter of days, and we just didn't sample enough early on to be able to understand what those kinetics are like. So if we had to do it all over again, absolutely we would try to get them, one, before they moved in together, and two, multiple times a day during the first few days of the study, because it looks like that may be all it takes for not all, but maybe the majority of these transfers to take place.
Thank you so much. I think there's one more question.
Yeah, Sergio, we have a question from the chat from Breck. Very nice results from the high-fat diet work. How do you uncouple phage-specific effects from those effects that might be mediated by small metabolites that could co-purify with LVPs?
Yeah, I mean, that's a really good question, and that's something we sort of have wondered about really ourselves as well. So, you know, our phage purification protocols are pretty harsh, to be honest with you. So we're actually using PEG purification, and we're going through multiple rounds of purification. So it would be fairly difficult through all of the purification steps that we take to be carrying over metabolites into the mice. But again, it's certainly not impossible, but it would seem to be hard. With that said, you know, the heat treated being different from the heat kill being different from the I guess the live that we have observed in our other system gives us a little bit more confidence as well, at least you know, unless the metabolite is of course heat sensitive. So you know, it's a difficult question for us to answer, and the other thing that to some extent might even support what you're saying is that particularly in our in vitro systems, the second we put our, even though we go through multiple rounds of purification, the second we put these phages in, we can observe a phenotypic difference even in these chemostats where they just start bubbling up, and it's kind of wild to watch. You know, so we definitely have wondered whether or not we might be putting some type of metabolite in there, and we've got some ongoing studies to try to figure that out to some extent. We haven't really found anything just yet. You know, maybe the bubbling really is, you know, some type of phage host interactions. We just don't know.
Thank you very much, David. That was wonderful. Okay, so we'll now move to the last talk of this symposium, and I'm very happy to introduce Mariana Beindlos, who works in pathology, microbiology, and immunology. She received her veterinary medicine degree and PhD from the Universidade Federal de Minas Gerais in Brazil, and she completed her postdoc at the University of California, Davis. She currently leads her lab at Vanderbilt University Medical Center. Her research focuses on how inflammation-mediated changes in the gut epithelial metabolism leads to gut dysbiosis and an increased risk of disease. She has over 70 scientific publications, and the title of her talk is Disruption of Microbiota and Intestinal Epithelial Interactions as a Key Driver of Non-Communicable Diseases. Mariana, the floor is yours.
Thank you. I'm share my screen. Actually, I apologize in advance because I decided to change my talk last minute because I was actually very excited about this is going to be all unpublished data from my lab, and I figured that this would be the best audience to actually present this data for the first time. So this is actually the first time I'm presenting this data to an audience, and any feedback would be very much welcome. So we're still talking here about, and thank you obviously for the organizers for the invitation. So here, we're still gonna talk about disruption of microbiota and intestinal epithelial interactions, but I'm gonna focus today a lot on more in the context of what happens during early life and how that may lead to obesity. So my lab is really, in general, is really interested in understanding how the host and the microbiota are talking to each other to promote host health. And in particular, we're interested in the microbiota and intestinal epithelial cell crosstalk and how that promotes health, and also what happens when this beneficial interaction gets disrupted through infection with enteric pathogens or through environmental factors such as diet and antibiotics, and how this disruption may lead to disease. And this talk today is definitely going to focus more on environmental factors. And we are very interested in obesity and how changes in the microbiota can lead to obesity, because in a world without COVID, imagine, right? It would be awesome. Obesity was considered the epidemic of the 21st century. And you can see here that in the 2000s, about 30% of the US population was considered obese. This number went up in 2019 to about 30 to 40%. And the prediction is that nothing is done, nothing changes, about 30%, about 60% of our population, especially in the South where we are, Vanderbilt is gonna be obese. So definitely there's urgency in understanding the factors that drive obesity. And not only that, if we look particularly in childhood obesity, we'll see that about 20% of the US population has childhood obesity, right? And the number again is much higher in the South. And children can become obese through multiple factors. Obviously, being sedentary and watching too much TV can cause obesity, but two important factors that drive obesity is dietary habits and exposure to antibiotics. And work by other groups have really shown that both subclinical dose exposure to subclinical doses of antibiotics or intermittent exposure to clinical dose of antibiotics can drive obesity. But one important aspect of most of the studies that have been performed so far is that they really focused on how one individual risk factor can drive obesity, right? And that makes sense because it's much easier to understand when we single out one risk factor to understand how that is impacting health. And through those studies, it's been shown that dietary changes drive obesity, and antibiotic use also drive childhood obesity. But what we notice is that if you think about the epidemiology of obesity, we start to see a pattern, right? Here is the graph for childhood obesity, and you can see that the areas in the country that have high childhood obesity are also areas in the country when there's increased consumption of fast food, and in areas where what they call supermarket deserts, right? So when people have to drive a lot to get fresh food. And not only that, all those areas are also mirrored by the areas in the country in which antibiotics are prescribed the most. So based on this idea, we start to think about what happens when we expose young individuals to more than one risk factor at the same time, right? What happens when you expose young individuals to maybe a high-fat, obesogenic diet and antibiotics at the same time? So that was a simple question that our lab started asking. And that's the work by my PhD student, Katie Shelton, which is a fantastic PhD student in the lab. So the model of antibiotics that we chose to use was a model that has been published by the Blazers group, which is exposing animals to a low dose, subclinical dose of penicillin. And that's because other groups have shown that this drives obesity. So what we decided to do is get young mice, so they're at weaning, and follow them to they're about 8 weeks old when they're actually kind of becoming adult, right? So what happens when you expose young mice to these environmental factors for a certain period of time? And what we decided to do is put the mice in a control low-fat diet, expose the mice to penicillin, expose the mice to a high-fat diet, and do this combination of high-fat diet and low-dose penicillin. And for the interest of time, I'm not going to show the data with the clinical dose, but all the results that I'm showing here today could be replicated if we did clinical doses of penicillin, so pulses of clinical doses of penicillin. So the first thing that we decided to do is the combination of high-fat diet and antibiotics drive obesity, right? So you can see here is that indeed the mice that were exposed to high-fat diet and antibiotics at the same time, they gain weight much faster than the other mice. Not only that, we also measured fat, and what we see is that it mirrors the weight gain. The mice that are exposed to high-fat diet and antibiotics, they have increased fat accumulation. And what is interesting here, and I wanted you all to note, is that in our hands, antibiotics alone do not drive obesity. And we also decided to see what's happening with glucose levels in the mice. And again, the data mirrored what we had seen with fat accumulation, is that mice fat exposed to high-fat diet and antibiotics together start to see an increase in glucose and fasting glucose levels much earlier than any of the other groups. And the last data that caught our attention is that when we looked for fat accumulation in the liver of the mice, you can see here that antibiotics alone in the background of a low-fat diet did not increase fat accumulation in the liver. The high-fat diet started to see a small accumulation of fat in the liver, and that was amplified in the group that received both antibiotics and high-fat diet. And to really see if this exposure was if these effects were long-term, we did an experiment in which we exposed the mice to the combination of high-fat diet and antibiotics, and then we followed them for an additional 5 weeks by removing the antibiotics but keeping them in a high-fat diet. And what you can see here is that this effect is long-lasting. Even after you remove the antibiotics, the mice that were exposed to the combination of risk factors, they gain more weight and they still have more abdominal fat, even 5 weeks after. So together, this initial data was suggesting to us that the combination, when you're exposed to more than one risk factor at the same time, there's an increased risk for obesity, at least in the mouse model, right? So here we're talking about mice. But the question here is why? Why is this happening? And this is a microbiome symposium. So of course, the first thing we thought about is microbiome, right? Because we know that microbes can play an important role in obesity through different mechanisms. And for us, we're particularly interested in this altered metabolic signaling. And we're also interested in the microbiota because preliminary data in germ-free mice from our group suggests that the phenotypes that I showed you earlier, they're not present in germ-free mice. Really making us start to think about that this could be a microbiota-driven effect. And what we also knew is that from beautiful work from other groups is that antibiotic treatment alone and exposure to a obesogenic diet alone can alter the gut microbiota composition. So we wanted to understand what happens to the microbiota in animals that are exposed to both antibiotics and a high-fat diet. So what is important here is that when we start doing this study, we decided actually to focus on the small intestine microbiota. And the reason is because we know that the small intestine is the site in which there's more absorption of fat and lipid metabolism happening. So we really wanted to see what is happening to the small intestinal microbiota in our model. This is just a simple graph just to show overall the major changes that we saw, is that actually antibiotics alone don't seem to change the microbiota that much in the background of a low-fat diet. But mice fed a high-fat diet, we actually start to see the microbiota change, and that has been shown by other groups too, right? And we see an increase in lactobacillus in terms of relative abundance. And actually, this increase in lactobacillus goes away in mice fed penicillin. And And we actually wanted to confirm that this was not just a decrease in relative abundance of lactobacillus. So what we did, we used a MRS agar, and we selected lactobacillus, and we plated lactobacillus from the small intestine of those mice. And we saw that the data replicates what we've seen in by 16S sequencing, that indeed in the combination of high-fat diet and antibiotics cause a significant depletion of lactobacillus in the gut microbiota. So this, I wanted you guys to remember this data because I'm gonna go back to this later in my talk. But then we knew that the microbiota was changing in our mice, but that can cause obesity for multiple mechanisms. But one specific data set kind of gave us a hint of what may be happening. What we saw is that the mice that were given a high-fat diet and antibiotics, they had significant higher levels of lipids circulating in their serum. So they really have a high abundance of lipids. And that is when we went back to our favorite site, which is the intestinal epithelium, right? So that since the intestinal epithelium in the small intestine in particular is a site in which lipid is absorbed and metabolized and secreted, we thought that that could be an indication that this, the microbiota and intestinal epithelial interactions were disrupted in our model. And we actually looked into what's happening to the small intestinal epithelium in these mice. And what we saw is that specifically mice fed a high-fat diet and giving antibiotics, there was disruption of the intestinal epithelial morphology with decreased length and an infiltration of lymphoid cells that we can actually see here by histopathology scoring. So that data prompted us to see what are the major changes in the intestinal epithelium in these mice. So what we did RNA-seq, specific in the epithelial cells. And just to summarize, we obviously saw huge changes in different genes, but one group of genes that caught our attention was genes related to lipid transport and metabolism. So you can clearly see that multiple genes related to lipid transport and metabolism in the intestinal epithelium were upregulated in mice that were exposed to a high-fat diet and antibiotics. And we confirmed that by qPCR. So based on this data, with the amount of genes that were altered, we started to think that maybe specific pathways that control so many lipid genes could be affected in our model. And that's when we start thinking about PPARs, or peroxisome proliferated receptors, because they are major regulators of lipid metabolism in multiple cell types. So we're particularly interested in PPAR gamma because work from our group and other groups have shown that microbial metabolites can regulate PPAR gamma function. We've shown the short-chain fatty acids in the large intestine regulate PPAR gamma function, but it's obviously a chance that unidentified metabolites also do, especially in the small intestine. And we also shown that antibiotic treatment actually decreases metabolites that affect PPAR gamma function in the large intestine. So we went back to our mice, and the first thing that we wanted to ask is PPAR gamma signaling altered in our mice fed a high-fat diet antibiotics? We looked at it different ways, but this is just immunohistochemistry. And to show that really in mice fed a high-fat diet and antibiotics, PPAR gamma signaling is significant decrease in the small intestine, right? We lose this PPAR gamma staining. So this was an initial indication to us that maybe PPAR gamma signaling in the intestinal epithelium may be driving our phenotype. So we wanted to confirm that this is happening. So we used a genetic model in which we deleted PPAR gamma specifically in the intestinal epithelium of the mice. And so this is the data just to confirm what we had seen previously. First, we put the littermate control flocks mice in a high-fat diet, or put the littermate controls in a high-fat diet and antibiotics. And we looked for weight gain and accumulation of fat. And what we see is that we had seen before that wild type, the control mice, the littermate control mice have increased adiposity, adipose tissue accumulation if exposed to a high-fat diet and antibiotics. But what's interesting is that when we put the PPAR gamma knockout mice, epithelial knockout mice in a high-fat diet, those mice became obese even in the absence of antibiotics. So they actually became as obese as the wild type mice that received antibiotics. And to really confirm that this was happening in the same pathway, we exposed the PPAR gamma knockout mice to antibiotics. And what we saw is that in that background, antibiotics did not even increase even further fat accumulation or obesity, suggesting to us that maybe the antibiotics are really acting through the PPR gamma pathway in the intestinal epithelium. And this was not because lactobacillus levels were different in the groups, because we confirmed that antibiotics reduced lactobacillus both in the wild type and in the PPR gamma knockout mice. So what this led us to think is that what's happening maybe here is that when young individuals are exposed to a high-fat diet, the microbiota has protective mechanisms that may activate PPR gamma in the intestinal epithelium, and that controls fat absorption, decreasing fat accumulation and obesity. However, when there's a combination of high-fat diet and antibiotics, maybe we're altering the microbiota in a way that these protective mechanisms are downregulated, leading to increased obesity. But the question here is how this is happening. And that's when I go back to the initial data showing the lactobacillus is decreased in the mice that have antibiotics and a high-fat diet. And we're particularly interested in this because lactobacillus are known probiotics, right? They are known to modulate intestinal epithelial function, and they're known to activate peroxisome proliferator receptors. So what we decided to do is go back to our mice and isolate lactobacillus strains from them and ask the simple question: can these strains activate PPR gamma in intestinal epithelial cells? And obviously that was a long story, but it turns out that we identified a strain of lactobacillus, which is now named Legilactobacillus murinus, that can actually activate PPR gamma in intestinal epithelial cells. And what was interesting here is that I don't have time to show the data, is that the same strain was able to reduce lipid uptake by intestinal epithelial cells in a PPR gamma dependent manner, suggesting to us that really somehow this particular component of the small intestine microbiota of young mice is activating PPR gamma and seems to be modulating fat absorption. But the question here again is how this is happening, right? So what we went back and we did an experiment in which we exposed epithelial cells to live lactobacillus strains and to the supernatant of the strains. And we saw that the supernatant of the lactobacillus strains had the same effect as the live microbe, giving us a hint that maybe it was a lactobacillus-derived metabolite that it was driving our phenotype. So to determine which metabolite it was, we went back to our in vivo model and we decided to run targeted metabolomics. And obviously what we saw is that a lot of metabolites were different between mice that young mice were exposed to a high-fat diet and young mice were exposed to a high-fat diet and antibiotics, right? You can see here. And we're still going through all this data, and I think there's a lot of interesting pathways that we could follow up. But one metabolite in particular caught our attention, which is phenylactic acid, or also known as PLA, which was significantly depleted in small intestine content of mice that have high-fat diet and low-dose penicillin. And this was interesting to us because PLA is actually a metabolite that is present in high abundance in fermented foods, such as kimchi and kombucha, that are thought to promote health. And not only that, this was interesting to us because we knew that our strain of lactobacillus that activates PPR gamma can produce PLA. So when we went back, and there's one paper published not too long ago that also showed that PLA can activate PPR gamma in adipocytes, but nothing was really known about PLA activating PPR gamma in intestinal epithelial cells. So what we did, we went back to our in vitro model and we simply treated the cells with PLA. And what we saw is that through immunofluorescence and other assays, is that PLA indeed does activate PPR gamma in intestinal epithelial cells, and it also helps modulate lipid uptake by intestinal epithelial cells, which is really interesting, right, to us. So the question then is like, okay, we've identified this particular metabolite that is microbiota-derived metabolite that is missing in the small intestine of mice that are exposed to antibiotics, and that may be contributing to this early accelerated obesity that we are seeing in these animals. So our question then is, could PLA then maybe prevent this antibiotic-associated early life obesity that we are seeing in our model? So what we decided to do, again, go back to our model and actually give PLA to the mice. And for most of my data, we were really focusing on mice that received a high-fat diet because we had seen previously that antibiotics alone didn't drive our phenotype, right? There was a phenotype driven by really the presence of increased fat. So we really focused on the combination of fat and antibiotics. So what we did, we supplemented PLA on the mice, and this is the data just to show you all that we're able to rescue the levels of PLA in mice treated with antibiotics here in the dark gray to the same levels of the mice given a high-fat diet. So the PLA supplementation did work. And we also wanted to make sure that this PLA supplementation was activating PPR gamma in the intestinal epithelium. So here's the data for mice given a high-fat diet of PLA. The PPR gamma is present in the intestinal epithelium. And this is what happens in the mice that are given antibiotics. PPR gamma, specifically in the intestinal epithelium, goes down. You can actually see what is interesting is that PPR gamma staining in immune cells is still present, suggesting to us that this is really driven maybe by intestinal epithelium specific. And we can see that, so this high-fat diet plus low-dose penicillin plus PLA, the labeling is wrong. We see PPR gamma light up again in the intestinal epithelium, suggesting that PLA is really activating PPR gamma in the intestinal epithelium also in vivo. But the question here is this affecting weight gain or adipose tissue, right? The position or obesity. The data that I'm showing here for adipose tissues is similar to what we see for weight gain, works the same way. So you can see here that PLA does not alter weight gain in mice high-fat diet alone, and the mice that are given antibiotics on a high-fat diet do have increased fat accumulation. And indeed, in the mice that are supplemented with PLA, this increase in fat accumulation goes away. Suggests that PLA is really preventing obesity. And it's not only preventing obesity, but it's also preventing fat accumulation in the liver of the mice. So this is another staining for fat deposition in the liver of mice, and the red is an oil red staining, so the red is really fat. And you can see here, as I shown you earlier, the mice that have antibiotics, they have a high-fat diet and antibiotics, have significantly higher levels of fat in the liver at a very young age, right? This is 8-week-old mice. We usually see that happening in much older mice. But what really happens is when the mice that were given PLA, this fat accumulation simply go away. This was honestly very shocking to me how black and white this phenotype is. And I obviously think that this is happening through the intestinal epithelium, but there's also a chance that PLA is affecting other cell types, and it'll be interesting to figure that out. So what we think is happening here is that we uncovered a mechanism by which the microbiota is, especially the small intestine microbiota, is protected in the context of a diet-induced obesity. So we think that when young individuals are exposed to a high-fat diet, members of the microbiota are producing metabolites that activate protective mechanism in the intestinal epithelium, in this case, peroxisome proliferator receptor gamma, that actually alter or controls fat absorption by the intestine, leading to decrease of fat accumulation and decrease obesity. However, when there's a combination of a high-fat diet and antibiotics, this combination causes depletion of specific members of the microbiota and specific metabolites, and the protective mechanisms are disrupted, leading to increased obesity. So with that, I wanted to thank the people that had done the work, especially Katie Shelton, who drove this project from the moment she was my first graduate student. She drove this from the moment that she started in my lab, and I'm very thankful for her dedication. And she was actually helped by 3 different undergrads. And our collaborators, our funding sources. And with that, I'll take any questions you may have.
Thank you, Mariana. That was terrific. And are there questions in the room, Christine? In the meantime, Mariana, you're talking about antibiotics in general. Do you know what happens if different antibiotics are given, and do they have different effects on PPAR gamma?
Well, so we haven't done that yet. So what we know from our work and work from Eric's lab, from Andreas Bromler's lab, from my postdoc advisor, is that streptomycin seems to affect it too, but we have not tested out antibiotics. What we do know is that this lactobacillus strain is actually vancomycin resistant. We learned that not too long ago. So I think it would be good to go back and do vancomycin treatment or other antibiotic treatments to see if they affect differently, but that would definitely be interesting.
Great. Christine, are there any questions?
Yep, I'm here. I'm here, Peter, or I'm here, Sergio. One in the back, Peter.
All right.
Hi, great talk. I guess I was wondering why you think the signaling is in the gut, and is it possible the phenolactic acid is absorbed and signaling directly on the adipocyte?
I think it's totally possible. So we focus in the gut because we've been studying PPR gamma for a long time. We focus in the gut because we didn't see changes in PPR gamma signaling in adipose tissue or in the liver. I didn't show the data here, but it seemed like it was really focused in the gut. That's why we focused there. But honestly, the PLA phenotype is so black and white that I would be lying if I didn't think that this metabolite may be activating, maybe playing a role in other cell types too. So we're repeating these experiments in the PPR gamma knockout mice, epithelial knockout mice, to see how much of this PLA signaling is through PPR gamma in intestinal epithelium and how much could be through in other cell types too, because definitely possible.
Is it known like how IEC, how does that pathway in the enterocyte communicate with the fat tissue? Is that mechanism known?
Well, in a simplified way, what we're thinking is that you just have more fat circulating. That's what it seems like in our mice have. It just seems like the intestinal epithelium is absorbing more fat, and then they just have more circulating fat that is accumulating in the liver and in the adipose tissue. Now, that's definitely a simplified version. I think it could be definitely cool to further characterize if there are other things happening that may be contributing to this fat accumulation. Yeah.
Terrific. Are there any more questions for Mariana? So one last from me, quick one. I think it's been described that PPAR gamma has different effects in the immune system, whether it's male or female. So this seems to have a gender difference. Do you find your effect to be different on the combination of antibiotics and diet based on gender? And is that depending on PPAR gamma?
So actually it happens in females too. Females at that point, they don't see the weight gain, but we see the accumulation in fat. And the females PPAR gamma knockouts also have this increase in fat accumulation. So it seems like it's happening in both males and females in our hands.
Great. Okay. So thank you so much. And boy, we've had quite a ride today. I'm really, first of all, grateful to all our speakers, and I just want to summarize very quickly what we've learned and what I've learned today. I think we've gone through a fantastic lineup, starting with Iran Sigal's description of the mega project of the Phenome project, which he's conducting. Doing bacterial genome-wide association analysis in multiple phenotypes, which looks like is going to be a very, very illuminating study. We then heard from Lisa Meyer on her work on the systemic mapping of drug microbe interactions and the interesting concept of antidotes as a concept to selectively protect gut microbes under antibiotic treatment. We then heard from Elizabeth Johnson, a fantastic talk on also drugging the microbiome, and particularly using dietary lipids as modulators of host microbiome interactions. She talked about the effect of milk lipids. So that was illuminating as well. We then heard from Sloan Devlin on the bacterial bile acid metabolites and how they affect host physiology. That was really a terrific talk, illuminating the aspect of gut bacteria, how secondary bile acids are produced and how they can be modulated by the microbiome. Then in the second part of the morning talks, we heard about from Ines Thiel on a terrific platform that she calls Agora 1 and 2, in which she's reconstructing whole genome scale metabolic networks. Then we have from Paul Blanley, in which in his talk how he's using this innovative microfluidics techniques to explore microbial interactions and environmental dependencies. We also heard from Ophelia Venturelli, also a tour de force in bioengineering, where she's using really state-of-the-art techniques and reagents to analyze molecules that are produced by the gut microbiota and how they shape host health and the ecological dynamics. We also heard from Breck Dorikop on bacteriophages as antibacterial therapeutics, and also from David Pride, also looking at phages and how they can be used to identify the individuals he showed that bacteriophages are shared among cohabitants in a house, similarly to how the microbiome is shared, but also what are the implications for the coevolution of phages and bacteria from the microbiome, and what their implications are for health and disease. And finally, we heard from Mariana, her unpublished work on how intestinal epithelial interactions and the microbiota during early life can promote obesity, but she focused more on this unpublished data set on the antibiotics and diet, how may act through PPAR gamma to produce higher fat content in the blood and lead to obesity. So overall, I think this was a really terrific day of learning and of sharing. I really want to thank all the guest speakers for terrific talks and their commitment to making this program really successful. I want to thank also my co-chair, Peter. I want to thank also the ETS support, Dorothy Wang. I want to thank last but not least, Christine, Kaylee, without whom nothing of what you witnessed today could have happened. They really put this together. And with this, I would like to draw this symposium to a close. And for those who are in person, please join us on outside. There will be some refreshments and some food. And thank you so much, and keep in touch for the next one. Bye-bye.